tell you when. All right, welcome back. All right, we just had a quick little little opportunist break there just to get some audio issues. Welcome back to our 164th VRPAC meeting. Dr. Monto, you want to pick up where we left off?
Well, I finally got on again, so yes, it's my pleasure to introduce Dr. Adam McNeil, who is from the Division of Viral Diseases at the Center for Disease Control and Prevention, who will be talking to us about a very important topic, epidemiology of COVID-19 variants.
Hi, good
Dr.
morning.
McNeil.
Hi, good morning. I'm Adam McNeil. So I'm with the Division of Viral Diseases at CDC, and I'm a representative on the Epidemiology Task Force as part of the COVID-19 response at CDC. So I wanted to start first by giving a brief overview of our current global situation with SARS-CoV-2. Currently, there's been over 110 million confirmed cases of SARS-CoV-2 with almost 2.5 million deaths. The bulk of the burden has been in the Americas and the European regions of the world. And we are seeing positive trends in the right direction in terms of declining cases, although I would note that these spikes have occurred at various times in various regions. So I think we remain relatively concerned about the current epidemiologic situation. So I'm going to talk about the various variants of concern. I want to briefly talk about what constitutes or what are the criteria for defining variants, including variants of interest and variants of concern. Currently, various organizations are developing standardized definitions. This includes WHO. From the United States government standpoint, we have a definition that's currently being reviewed as part of interagency activities, and we hope to have that finalized soon. But in general, regardless of the formal definition, there are some key criteria that generally meet consensus for helping define particularly a variant of concern. And this concludes at least one of the following: evidence of immune escape, either due to vaccine or natural infection, evidence of convergent evolution, impact of the variant on diagnostics, impact of the variant on therapeutics, evidence of increased transmissibility, or evidence of increased disease severity. For this presentation, I'm going to focus on the epidemiology of three specific variants, which currently have general consensus as being characterized as variants of concern. These variants are the B.1.1.7 variant, which was first identified in the United Kingdom and likely emerged in September of 2020, the B.1.351 variant, which was identified first in South Africa around October of 2020, and the P.1 variant, which was first associated with Brazil and Japan and identified in January of 2021. Notably, all three of these variants have two distinct amino acid changes, N501Y and D614G. Importantly, the emergence of the same amino acid changes in all three of these variants is suggestive of convergent evolution, in other words, suggestive of a potential selective advantage. In addition, the B.1.351 and the P.1 variants have a notable amino acid change of E484K. And this amino acid change has been very much associated in vitro with very strong evidence of reduced neutralization to previous infection as well as vaccination. In addition, I want to note one characteristic of the B.1.1.7 variant that's relevant to some of the current data we have. This contains a deletion at amino acid 69 and 70 of the spike gene, which impacts the S gene on multiple diagnostic PCR assays, which results in what has been referred to as an SG target failure, or SGTF. Due to this distinct pattern, it's allowed as an effective tool for screening or potentially enriching for identification of B117 using PCR assays. So since transitioning specifically to B117, since being identified in November 2020, B117 has rapidly been broadly identified across the globe. Notably, there's a high density and a high overall number of cases of infection of this variant in Western Europe, particularly in the United Kingdom. Although I would caution we look at this distribution that in many parts of the world, actually coverage with sequencing is relatively limited. So we have to realize this probably does not represent the full distribution of B117. Relatedly, if you look on the table on the right, you can see that in many countries in Western Europe, B117 has actually become the predominant virus in circulation. So as mentioned, B117 was first detected in England of 2020 and likely actually emerged in the southeast part of the country in September of 2020. The United Kingdom has used the S gene target failure PCR pattern as a way to actively monitor spread of the variant. And we observed in late 2020 a very rapid expansion of this variant throughout the United Kingdom. Based on various modeling studies, it has been estimated that the reproductive number of the B117 variant is approximately 1.5 times higher. Or in other words, this data suggests that B117 is about 1.5 times higher in terms of transmissibility in comparison to the previous dominant variant. I also want to briefly touch on the evidence suggesting potential increased severity of disease associated with B117. This data is pulled from a previously this evidence is pulled from previously unpublished data that was reviewed by the New and Emerging Viruses Threat Advisory Group in February 2021. And it was based on a composite analysis of 22 different related analyses or studies. And the data used a combination of either B117 confirmed variants based on sequencing or data using the S gene target failure marker. Based on review of all the evidence, it was concluded that there was evidence from these analyses of multiple different data sets that have infection with B117 that's associated with an increased risk of hospitalization and death compared to infection with non-variant of concern viruses. Results, if you look at this preliminary report, varied, but some outcomes were statistically significant, with ratios of hospitalization and death up to 1.7 times higher for this variant as opposed to baseline viruses. However, it should be noted that one of the key conclusions of this report is the absolute risk of death per infection remains low even with B117. Moving on, I want to talk a little bit about the global distribution of the B1351 variant. This variant was first identified in South Africa in October of 2020, and South Africa continues to have the highest case counts. Although, like I mentioned previously, I caveat this by the fact that many countries currently have limited sequencing coverage. You can see that in addition to South Africa, multiple countries in the southern part of the African continent have had cases identified. Similarly, there are a number of cases that have been identified in Western Europe, and small number of cases have been reported from the Americas, Asia, as well as Australia. Looking at this variant, specifically using one case study, a recent MNWR report documented the potential epidemic spread of B1351 in Zambia. This figure demonstrates the trend in the overall number of confirmed COVID-19 cases in Zambia. Starting in December, as you can see, a large spike in overall case numbers was noted within the country. Sequencing performed on a selection of diagnostic specimens from mid-December identified a high proportion of the B1351 variant, with 22 of 23 specimens being this variant. Notably, these specimens were obtained from 4 different provinces, indicated likely broad or wide distribution of this variant throughout the country. Similarly, prior to December, no B1351 cases were identified among 245 specimens that were previously sequenced within the country. Moving on to looking at the global distribution of the P1 variant. This virus was first identified among travelers from Brazil in January of 2021, and Brazil really remains the epicenter of transmission, which I'll discuss more in the next slide. Additionally, this variant has been identified in North and South America, Western Europe, and there are a small number of cases that have been documented elsewhere. So I'd like to turn over to the Manaus region of Brazil, which presents interesting and I would say quite concerning situation. There was a wide, largely unmitigated outbreak of COVID-19 that occurred within the Manaus region of Brazil in mid-2020. And you can see on the figure A, on the upper half of this, the large peak in excess mortality that occurred within this region of Brazil in 2020. A study used blood donor serology to estimate the actual seroprevalence of SARS-CoV-2 in Manaus, an estimated seroprevalence in October of 2020 to be around 76%. Notably, even with waning immunity, one would expect that this highest seroprevalence would likely have put this region in the position to be able to establish herd immunity. However, a second large peak in hospitalizations and excess mortality began being documented in January 2021, as you can see on the far part of figure A. P1 variant was detected in circulation in Manaus in January 12, 2021, and as shown in the previous slide, this really represents kind of the epicenter where now numerous instances of P1 have been detected in this region. Well, waning immunity from the previous large outbreak may partly be contributing to this overall second spike. I think it's important to point out that largely this data is probably suggestive of a certain degree of antigenic escape associated with P1. I would note that a number of further investigations are currently going on to better understand the situation within Manaus. I want to turn over to the SARS-CoV-2 situation in the United States now. As of earlier this week, we were hitting around 70,000 new cases being reported on a daily basis. This is down from a peak of over 300,000 being reported on a daily basis, which occurred in December and January of last year and earlier this year. In total, almost 28 million cases of SARS-CoV-2 have been reported within the United States. So as you can see from the curve on the right side of the slide, we are moving in the right direction with certainly a strong downward trend in the number of cases. However, I would caution we are certainly not out of the woods yet, and we need to continue our focus on mitigation measures and trying to stop the current outbreak. So as I get into talking about some of the approaches we're taking towards genomic epidemiology within the United States, I want to talk about how we're thinking about this in terms of key objectives and approaches. This can broadly be grouped into 3 categories. First of all, using genomic epidemiology for situational awareness or surveillance. So this would be to understand the prevalence and spread of variants and potentially use this for broader public health decisions. I would mention that for surveillance and situational awareness, this does require widespread sampling of representative specimens for sequencing, and the overall number of specimens is largely dependent on the burden of infection. Second is using genomic epidemiology to allow for novel variant detection. The main focus of this is to identify the presence of novel variants for further investigation. And as I'll talk about in a later slide, this is more focused on using a relatively fixed sample size within a defined population for detection of variants. Finally, we are using genomic epidemiology for focused studies. This includes trying to better understand the transmission, clinical outcomes, and vaccine effectiveness associated with variants. And for these studies, it requires extensive sampling and sequencing within a targeted population. So I do also want to touch on some of the inherent challenges of using genomic epidemiology to characterize SARS-CoV-2. First of all, in our current situation, we have to acknowledge that only a small proportion of viruses are being sequenced. And as I'll touch on, we are really rapidly scaling this up, but the reality is even if we can get to extremely high numbers with current incidents, we may only be sequencing 5 to 10% of specimens. So the reality is that we can have a certain degree of evidence, but we may not ever know the full situation in terms of what is going on with the virus. Second, there is an inherent time lag between sample collection and sequencing results. And while we are trying to push this time lag down, it does remain a significant challenge. So thus far, sequencing is not a rapid diagnostic tool. It has limited current utility from a clinical standpoint for clinical monitoring. And it currently represents a challenge for informing immediate public health action. Oftentimes, by the time we are ever able to actually confirm a variant, it may be late in terms of the opportunity for conducting contact tracing. I'd also like to note that we have not yet demonstrated sequencing as an effective containment strategy, and this is just evidenced by the fact that we have seen broad global spread of these 3 SARS-CoV-2 variants that I've discussed. Finally, sequencing by itself has limitations in terms of predicting epidemiologic outcomes. So key to mention that sequencing needs to be linked with supporting virologic and immunologic studies, as well as clinical and epidemiologic data. And acquiring clinical and epidemiologic data does take substantial sample size numbers and often takes a decent amount of time to fully characterize. So going on and looking at what sample sizes are needed to actually detect variants of concern. So this approach was adapted from the influenza virologic surveillance right-size roadmap, which has really been our long-term approach for estimating sample sizes for influenza surveillance. So this represents a disease-agnostic sample size calculator. And I would So as prevalence goes up, the number of sequences needed to detect a variant decreases. So we are currently taking a number of different approaches to evaluate genomic epidemiology of SARS-CoV-2. First is really kind of our backbone surveillance program, which we call National SARS-CoV-2 Strain Surveillance, or NS3, which represents a random selection of specimens submitted by public health laboratories for sequencing at CDC. In addition, we're taking further efforts to scale up sequence numbers by partnering with commercial diagnostic laboratories, conducting focused epidemiologic studies, developing contracts and partnerships with state and local health departments and universities, and finally by supporting the VIRS Consortium, which represents a consortium of over 160 partners that are working to standardize metadata and ensure that there is a large number of SARS-CoV-2 sequences available in the public space. So going on and looking at sequences in the public repository. In this slide, I'm showing numbers of sequences from specimens in the U.S. currently available in public repositories. And you can see in the orange line the number of sequences from the United States that have currently been submitted to GISAID, which is currently around 100,000. And we do hope that this will rapidly continue to increase as we are scaling up our sequencing efforts within the United States. Further, I want to note that we have a number of pre-existing protocols and study platforms that we are currently adapting to try to better understand epidemiologic and clinical characteristics of the viral variants. And this is, being able to use these platforms is somewhat dependent on the prevalence of a variant. So to be able to pull out clinical characteristics of a variant with very low prevalence is challenging. But as we continue to see increasing proportions of all cases that are represented due to variants, we anticipate having further statistical power to be able to tease out various epidemiologic characteristics. Furthermore, I would note that we are conducting surveillance and investigation of vaccine breakthroughs to understand the actual impact that viral variants have on occurrences of vaccine breakthrough. So looking at actual current numbers of SARS-CoV-2 variant cases detected in the United States, currently there are approximately 1,600 reported cases of B117, 22 cases of B1351, and 5 cases of P1. And you can see from these maps the current distribution of these cases. I would note that due to sequence coverage, as well as looking at the distribution, that these variants are probably much more widespread throughout the country. And I think we have to assume, in the absence of other information, that these variants probably could exist throughout the entire United States. So in order to better understand the potential impact of B117, assuming increased transmissibility of this variant, we developed mathematical models to look at the dynamics of viral transmission. In this slide, 2 scenarios are shown. In the figure on the left, a baseline reproductive number of the dominant virus is 1.1 is plotted. And in the right, we use the scenario of a reproductive number of the baseline variant or baseline virus as 0.9. The incidence of disease is shown on the y-axis. In both scenarios, B117, as represented in light purple, eventually becomes the dominant virus in late to mid-March. While these are theoretical models, even a lower reproductive number, which may be closer to the current epidemiologic situation, B117 does eventually result in an overall uptick in case counts. So in these figures, we have used the same modeling assumptions as the previous slide. However, vaccine introduction is additionally added to the model. As with the previous scenario, B117 becomes the dominant virus in mid to late March. However, through scale-up of vaccination, the actual trajectory of case count is substantially blunted. I would similarly like to briefly pull in some empiric data from a combination of academic partners and a commercial diagnostic laboratory that looked at early introduction and spread of B117 and specimens being submitted through their diagnostic network. Based on their evidence, they noted that B117 likely arrived in the United States in November of 2020, and multiple introductions occurred. Geographically, B117, based on current data, is widespread and confirmed in 44 states. This data from early February estimated prevalence around 1 to 2%. However, when the actual trajectory of the increasing prevalence was plotted through January and early February, it did appear that the virus was at an exponential growth phase if you look at the proportion of cases due to B117. So this is also supportive of the notion that B117 is on a trajectory to potentially become the dominant variant within the United States. I want to move on and look at the overall burden of infections within the United States so we can think about the impact that viral variants may have on this. We have started conducting routine seroprevalence surveys using commercial diagnostic specimens starting in around June or July of this year, which we use to generate seroprevalence estimates every 2 weeks for all states within the United States. So in this slide, I'm showing our seroprevalence estimates from December of 2020. And you can see from this slide that many states are starting to approach close to 25% seroprevalence. I would note that seroprevalence may underestimate the overall burden of infection because of potential waning in immunity. So we have similar used probabilistic models to try to account for under-detection and under-reporting of infections and estimate the overall burden of infection. Through December of 2020, based on these models, it's been estimated that over 83 million infections have occurred within the United States, with 70 million estimated symptomatic illnesses and 4.1 million hospitalizations. So if you look at this number, 83 million infections, it would land on approximately 25% of the U.S. population previously being infected with SARS-CoV-2 by December of 2020. So how do these estimates stand with regard to herd immunity? So shown in this slide is a figure generated by Omer et al. Using empiric data to estimate herd immunity requirement of SARS-CoV-2. So based on estimates of the reproductive number, approximately 60% population immunity is necessary to establish herd immunity. So based on our current estimates, which I showed in the previous slides, through around December 2020, we're certainly nowhere close to having herd immunity. Would note since this time, obviously, vaccination has started, and hopefully this is moving us closer to filling the herd immunity gap. However, thinking about the potential impact of variants on viral transmission and population immunity, currently we know that the U.S. population, the majority of the U.S. population is not immune to SARS-CoV-2. And variants may affect, may cause this proportion of the population that is not immune to increase. Waning immunity has potential to continue to contribute to this pool of individuals who may be susceptible to infection or disease. Increased transmissibility of a viral variant would require higher proportions of the population to be immune to establish herd immunity, and decreased effectiveness of a vaccine to protect against infection by a variant virus could result in prolonged or continuous transmission of SARS-CoV-2. So if I wrap this up, I want to talk about some key public health messages to stress. First of all, we know that current mitigation strategies work, and they work against variant viruses. So this includes masking, social distancing, hand washing, quarantine, and public health policies. Variants demonstrate the need to further push these mitigation measures. Current epidemiologic data is moving in the right or downward direction. However, potential of increased transmissibility means that adherence to these mitigation measures needs to be higher in order to maintain this downward trend in cases. Additionally, I will stress the importance of vaccination and monitoring the impact of vaccination. Vaccination provides general protection for the population against SARS-CoV-2. The impact of viral variants on vaccine effectiveness is still being characterized, but even with decreased effectiveness, vaccination still may provide partial protection against variants. And this underscores the need for robust epidemiologic and virologic surveillance system to determine if vaccine updates are needed. So in conclusion, 3 variants of concern have currently been identified. As SARS-CoV-2 continues to evolve, we have to figure inherently additional variants will likely emerge. And this underscores the importance of genomic surveillance. Data suggests that variants may have increased transmissibility, increased severity, or the ability to evade immune responses from previous viral infections. Epidemiology indicates broad global spread of these variants, and containment of variants has thus far been unsuccessful. And finally, this underscores the importance of currently well-characterized mitigation measures. This includes use of well-fitted masks, hand hygiene, social distancing, avoiding crowded or poorly ventilated indoor spaces. And finally, focusing again on ensuring we scale up vaccination to all those who are eligible to receive the vaccine. Thanks, and I would be glad to answer any questions you have.
Thank you, Dr. McNeil. Very important presentation for the rest of our discussion. Dr. Rubin.
Thanks, Dr. Yeah, that was very interesting. When it comes to specifically to the vaccines and their efficacy, the concern, of course, is pre-existing mutations that increase efficacy and perhaps the appearance of new mutations in the vaccinated population because of the new selective pressures. And it seems to find those, you'd have to be systematically sampling the escape mutants, and that means rather intensively, and have a representative sample of the population to compare that with. So the I wonder about that about both those pieces. Are we systematically sampling escape mutants, and the 3,000 say sequences we're getting a week of that we're getting right now? Are they in any way? Do we know they're representative?
Yeah. Thank you, Dr. Rubin. I think first touching on this representative piece. So the underlying goal with the NS3, so the 3,000 specimens, is to be broadly representative. As we're standing this up, the proportion of specimens that we're receiving from state and jurisdictional labs is proportional to their representative population size. And we are requesting that these states and jurisdictions try as best as they can to submit random specimens. I would add on the commercial laboratory front, as we are scaling up the number of sequences being generated by commercial labs. Early on, a lot of the sequencing was focused on the S-drop pattern and trying to identify B117. We have shifted this to have our contract focus more on having large commercial laboratories doing random specimen sequencing. So we actually hope to be able to get relatively large numbers to have a representative set of specimens. But, you know, it does remain an ongoing challenge, and, you know, I think inherently we can never hope to be perfectly representative, but I think we are getting relatively close to what we need to be able to have a representative denominator and actually understand what the virus is doing in terms of background circulation. Regarding systematically looking at viral breakthrough, I would note a couple things. Both CDC, but largely more in the interagency space. We are actively working to combination of acquire and culture variants of concern as they arise, as well as collect and characterize immunological breakthrough using from previously vaccinated individuals. In addition, we have a currently established protocol, and we are currently conducting more passive, but from a somewhat active standpoint, also trying to acquire and characterize instances of vaccine breakthrough. And hopefully by evaluating those, both looking at the viral sequence as well as hopefully being able to acquire serologic samples from some individuals are encouraging vaccine breakthrough will continue to better characterize these instances.
Okay, thank you. Dr. Gantz.
Thank you so much. You started, this has been a really helpful, informative, thoughtful, and very important discussion. So thank you for bringing this to the forefront. You started to allude to some of the efforts that you're doing to understand breakthrough, and I was very glad to hear that you're actually trying to get some samples, some blood samples from the vaccine. This is a group of people, obviously, that is very well characterized, and so it's an opportunity. You talked about serologic. Can you just discuss a little bit more about the immunity studies that you're going to undergo in terms of how you're looking at this? Because I think what we're starting to see is some discordance in the humoral and T-cell immunity in some of these people. And so to understand that a little bit better would be nice to know. How that pattern is being evaluated, particularly as it pertains to these variants.
Yeah. Thanks. Great question. And I am going to caveat this because I am not working on the laboratory end. But, you know, from a broad standpoint, certainly a number of in vitro studies have focused on the serologic component and looking at correlates of breakthrough from infection. From a T-cell standpoint, you know, I know this is an area that's been discussed. Obviously, characterizing T-cell responses and even acquiring specimens from a relevant number of individuals is more challenging from a serologic standpoint. But I know that that is an area that there's also interest. I think we're looking at this from a broad perspective. I would use probably the closest analogy analogy is how we continually characterize influenza and the dynamics between that virus as it's circulating and the vaccine. And I would note this is a combination of CDC as well as a number of other organizations in the interagency space, including NIH, FDA, DOD.
Dr. Meisner.
Thank you. I'll add my compliments for a very clear, helpful presentation. I would like to ask you a little bit about serologic correlates of immunity and how that's going to be impacted by these variants. It would be nice if a serologic correlate of immunity could be established so that large efficacy trials, which are so expensive and so time-consuming, weren't necessary. And particularly thinking about children. If we had a serologic correlate of immunity, that might make it easier to evaluate vaccine usefulness in the pediatric age group. But it seems to me it's going to be hard to establish a serologic correlate of immunity if these variants continue to emerge, because the threshold of immunity will probably vary depending on the vaccine and depending on the variant that's circulating. So perhaps you could comment on that.
Sure, thanks. Great question. You know, I think the issues with the variants underscores that exact concern. You know, I think we've seen, and I realize those on this call know the clinical trial data better than I do, but you know, we've seen, particularly with B1351, that there's a lot of evidence that the vaccine does not provide as high levels of serologic protection. So as these viruses are evolving, it may be a moving target in terms of what the serologic correlate of protection is. You know, going back to the E484K amino acid change, you know, I think that was the one. It's been, I would say, there's been a logical scientific progression where around December, January, there was antigenic mapping, and this was, in essence, the amino acid that showed the highest potential for immune evasion. And then I think we've seen analogously evidence of, as this mutation has been present in both B1351 and P1, there's similar evidence. So I think there is a certain alignment between the in vitro studies and what we're seeing in vivo, but it is probably going to be a moving target. I think that's going to be one of maybe the fundamental questions as this outbreak progresses is how is this virus going to behave? Are we going to need annual updates like influenza? Will we need annual updates every 5 years, or will we have broad enough protection to be able to use, in essence, a steady state vaccine?
Thank you.
Thank you, Dr. Meisner, and thank you, Dr. McNeil. I'm going to have to close the question period right now. This is a topic which we are going to be returning to in our open discussion this afternoon. So please, if possible, stay around so we can follow up with additional questions at that point. Now I'd like to move to another issue, post-marketing surveillance from currently authorized COVID-19 vaccines. This is going to help us decide on what is working and what is not working. And we have a double-barrel presentation, first from CDC, Dr. Shimabukuro from CDC, who is Deputy Director of Immunization Safety Office, and then from FDA, Dr. Stephen Anderson, who is the director of the Office of Biostatistics and Epidemiology, and we'll have the two presentations in sequence and the question period following. Please go ahead.
Thank you. This is Tom Shimabukuro. Can you hear me okay?
We can.
All right. Good morning. Today I'm going to do an update on vSafe, one of our CDC's safety monitoring systems, then a vaccine adverse event reporting system update, a vaccine safety data link update, and then I'm going to spend some time focusing on COVID-19 vaccine safety in pregnancy. Starting off with vSafe. So just to remind folks, vSafe is our smartphone-based text and web survey monitoring system that CDC stood up just for COVID-19 vaccines, the COVID-19 vaccination program. It involves health check-ins that occur daily, the first week after vaccination, and then weekly, and then through 6 weeks, then 3, 6, and 12 months, and the process starts again when a person gets a second dose. It's a voluntary self-enrollment program. If on any health check-in, a registrant reports that they receive medical care, we will follow up through our call center with VAERS and take a VAERS report. The questionnaires also allow for identification of pregnant women, and we also have a pregnancy registry team that conducts follow-up for enrollment into the pregnancy registry. So as of February 16th, that's the analytic period that, for this presentation. There are roughly 55 million individuals who had received one or more doses of COVID vaccines in the United States. And we had about 3.9 million registrants in vSafe that had completed at least one health check-in. And that included just over 30,000 individuals that self-reported that they were pregnant on a vSafe health check-in. So this table is from an MMWR that was recently published, and I apologize if this is a little bit small, but the analysis that I want to draw your attention to is really looking at dose 1 versus dose 1 for the two vaccines, the Pfizer-BioNTech vaccine and the Moderna vaccine. And specifically, we're looking at reactogenicity, which is collected during week 1 following vaccination, of dose 1 day 1 of Pfizer versus dose 1 day 1 of Moderna. And you can see that the reactogenicity profiles are very similar. Injection site pain is commonly reported, and then systemic reactions are also commonly reported as well. Confirming what was observed in the clinical trials, these mRNA vaccines are reactogenic, and the reactogenicity profiles of the two vaccines for dose 1, day 1 look very similar. Then at the time of this analysis for the Pfizer-BioNTech vaccine, we had some dose 2 data. We didn't for Moderna because of the timing of the rollout and the longer period between doses. But for the Pfizer vaccine, we had information on dose 1, day 1 compared to dose 2, day 1. And if you look at the comparisons for the systemic reactions like fatigue, headache, myalgia, chills, fever, joint pain, nausea, there's substantially more self-reported reactogenicity symptoms for dose 2 compared to dose 1, up to 3 to 4-fold higher in some cases. And that's not unexpected. That was observed in the clinical trials. So the vSafe reactogenicity really kind of confirms the safety profile of these vaccines and confirms that it was similar to what was observed in the pre-authorization clinical trials. Moving on to VAERS. VAERS is our spontaneous reporting or passive surveillance system that's co-managed by CDC and FDA. VAERS is a national system. Basically, anyone who's eligible for a vaccine is in the covered population. It can rapidly detect safety signals and can detect rare adverse events. The main limitation of VAERS is that it is not designed to assess causality. It accepts all reports from anyone, regardless of the plausibility of the vaccine causing the event, or the clinical seriousness of the event. It's a hypothesis-generating system to identify potential safety concerns or signals that can be studied in more robust data systems. So at the time of the data cutoff for this analysis, which was February 16th, we had just over 100,000 reports to VAERS, of which 94% were non-serious and 6% were serious. Serious uses the regulatory definition. So here's two tables showing the most commonly reported adverse events to VAERS for the Pfizer-BioNTech vaccine on the left and the Moderna on the right. You can see that systemic reactions and local reactions are the most commonly reported adverse events. But importantly, there were no empirical Bayesian data mining alerts detected for any adverse event COVID-19 vaccine pairs as of the last data mining run that FDA performed on February 18th. I just want to draw your attention to a fairly recent publication which updated some of the anaphylaxis reporting rates for the vaccines. This is a little small. I've highlighted a statistic on the bottom there showing that the most current reporting rates we have for anaphylaxis were 4.7 per million doses administered for the Pfizer-BioNTech vaccine and 2.5 per million doses administered for the Moderna vaccine. So I think the take-home message here is that these are rare events, and anaphylaxis, although clinically serious, is treatable, and there is CDC guidance available on identifying, managing, and being prepared at vaccination locations to handle anaphylaxis when it occurs. Moving on to the vaccine safety data link. It's a collaboration between CDC and 9 participating integrated healthcare organizations. It has electronic health record and administrative data on a covered population of roughly 12 million persons per year, and it also has rapid access to charts to review to confirm cases if need be. As of through February 13th, there had been approximately 630,000 doses, dose 1 doses of any COVID-19 vaccine administered in VSD, and about 200,000 dose 2 doses. We do something called rapid cycle analysis in VSD. These are basically weekly analyses of the data as the data accumulate, and I'm showing this slide mainly as a reference slide. This shows all of the pre-specified outcomes for VSD rapid cycle analysis. These are outcomes that we've identified in advance and we are monitoring. The analysis I'm showing here is an unvaccinated concurrent comparator analysis. That's basically comparing vaccinated individuals with unvaccinated individuals for these adverse events, and they're matched on certain characteristics, vaccinated and unvaccinated individuals. The preliminary results of the unvaccinated concurrent comparator analysis after any dose of an mRNA vaccine showed no statistically significant increased risk detected for any of these pre-specified outcomes. So what I'm showing here is a different kind of analysis. This is a sequential vaccinated concurrent comparator analysis. This is comparing vaccinated individuals and looking at events in the risk interval versus events in control interval. And I'm only showing outcomes for which there are events in the risk window. So if you don't see an outcome on here compared to the previous slide, that means there was just no event in the risk interval. And the preliminary results of the sequential vaccinated concurrent comparator analysis was that there were no statistical signals detected. So next steps for VSD RCA, we're going to do a dose-specific analysis, product-specific analysis in 2 risk intervals, 1 to 21 and 1 to 42 days, and a historical comparator analysis that's expected to start in the latter half of March. Moving on to pregnancy. The VSafe pregnancy, VSafe participants who self-report pregnancy are actively contacted and enrolled. The outcomes of interest include fetal demise, pregnancy complications, maternal intensive care unit admission, adverse birth outcomes, neonatal death, and infant hospitalizations, and major birth defects. So we have currently enrolled just over 1,800 individuals in the V-Safe pregnancy registry. Moving on to VAERS data. As of the 16th, there were 154 reports to VAERS, and there were the median maternal age in these reports was 33, median gestational age 13. Just over half of these reports involve vaccination in the first trimester, and you can see the vaccines there below. Of these 154 reports, most of these, and in fact 73%, were non-pregnancy-specific adverse events that you would expect, like headache, fatigue, chills, local reactions. Of the 42 pregnancy or neonatal specific conditions, most were spontaneous abortion or miscarriage. I just want to point out that the frequency of spontaneous abortion and miscarriage is actually quite common, 10 to 20% based on age. So there are other maternal vaccination safety activities, which I'm not going to cover in detail, but they include studies and surveillance activities in VSD and in the Clinical Immunization Safety Assessment Project. So to sum things up, as of February 16th, just over 55 million doses had been administered in the United States. The reactogenicity profiles of the mRNA vaccines in V-Safe are consistent with what was observed in the clinical trials. Systemic and local reactions are most commonly reported to VAERS. Anaphylaxis does occur, though rarely, and there's no safety signals for any serious adverse events. And there are no safety concerns identified among VSD rapid cycle analysis outcomes. Most reports to VAERS among pregnant women involved non-pregnancy specific adverse events. Miscarriage is most frequently reported, the most frequently reported pregnancy-specific adverse event, but the number was not concerning considering expected background rate. And safety monitoring in pregnant women is ongoing or planned in V-Safe, VSD, and CISA. Thank you. That concludes my presentation.
All right, I'm going to just give an update on FDA's monitoring of the COVID-19 vaccine safety and effectiveness work that we're doing. So Tom has presented information from this slide. Just wanted to note the 55 million doses administered. We're using the same set of numbers for our presentation. Since Tom has already cut, this is a slide of our current vaccine surveillance programs, and since Tom has already covered the passive surveillance and VAERS systems, I'm not going to cover that. I'm really going to focus on the bottom portion of the slide, the active surveillance component, talking about our CMS work, our work on background rates, our work on study protocols, and then talk about next steps. So just launching into the FDA CMS work, we're going to talk about our rapid cycle analysis, specifically talking about the approach, which is to monitor 20 or more outcomes, which Tom sort of mentioned in his presentation. FDA is identifying the elements of our RCA we're identifying and using 15 possible adverse events of special interest. I wanted to then talk a bit about getting sufficient counts in the CMS database to start the analysis, the background rates, and then talk about where we are as far as conducting the RCA analysis and CMS data. So these are the adverse events of interest that FDA is focusing on. I just wanted to mention that these have been studied in other vaccines, but they haven't been associated with the COVID-19 vaccine in pre-authorization studies. So some of the things you've seen in previous studies, like Guillain-Barré syndrome with Bell's palsy, et cetera. I just wanted to talk about the rarity of these events, 1 in 10,000, 1 in 100,000 or less, and so they're rare. and so need large databases in many cases in order to get significant power in order to analyze these with millions, usually with millions of patients. The CMS data, just to remind people, I think I've shown this slide at the previous presentation to this committee. The data covers nearly all of the 55 million elderly U.S. beneficiaries over 65 years of age in the United States. All right, so here's the counts that we're getting. So in the CMS Medicare data, we've got 4.8 million total doses. And just to sort of orient you on the graphics, the far left, this bar is the total number. The middle is the first dose. The right bar in the set is the second dose. And as you can see, for Pfizer, there's 2.8 million doses and 2 million doses for the Moderna vaccine. And the time period given for this analysis is listed in the bottom left corner. All right, so as far as those vaccine counts, what does the age distribution look like? So this is just a check, and so you can see that most of the counts end up in the age 65 years of age and older. Medicare does cover younger populations, persons with disabilities, and kidney disease, and so so you'll see some of those represented in the lower age populations in this study. All right, so background rate analyses. So background rates, why are we talking about them right now? So the background rates for AESIs provide us with information on expected rates or an estimate of baseline for comparison to see if there's an elevated risk of for an AESI, then we need to compare that to some sort of baseline historical number. So for our analysis, I think it's important to mention that COVID-19 vaccines are new, so we lack kind of that historical information that you might have for a vaccine like influenza, where we have years and years of data where we can understand background rates for these AESIs. But what this work does in the third bullet point is really requires us to go ahead and generate new background rates for the selection of comparator groups. Just going down to the bottom bullet point, so we've actually generated information on background rates for 4 different populations. And I'm sorry this type is small, but what we've done is we've looked at, in the CMS data, at the population overall, the age 65 years of age in that group. And then we've also looked at influenza vaccinees, age 65 years of age and older, and gotten the rates for those specific AESIs in that population. The time periods may be important too, so we've looked at that. So we looked at the pre-COVID period, and that means for the years 2017, 2018, and 2019, and that should give us information prior to COVID. And then we're also looking at the peri-COVID or sort of the COVID period, which we think is another important consideration. And why are we doing that? Well, so in the first bullet point here, the COVID-19 pandemic may have impacted healthcare utilization, and that's been published in the literature that for like infections like, for conditions like heart attacks or AMI, reports dropped initially by 50% in the first few months of the pandemic, and then initially rose back to sort of more pre-pandemic levels. So that's of interest to us if we're trying to evaluate what's the relevant background rate to use for the analysis. So we've assessed background rates in these populations, and what I wanted to show you was some of the results, and these are just sort of representative. So the top 3 lines on this left graph represents 2017, 2018, and 2019. And then this is for colonoscopies, and as you can see, and you might have expected this, was during the first few months of the pandemic, colonoscopies dropped by almost 75 or 80%. But then by about September of the fall, they started to climb back up to rates that were equivalent to prior to the pandemic. Now, if we look at something like stroke in the graph on the right, you can see again those 3 lines in blue, orange, and gray sort of show you that the rates for 2017 through 2019. And by comparison, the line in yellow, you can see there's a dip. Again, similar to what colonoscopies, but not so, not such a strong relation, not such a strong drop for hemorrhagic strokes. And then what you can see is that it's really popped back up again by late summer to sort of the pre-COVID rates. So what does that mean for us? So for a majority of the ASIs in our analysis, we selected the pre-COVID-19 background rates among persons 65 years of age and older in our CMS data. For a few of these ASIs, like less than 5 of those AESIs, where the rates didn't recover, then we used the pre-COVID-19 levels for our background rates. And then just to note, the background rates are also been standardized against for age and other demographic characteristics. So I just wanted to talk about where we are as far as our RCA analysis. The FDA's done this foundational work on the counts monitoring and then the background rates, and that work is complete. So we just sort of started to on the preliminary runs. Those have been underway for the last few days. And we're evaluating our early results. So our expectation is we'll have results probably end of the week, early next week, into the weekend. And then our expectation is to be able to sort of fine-tune our analyses and then conduct runs every 1 to 2 weeks to kind of achieve our goal of near real-time monitoring of safety for these 15 outcomes. I just wanted to mention that we have these surveillance study protocols that sort of support the work that we've done. The first bullet points we've done looking at the background rates, as I've mentioned. The second protocol that we've done is the active monitoring, which is really just describing the rapid cycle analysis protocol used. Each of those protocols were posted on the bestinitiative.org website. And so we received comments for about 10 days on each of those. And then I think we posted the latest version or the latest update on February 10th and 11th. So you can go to that website and see those. We're also developing additional protocols for inferential studies. So if we do signal in these RCAs, we need to follow up with epidemiological studies, and the goal would be to these protocols have those protocols listed that we would be using to follow up on any signals identified in the rapid cycle analysis or any signals identified in bears like the anaphylaxis and others. There's also testing where we're evaluating the performance of testing codes as well, and that protocol is under development as well. And I just wanted to say sort of at the bottom of this point number 2 is we're developing a vaccine effectiveness study, and obviously many considerations there. For instance, effectiveness by vaccine, comparative effectiveness by dose, duration between doses, duration of protection, and a number of other factors for considering and developing those protocols. And then just to get to the next steps, I wanted to say that we talked about persons 65 years of age and older. What we need to do then is focus on 18 to 64-year-old persons. And we're going to be conducting additional rapid cycle analyses. And if you go down to the bullet points in the middle here, we're going to be doing these analyses in Optum and then CVS Healthagen. And that should cover approximately, I think, 25 to 30 million persons overall is our hope. And then to add other claims databases as soon as we can bring those online. And our plan is we would start analyses in late March for the 2 databases listed, followed by in subsequent weeks with other databases. And again, I think Tom mentioned what they're doing by brand. They're analyzing their ASIs by brands, by risk intervals, doses, et cetera. I just wanted to mention quality assurance. So I think the power of the government approach is that we're able to compare our results that we get with the FDA systems with those of the CDC's VSD but also the Veterans Administration is running similar analyses. So we can do this cross-comparison, see what we're getting. If one gets a signal in their system that others don't get, we can do validation of those outcomes and verify signals that are identified. And then I just wanted to acknowledge that this work is a huge amount of work by a number of colleagues and a lot of individuals in CBER, as well as our CDC colleagues, our CMS colleagues, VA colleagues, and many other FDA partners. And with that, I will stop. So thank you very much.
Thank you both. We have time for a few questions. Dr. Marasco.
Dr. Marasco, please make sure you're unmuted. All right, let's go to the next.
Well,
let's go to the next one. Yeah.
Dr. Pergam.
Thanks for those presentations from both of you. Dr. Anderson, I wanted to ask you specifically about how you're tracking and how the FDA, and maybe this is for you, Tom, as well, how you guys are monitoring people who are getting a second dose that is not the same as the primary dose of the vaccines they've received. And I think that's particularly important when we get into the Janssen vaccine that might potentially be a different mechanism. So do you guys have a plan or a system of how you're tracking that information and potential differential effects of that situation?
Sure. I'll ask Rich Borshi also to get on this, get online as well. So Rich, do you want to explain for first dose, second dose?
Absolutely. So thanks for that question. We have been working with the American Medical Association and CMS since the start of 2020 to make sure we had appropriate codes in place to deal with the kinds of situations that you were discussing. So each of the vaccines that receive an EUA is going to have a specific CPT code, and not just for the vaccine, but also for the administration and the administration by dose. So currently for the Pfizer-BioNTech and the Moderna vaccine, there are specific CTP codes for first dose of Pfizer, second dose of Pfizer, first dose of Moderna, second dose of Moderna, and the same thing's going to happen for other vaccines that are approved. So when the claims are submitted, we will know exactly which vaccine and which dose people have received. And so that will allow us to look at some of the safety and effectiveness questions that you outlined with regard to mixing different vaccines. I'll pause there and see if there's any other follow-up questions.
Dr. Gantz.
Hi, thank you for this very important presentations and actually very satisfying in terms of how you're looking at these. My question for you is one way that we actually look at the vaccine in terms of the safety is actually not only to look at the baseline rate in a population of specific outcomes that you listed very nicely, but is actually to look at those outcomes also from the disease itself, so those with active infection. And I'm wondering if those analyses are going on, because if you can show differences also in rates of some of these outcomes as comparison to disease, that's an important marker of how we're sparing disease and how safe these are in relation to disease entities.
So one of the things that we have already done.
Tom, did you want to start?
Or I think I'm going to have. I was going to ask to repeat the question,
Rich,
unless you want to go
Rich,
ahead,
did
Steve,
you wanna,
if you want.
Rich, did you want to take that one as well? Because
oh. Yeah, so I can start that discussion. We have published a first paper, and we have others in the line characterizing the natural history of the COVID-19, essentially trying to look at the risk factors that put people at greater risk for serious outcomes. And so we're already related outcomes, and then that will give us a position where we can look at vaccinated versus unvaccinated and some of these serious COVID-19 outcomes. So that, in fact, a big part of the reason we did the natural history study was to make sure that so Dr. Gans, is that addressing your question, or is there another dimension to it?
Yeah, no, thank you. I just, I'm encouraged to hear that those studies are in place and will go forward, because I think that's a really important.
Oh,
go ahead, Tom.
I was just going to mention, and I know that the CDC and FDA and the VA apply certain, this is a little bit different than the question you asked, but apply certain exclusion criteria based on a history of natural disease, so the natural disease doesn't confound our safety monitoring.
And the only other thing that I was going to add is...
Okay, I think I'm going to have to break in. We're running over, and this is a very interesting discussion. And again, it's going to come up later, especially concerning how we can rapidly figure out vaccine effectiveness against some of the variants prevalent. So it's time for a break. And since we've been going so well for this period of time, let's try to reconvene past the hour. That's Eastern at 11:20, which will be 10 minutes, 10-minute break. So break time right now.
All right, Dr. Monto, I will get us. Oh, hold on.
15 minutes.
And you like how much time, Dr. Monto? For
10 minutes.
10 minutes, okay. Johnson, Janssen, anybody who is going to possibly be speaking during the sponsor section, raise your hand quick so we can confirm your audio. I want to make sure every one of you is connected that needs to be. So far we have Greg. I'm going to go down the line. Greg? Dr. Greg?
Yep, I'm on. Thanks, Mike.
Perfect. Dr. Makaya?
outbreak may partly
All right, welcome back from break. We are now going to go into our sponsor section. So I'd like to bring in our chair, Dr. Monto.
Welcome back from our extended break. Next is our very important sponsor presentation. We're going to hear from the team at Janssen, and I'd like to turn this over to Dr. Johan van Hof, who will introduce the other members in turn of the Janssen team. Dr. van Hof.
Thank you. Thank you, Mr. Chairman, and good morning. My name is Johan van Hof. I'm the head of Janssen's Vaccines Research and Development Organization. I want to thank the committee and FDA for the opportunity to present data from our development program as we seek emergency use authorization for our COVID-19 vaccine candidate. As we all know, while the FDA has authorized 2 vaccines for emergency use, there remains an urgent need for additional vaccine availability in order to vaccinate a majority of the US population, ensure protection against serious disease, and subsequently reduce the burden on the healthcare system. If authorized, Janssen's vaccine candidate would play a critical role in the global effort to fight COVID-19. S26CoV-2S was studied in a large phase 3 study. We enrolled more than 44,000 participants and conducted the study in multiple countries during the height of the pandemic. It offers substantial protection, especially against severe COVID-19, including hospitalization and death, irrespective of the variant. It is well tolerated and safe, and it is a single-dose regimen with storage and transportation conditions that are compatible within existing distribution channels. Specifically, Janssen's single-dose vaccine has demonstrated early onset of protection against symptomatic COVID-19. Particularly important in the context of an outbreak, we observed 85% vaccine efficacy against severe COVID-19 globally, including the United States, at least 28 days after vaccination. For this secondary endpoint, the effect was consistent across all geographic regions, including in South Africa, where 95% of the strains were an emergent variant of the B1351 lineage. Importantly, the primary analysis, there were no COVID-19 related hospitalizations in the vaccine group versus 16 in the placebo group. There were no COVID-19 related deaths in the vaccine group compared to 5 in the placebo group. In the United States, we have shown 72% vaccine efficacy at least 28 days post-vaccination. We enrolled more than 19,000 participants in the US and paid particular attention to include a diverse population. Our primary endpoint was achieved with 66% vaccine efficacy for moderate to severe critical COVID-19 for the overall study population after day 28. And protection was observed as early as 2 weeks after vaccination. And we saw consistent efficacy across age, comorbidities, race, and ethnic subgroups. These results are particularly important when one considers when and where we studied our vaccine. Our trial was conducted under challenging epidemiological circumstances. Our study sites were located in areas where COVID-19 incidence was highest and where variants were emerging, including in South Africa, Brazil, and the United States. And still, the vaccine efficacy against severe critical COVID-19 was high. Based on sequencing of approximately 72% of the COVID-19 cases in our study, it is evident that prevalence of the new variants was close to 70% in Brazil and greater than 90% in South Africa. Of note, we did not observe the P1 lineage in our Brazil sites. Also, in line with what you will hear later, these efficacy rates are based on the total data set, including the non-centrally PCR-confirmed cases. During the last 2 months, we have all seen that it is critically important to manufacture and distribute vaccines quickly and efficiently. And Janssen's vaccine offers logistical and practical advantages to help simplify distribution and expand vaccine access. Each person receives a single injection of 0.5 ml. The application of a single-dose regimen offers the ability to vaccinate the population faster. Each vial includes 5 doses and no dilution is required. The vaccine can be stored for 3 months at normal refrigeration temperatures and has a 2-year shelf life when kept frozen. We have continuously improved our manufacturing and formulation processes and started last spring to prepare for large-scale manufacturing. Based on this, we expect to supply 100 million doses to the US government in the first half of 2021. And it can easily be shipped using existing supply chain infrastructure. In addition to these key features, it is important to note that in the context of a vaccine that would be administered to millions of people, it is reassuring to note that Janssen has substantial clinical experience with more than 193,000 people who have been exposed to our At26-based vaccine. These studies and programs were conducted across continents, including participants of various ages, races, and ethnicities. This also included vaccination of pregnant and breastfeeding women in our Ebola program. Our Ebola vaccine was licensed in Europe in July 2020 and is currently part of a mass vaccination program in Rwanda. Regular reviews of the safety database have shown overall good tolerability and safety. Local and systemic reactogenicity are in line with what is seen with licensed vaccines. And the database searches focused on adverse events of special interest did not reveal any safety signal. Let me provide an overview of the key studies in our comprehensive development program which led to the current emergency use application. We conducted numerous animal studies, including non-human primates, to study vaccine immunogenicity and efficacy. Our phase 1-8 study led to the dose selection for our phase 3 studies. Study 2001 is a phase 2 trial investigating a range of dosing regimens. Our ongoing pivotal phase 3 trial, COV 3001, is examining the efficacy, safety, and immunogenicity of a single-dose regimen. This is the data being submitted to support our application for emergency use. Today's presentation shares with you the first look of this ongoing study. Since our initial analysis, some additional information on cases observed has become available, which will help to address some of the questions those initial results could have triggered. We will also share this with you today. We also have a series of planned studies which are not part of the data package for EUA. Let's review those. We are evaluating the efficacy of the 2-dose regimen in study 3009. We will conduct several immunogenicity and safety studies in children from birth up to 17 years of age. The study in adolescents will open for enrollment soon. The start of a study in pregnant women is planned for end March, early April, and we also plan to begin studies in immunocompromised individuals in the third quarter of this year. In addition, Janssen plans several post-authorization observational studies to assess vaccine safety and effectiveness in the real world. This also includes development of a pregnancy exposure register. With this in background, let me present the outline for the rest of our presentation. Professor Hanneke Schuitenmaker will describe the vaccine design and immunogenicity data. Dr. Makaya Dwogi will review our efficacy and safety data. And Dr. Craig Holland of the Mayo Clinic will provide a benefit-risk assessment on granting emergency use authorization for Janssen's At26 COVID vaccine candidates. All external experts have been compensated for their time preparing for today's study. Let me now turn over the presentation to Professor Schuitenmaker.
Thank you, Dr. Van Hoof.
My name is Hanneke Schuitenmaker and I'm the Global Head of Viral Vaccine Discovery and Translational Medicine and the Viral Vaccines Disease Area Stronghold Leader at Janssen. I'm also a professor in virology at the Amsterdam University Medical Center. In this presentation, I will explain our established EdVac technology platform, of which Ed26 is at the core of the Janssen COVID-19 vaccine, and provide an overview of how we designed our COVID-19 vaccine candidate and its immunogenicity in non-clinical and clinical studies. By design, the Ed26 vector is replication incompetent. The E1 region, shown in blue, and the E3 region, shown in yellow, are important in the development of our vector vaccine. By deleting E1 from the adenovirus DNA genome, the virus irreversibly loses the ability to replicate in human cells. We have also deleted most of the E3 gene. This creates more room in the genome for a transgene, shown here in purple, that codes for the protein that triggers the desired immune response. For the production of the replication incompetent viral vector, we use the well-characterized Percy6 cell line that complements for the missing E1 gene. The vector can only replicate in an E1 complementing cell line and again cannot replicate in the human body. Of note, the Percy6 cell line grows in a medium free of animal components, and ultimately a vial of our Ed26 vaccine additionally only contains a buffer with commonly used ingredients in vaccines. There are no added adjuvants, antibiotics, or preservatives. We chose to target the immune response against the spike protein of SARS-CoV-2. This was based on knowledge gained from previous SARS-1 experience and literature that show that antibodies directed against the spike can neutralize the virus and that T cells against epitopes in the spike protein play a role in the protection against disease. Therefore, we evaluated multiple transgenes encoding different spike designs, allowing us to select the vaccine candidate with optimal stabilization, expression, immunogenicity, and non-clinical efficacy. The selected spike protein is membrane-bound and contains 2 proline mutations and a knocked-out furin cleavage site for optimal stability in its pre-fusion conformation. And so our lead candidate was selected based on these factors and also on its optimal manufacturability. Now let's take a look at how Ed26 CoV2S may work in the body. First, a single dose is injected into the muscle in order to deliver the transgene to a diversity of cells. The transgene encoded spike protein is then expressed on the surface of these cells. Innate immune responses triggered by the Ed26 vector provide the optimal microenvironment for the immune response against the spike protein. Attracted antigen-presenting cells pick up the spike protein and migrate to the lymph node, eliciting both humoral and cellular immune responses. The CD4 T helper cell responses are predominantly of a Th1 phenotype and stimulate B cells. Spike-specific antibodies are then produced by plasma cells. These antibodies have SARS-CoV-2 neutralizing activities and or Fc-mediated antiviral effector functions and play a key role in vaccine-elicited protective immunity. In addition, spike-specific CD8 T cell responses are triggered. CD8 T cells mature into cytotoxic effector cells with the ability to kill virus-infected cells. This is an important effector mechanism of vaccine-elicited antiviral immunity. Finally, FDA guidelines classify adenoviral vectors as non-integrating, meaning they do not have the propensity to modify the host genome. Let's turn to the non-clinical data. A single dose of Ed26 CoV2S gave full protection against SARS-CoV-2 challenge in non-human primates. We observed near complete protection against viral replication in the nose and full protection in the lung. This protective efficacy was durable for at least 6 months, and high-level protection against lung viremia was seen even after vaccination with a 4-fold lower dose in the face of lower antibody titers. In addition, a single dose also gave near complete protection against viral replication in the lungs of aged non-human primates. To complement our NHP studies, we tested our vaccine in Syrian golden hamsters, where it also demonstrated protective efficacy. And histopathology in animals with low-level breakthrough infection demonstrated no evidence of vaccine-associated enhanced disease. You can find more information on this in your briefing materials. Overall, these results satisfied the FDA guidance criteria to allow for progression to human clinical trials. Turning now to our phase 1/2a study. Study 1001 is a randomized, double-blind, placebo-controlled trial and was the first in human dosing of F26CoV2S. The study initially enrolled 2 groups of healthy adults ages 18 to 55 and healthy adults 65 years and older. We evaluated 2 dose levels, 5 times 10 to the 10th and 1 times 10 to the 11th virus particles, which were administered intramuscularly in either a 1-dose or a 2-dose regimen. As both dose levels demonstrated similar immunogenicity, I will focus on the single vaccination with the lower vaccine dose, as this regimen was selected for our first phase 3 study. The interim analysis was conducted at day 29, which was 28 days after administration of the first dose, and evaluated safety and immunogenicity. The study demonstrated similar and durable humoral immunogenicity in adults 18 to 55 years of age and those 65 years and older with a single dose of F26CoV2S. A neutralizing antibody response was observed in 96% of participants by day 29 in both age groups. This response was maintained up to at least day 85, suggesting good durability of humoral immunity. There are additional features of F26CoV2S elicited humoral immunity. In line with our platform data, F26CoV2S elicited antibodies also demonstrated non-neutralizing Fc-tail-mediated functionalities. These could have important antiviral effects or function, including against emerging SARS-CoV-2 variants. Indeed, in contrast to neutralizing antibodies, these antibodies are not limited to epitopes in the receptor binding site or N-terminal domain, where most of the amino acid substitutions in new SARS-CoV-2 lineages seem to occur. A common concern is that natural immunity against F26 may interfere with the immunogenicity of F26-based vaccines. The phase 3 study demonstrated that F26CoV2S immunogenicity was similar across the highest enrolling countries. This included Brazil, with 33% of participants having F26 neutralizing antibodies at baseline, South Africa with 69%, and the US, where F26 seroprevalence at baseline was below 2%. The results are in line with our experience across our F26-based vaccines. Turning now to cellular immunogenicity. A single dose of F26CoV2S elicited CD4 and CD8 T-cell responses, which could be detected by day 15. Spike-specific CD4 responses were detected in 71% of younger adults and in 69% of the older adults, and were predominantly of a T helper 1 phenotype. CD8 T-cell responses were detectable in 46% of the younger adults and 27% of the older adults on day 15, further increasing to 61% and 51% by day 29. Both CD4 and CD8 T cells had a memory phenotype, were present, which is obviously important for anamnestic responses and durability of protective immunity. In summary, following a single dose of F26CoV2S, Neutralizing antibody titers were elicited in the vast majority of adults, independent of age. Titers were detected as early as 14 days post-vaccination, which increased in the following weeks and persisted at least up to day 85. This was irrespective of the vaccine dose used. Both CD4 and CD8 T-cell responses were observed. The Th1 dominance of the CD4 T-cells in combination with the neutralizing antibody response minimizes the risk for vaccine-associated enhanced disease. In addition, both vaccine dose levels had a favorable safety profile with no safety concerns. However, the lower dose had a more favorable reactogenicity profile. Based on these results, the lower vaccine dose of 5 × 10⁻⁻ virus particles of F26CoV2X was selected for the phase 3 study called 3001. Thank you. Dr. Duogi will now present the efficacy and safety data from our clinical trials.
Thank you, Professor Schoutemacher. Good morning. My name is Makaya Duogi. I am the head of clinical development and medical affairs for vaccines at Janssen. This morning, I'll be presenting results from our phase 3 study, COV 3001. Our primary analysis shows that the study met its primary endpoint, demonstrating the ability of a single dose of Ad26-CoV2F to protect against moderate to severe COVID-19 in adults, and that it has an acceptable safety and reactogenicity profile. From here on, I will refer to our vaccine as Ad26. I'll start with the study design. 3001 is a randomized, double-blind, placebo-controlled phase 3 trial that's evaluating the efficacy, safety, and immunogenicity of a single dose of Ad26. We randomized participants 1-to-1 to receive a single injection of either vaccine or saline placebo. Randomization was stratified by site, age, and absence or presence of comorbidities. The first part of the study consisted of a safety run-in phase. Stage 1A enrolled 2,000 adults up to age 59 without comorbidities. Following a planned safety review, enrollment progressed to Stage 1B to include participants up to 59 years with and without comorbidities. Stage 2A began in parallel to Stage 1A and enrolled 2,000 participants 60 years and above without comorbidities. Note that Stage 2A took a little bit longer to recruit. After a planned safety review where the DSMB identified no safety concerns, we progressed to Stage 2B to include those 60 years and above, both with and without comorbidities. Since those 60 years and older are at higher risk for severe COVID-19, we targeted at least 30% of the total population to be in this age range. The co-primary endpoints are vaccine efficacy to prevent moderate to severe critical COVID-19 with onset at least 14 days post-vaccination and also with onset at least 28 days after vaccination. The primary hypothesis is that the lower limit of the 95% confidence interval of the point estimate is greater than 30%. This had to be met for both co-primary endpoints in order to declare success. The case definition for moderate COVID-19 was a positive RT-PCR or molecular confirmation of SARS-CoV-2 infection, and at any time during the observation period, at least one new or worsening sign or symptom, as listed in the panel on the left, or at least 2 new or worsening signs or symptoms suggestive of COVID-19 on the right. For example, if a participant had a sore throat and a headache, this was sufficient to be considered moderate. Here's the case definition for severe critical COVID-19. For this, participants must have a positive RT-PCR or molecular test confirmation of SARS-CoV-2 infection and any one of these listed signs at any time during the observation period. We have a blinded clinical severity adjudication committee to evaluate severe and all moderate COVID-19 cases with at least 3 signs or symptoms. Classification of a case as severe critical by this committee is considered definitive. Next, I'll move to the disposition and efficacy results. Beginning with disposition, a total of 44,325 participants were randomized, of which 43,783 received an injection of either Ad26 or placebo, making up the full analysis set. This is the safety population. The per protocol population is the primary efficacy population and includes all participants who were seronegative at the time of injection and had no other major protocol deviations judged to possibly affect the vaccine efficacy. Participants were also excluded when they had a positive PCR test at the day of injection. The per protocol at-risk set includes all participants in the per protocol population, but excludes those with a positive PCR test between injection through day 14 or day 28. This slide shows the overall demographics of the study population at baseline. Looking between the two groups, we see no relevant differences. Efforts were in place to reach pre-groups with good representation in terms of age, race, ethnicity, and sex. And I'll just flag for you here the numbers of participants who are 60 years and older across both groups. Enrolling significant numbers of participants in this age range was important for evaluating vaccine efficacy. And as I noted, we had a target of 30% for the whole study population. Of note, nearly 20% of the full analysis set were frontline essential workers or healthcare professionals. And here's the same table, but looking at baseline demographics for participants from the US. Again, we see no relevant differences between groups. We see a similar proportion of participants 60 and older in the US as we saw globally. And importantly, participants are representative of the US population in terms of race and ethnicity, reflective of the diversity of the population. Understanding that people who are most at risk for developing severe COVID-19 are those with pre-existing comorbidities, it was important to enroll participants with key risk factors. Approximately 41% of those enrolled had at least one comorbidity. This table shows the most common comorbidities across the global population, with at least 2% in either group. As you can see, participants who are the most vulnerable for developing COVID-19 related symptoms are well represented in the study overall. And then looking at the US subgroup specifically, the percentages are similar to the overall trial population and are distributed between the vaccine and placebo groups. The full table is presented in the briefing document. Let's now turn to the co-primary endpoint results. Study 3001 met both of its co-primary endpoints, accruing 464 primary endpoint cases between September and January. The co-primary endpoint analysis includes all PCR positive cases that were confirmed at a central laboratory. The vaccine efficacy against moderate and severe critical COVID-19 is approximately 67% after day 14 and 66% after day 28. The lower limit of the 95% confidence interval was well above the FDA requirement of 30%. Over 99% of accrued cases fell within the defined moderate to severe COVID-19. Therefore, the primary endpoint is representative of nearly all symptomatic COVID-19 cases. When looking at the co-primary endpoints in the US, we see that vaccine efficacy against moderate to severe COVID-19 is 74% after day 14 and 72% after day 28. Note that the onset of protection was apparent as early as day 14 after vaccination. Now here we see the cumulative incidence curves for moderate to severe COVID-19 begin to separate between the AD26 and placebo groups at around 14 days after vaccination. The circles represent the severe cases for each group. Due to the high COVID-19 incidence rate during the conduct of the study and the time it took for central lab confirmation of local PCR tests, not all cases could be confirmed by the central laboratory at the time of the primary analysis. As a result, there are two data sets. The first data set consists of PCR positive COVID-19 cases confirmed at a central laboratory and is used in the co-primary and secondary efficacy analyses. However, we anticipated that subgroup analyses would require a larger data set in order to be able to draw conclusions. And we wanted to look at all confirmed COVID-19 cases that required hospitalization or led to death. So we pre-specified that a second data set could be used, which included all COVID-19 cases with a positive PCR by any FDA-approved test, regardless of central confirmation. To justify the use of the second data set, we looked at agreement of the PCR positive results from the central lab versus all other sources. We found that these data sets had high concordance. We also wanted to test for consistency between the data sets with regards to vaccine efficacy, and found that there was less than a 1% difference between them for the co-primary endpoint criteria. This is true for both time points. Therefore, the use of the larger data set was justified. All secondary endpoint results can be found in your briefing material. In the interest of time, I'll just walk through our key secondary endpoints. Overall, vaccine efficacy against confirmed severe COVID-19 occurring after day 14 was high at approximately 77% and increased to about 85% after day 14. There were 14 versus 60 cases of severe COVID-19 occurring after day 14 in the AD26 group versus placebo, respectively, and 5 versus 34 cases occurring after day 28 in the AD26 group versus placebo. When we look at the cumulative incidence of molecularly confirmed severe COVID-19 cases, we notice 3 important characteristics. Vaccine efficacy starts at day 7 or earlier, indicating early onset of protection. Protections continue to increase over time, and this demonstrates the potential for longevity. Notice that after day 48, there were no more cases in the vaccine group versus 13 in the placebo group. Analyses do show that vaccine efficacy against severe COVID-19 increases over time, as denoted here by the black line. Looking at day 56, we see an estimated increase to approximately 92%, with no indication of waning thereafter. The gray area reflects the uncertainty around this estimate, which increases beyond the median follow-up of 58 days due to the small numbers of participants at those time points. Data also demonstrate substantial effects on the prevention of COVID-19 related hospitalization, with 93% vaccine efficacy for all positive PCR cases from any source after day 14 post-vaccination. And the protective effect is even more pronounced after day 28, where we see 100% vaccine efficacy. 19 deaths occurred in the study, 3 in the AD26 group and 16 in the placebo group. 5 of the deaths in the placebo group were confirmed to be COVID-19 associated and were reported prior to the January 22nd cutoff. None of the deaths in the vaccine group were COVID-19 related. A 6th COVID-19 related death occurred in a participant in the placebo group who had a positive SARS-CoV-2 RT-PCR test at baseline. As such, according to protocol, this participant is not included in the COVID-19 related deaths. We did look at deaths after the primary analysis and identified 6 more between the initial cutoff date and February 5th, 2 in the vaccine group and 4 in the placebo. One of the deaths in the placebo group was confirmed to be COVID-19 associated, compared to none in the vaccine group. All COVID-19 associated deaths occurred in South Africa. We also evaluated the effect of AD26 against asymptomatic, undetected SARS-CoV-2 infection to gain insight into the potential benefits of vaccination. Based on SARS-CoV-2 and IgG testing alone, 18 participants seroconverted in the AD26 group compared to 50 in the placebo group, resulting in a vaccine efficacy of 66%. A sensitivity analysis was performed to remove all participants with a positive serology result at day 71 who had symptoms between day 1 and day 71. 10 seroconversions occurred in the AD26 group and 37 in the placebo. These findings are preliminary, and while further follow-up is needed to assess whether or not they are confirmed in the larger data set, they do suggest a protective effect of the vaccine on asymptomatic SARS-CoV-2 infection. We also performed additional analyses where we looked at vaccine efficacy by key demographics and by country. And here we see that vaccine efficacy against moderate to severe critical COVID-19 is consistently observed across all pre-specified groups. This analysis includes breakdowns by age, 18 to 59, and 60 and older, by comorbidity, by sex, and by the largest racial and ethnic groups. We do note that a lower vaccine efficacy point estimate with wide confidence intervals was observed for the subgroup of participants 60 years and older that had comorbidities compared with the overall population. At the same time, our assessment is aligned with that of the FDA that there is an observed trend of increasing efficacy with narrower confidence intervals as numbers of cases in the analysis increase. And therefore, we are also aligned with the FDA in that those 60 years and older with comorbidities are similar to any other subgroup and would benefit from vaccination with Ad26. Across 3 key countries, vaccine efficacy against moderate to severe COVID-19 was consistently high. The majority of participants were enrolled in the US, Brazil, and South Africa. They were also the countries with the highest incidence of moderate to severe COVID-19 cases in our study. The forest plot illustrates that Ad26 consistently protected against moderate to severe COVID-19. Vaccine efficacy after day 28 ranged from 64% to 72% across 3 countries. The vaccine efficacy against severe COVID-19 was consistently high across these countries as well. Looking at South Africa, for example, protection after day 28 was about 82%. Of note, 95% of the sequence samples in South Africa were associated with the new, highly transmissible variant from the B.1.351 lineage. Taking a closer look at South Africa, we found that there were no hospitalizations in the Ad26 group and 6 in the placebo group. There were no deaths in the Ad26 group and 5 deaths in the placebo group. These findings suggest that Ad26 is efficacious against this newly emerging and rapidly spreading strain. To summarize efficacy, a single dose of Ad26 offers substantial protection against COVID-19, including against hospitalization and death. Across all countries, Study 3001 generated high-quality, robust data at a time when the incidence of SARS-CoV-2 was increasing and new, highly transmissible variants were emerging. Janssen's vaccine demonstrated 85% vaccine efficacy against severe disease with an early onset of protection as early as 7 days after vaccination. Importantly, for the primary analysis, there were no COVID-19 related hospitalizations in the vaccine group versus 16 in the placebo group, and there were no COVID-related deaths in the vaccine group compared to 5 in the placebo group. For protection against moderate to severe disease, there was 66% vaccine efficacy across all countries, and the onset of efficacy here was evident as early as day 14, increasing through day 56, especially against severe disease. In the United States, where study participants reflected the diversity of the overall U.S. population, vaccine efficacy against moderate to severe COVID-19 was 72%. Protection against all symptomatic disease was consistent with the primary endpoint, and the high levels of protection were consistent across subgroups, countries, and regions in particular areas of high incidence of circulating variants. Now let's turn to safety. A single dose of Ad26 was demonstrated to have an acceptable safety and reactogenicity profile. As expected, results were consistent with the tolerability and safety of our other adenovirus-based vaccines. We also have plans in place for continued safety monitoring following emergency use authorization. Now, reorienting us to the study population, serious adverse events, medically attended adverse events leading to discontinuation and death were collected on the 43,783 participants who make up the full analysis set. In addition, all solicited and unsolicited adverse events were collected in a subset of individuals, referred to as a safety subset. We conducted our primary analysis after meeting the FDA guidelines for reaching a median follow-up of at least 2 months. The median follow-up after vaccination was 58 days. More than half the participants in the full analysis set had at least 2 months of follow-up. In the safety subset, nearly all participants completed the post-vaccination period of day 1 to 29. Now I'll turn to the solicited adverse events collected during the 7-day post-vaccination period. Solicited local adverse events, stratified here by grade and age, were transient, and more than 99% were grade 1 and grade 2 in severity, and all resolved within 2 to 3 days after injection. The most frequently reported local adverse event was injection site pain. The frequency of grade 3 adverse events was very low overall, with a higher incidence in the AD26 group compared to the placebo group. For participants in the AD26 group, the most frequently reported grade 3 event was injection site pain at 0.4%, and there were no grade 4 AEs reported. In the AD26 group, the frequency of solicited local AEs was similar between those who were seropositive for SARS-CoV-2 at baseline, pre-vaccination, and those. The frequency of solicited systemic AEs was higher in the AD26 group compared to placebo during the 7-day post-vaccination period. Most were transient and had a median duration of 1 to 2 days after vaccination with AD26. The most frequently reported symptoms in the AD26 group were fatigue, headache, and myalgia. Approximately 98% of solicited systemic AEs were grade 1 or grade 2 in severity. Grade 3 events were infrequent and reported in about 2% of participants in the AD26 group. There were no grade 4 events reported. Fever was reported in 9% of participants in the AD26 group, with 0.3% being grade 3 among those 18 to 59 years of age, and 0.1 in those 60 years and older. All fevers were reported to have started on the day of vaccination or the day after and had a median duration of 1 day. You'll note that the reactogenicity profile overall is milder in the older age group compared to the younger age group. Now turning to unsolicited adverse events, as you can see, rates for unsolicited adverse events, serious and non-serious, in the safety subset 28 days after immunization were balanced between arms. Rates were also balanced in the full analysis set for medically attended adverse events, any SAE, any SAE due to non-COVID related AEs, or any death. SAEs, medically attended AEs, and deaths were numerically higher in the placebo group. The imbalance between AD26 and the placebo group is mostly driven by the number of AEs associated with SARS-CoV-2 infection. None of the deaths in the AD26 group or placebo group were considered causally related to the vaccine. In addition, there was no evidence of vaccine-associated enhanced respiratory disease following vaccination with AD26. The clinical findings confirm the non-clinical observations that the theoretical risk of vaccine-associated enhanced respiratory disease with AD26 is low. Data demonstrate clear Th1 dominant immune responses. Breakthrough infections in those receiving vaccine were milder than those in the placebo group. The DSMB continuously monitored all cases of COVID-19 for patterns that are suggestive of vaccine-associated enhanced respiratory disease, and none were found. Janssen analyzed the occurrence of various adverse events of interest by regulatory agencies and medical and scientific organizations such as the Brighton Collaboration. These events include neuroinflammatory and others, including those where there's a numerical imbalance with numbers higher in the AD26 group. I'll provide more details of hypersensitivity reactions as well as arterial and venous thromboembolic events in my following slides. For the other events of convulsions, tinnitus, peripheral neuropathy, Guillain-Barré syndrome, and Bell's palsy, no causal relationship with the Janssen COVID-19 vaccine could be determined. The assessment of causality was confounded by the presence of underlying medical conditions frequently present in individuals with these adverse events of interest. And these events are included for further monitoring in our comprehensive pharmacovigilance plan. Hypersensitivity adverse events were reported in 0.4% or 77 vaccinated individuals and 0.3% or 65 individuals who received placebo. A given participant may have reported more than one sign or symptom. As shown in the table, non-serious dermatologic manifestations, particularly rash and urticaria, were the most common hypersensitivity AEs reported. Rash and urticaria, both localized and more rarely generalized, are considered likely related to vaccination. In the events reported as related by the investigator, the mean time to onset after vaccination was about 6 days, and the mean resolution time was 13 days. The vast majority of events were grade 1 or grade 2. There were 2 serious adverse events in the AD26 group and 1 in the placebo group. A single SAE of hypersensitivity was reported in 1 vaccine recipient with urticaria beginning 2 days following vaccination, and angioedema of the lips with no respiratory distress, which began 4 days following vaccination. The event was likely related to the vaccine. One SAE of angioedema occurred 23 days after vaccination and was considered unrelated by the investigator. Both participants recovered without sequelae. The results are similar to what we have observed with other Ad26 vaccines. As of February 22nd, no cases of anaphylaxis meeting the Brighton collaboration criteria had been reported in our vaccine clinical program. On Wednesday of this week, however, we received preliminary reports of 2 cases of severe allergic reaction, one of which was anaphylaxis from an ongoing open-label collaborative study in South Africa that has vaccinated approximately 40,000 healthcare workers to date. We will continue to closely monitor for these events as outlined in our pharmacovigilance plan. The overall incidence of thrombotic and thromboembolic events, arterial and venous, is similar across Ad26 and placebo groups. A numerical imbalance was observed for the venous thromboembolic events. Most of these participants had relevant underlying medical conditions as well as predisposing factors that may have contributed to the occurrence of these events, such as COVID-19 infection, prior history of DVT, new estrogen use, family history of DVT, prolonged air travel, or stopping anticoagulants. There's insufficient evidence to determine a causal relationship between these events and the Janssen COVID-19 vaccine. These events are included for further monitoring in the pharmacovigilance plan. In summary, the known and potential benefits of Ad26 outweigh the known and potential risks. Overall, safety data from the 43,783 participants in our phase 3 study demonstrate that a single dose of Ad26 has an acceptable safety and reactogenicity profile. Reactogenicity was demonstrated to be mild and transient in nature, and grade 3 events were rare. Most AEs were mild or moderate and generally resolved within 1 to 2 days post-vaccination. Adverse events of interest were thoroughly evaluated, and we will continue to monitor for these events in our comprehensive pharmacovigilance program. The safety profile is further supported by data from more than 193,000 individuals who have received at least 1 dose of Janssen's Ad26-based vaccines in our other clinical studies and programs. If authorized, Janssen will amend the 3001 study protocol to facilitate crossover of participants who receive placebo to receive 1 dose of the vaccine as fast as operationally possible. All participants will be encouraged to stay in the study for up to 2 years for ongoing assessment of efficacy, safety, and immunogenicity. The amendment will allow us the opportunity to continue collecting long-term data and assess the duration of protection and immunogenicity of a single dose, comparing the results of 2 groups vaccinated approximately 4 to 6 months apart. I'll now review our safety and effectiveness monitoring activities in the post-authorization period. Janssen has developed safety and effectiveness plans to complement and utilize the U.S. government and other established programs to monitor and quickly identify any potential safety signals. This plan includes surveillance of adverse events following immunizations, a pre-specified list of AEs of special interest, and other known concerns associated with vaccines in general. We plan to identify and assess any new safety signals by monitoring our own global safety database, along with reviewing external databases, including the FDA VAERS database. In addition, we will monitor long-term safety and effectiveness by conducting observational and active surveillance studies utilizing health insurance claims databases and electronic health records here in the U.S. and in Europe. For patients who opted in to have digitization of their records, they will be followed long-term for efficacy and safety. Perspective.
I've been a practicing internist for nearly 40 years, a PI of roughly 40 vaccine clinical trials, and exposed to hundreds more in my role as editor-in-chief of the journal Vaccine. Unfortunately, my experience also includes a front row seat to this fast-moving and deadly coronavirus. both as a researcher and a care provider. And so I'm very pleased to share my clinical perspective on the positive benefit-risk profile of Janssen's vaccine candidate and its role in protecting more Americans against COVID-19. As of today, COVID-19 continues to spread at alarming rates, and a large proportion of the U.S. population still needs access to safe and effective vaccines. In fact, we have periodically reached exponential phases of spread where the virus is no longer increasing on a linear scale, but is instead periodically spiking at a rapid rate. The consequence of this is that there are limited options to control the virus. In fact, there are only 3 ways the pandemic can be controlled. First, a hard lockdown with mandatory masking and social distancing. And we know this has largely been unpopular and less successful in the United States. Second, the virus mutates to be less transmissible. But in fact, more transmissible variants are already emerging and circulating in the U.S. and the world. Or third, the development of highly efficacious vaccines that are widely used. We need vaccines that are effective and well tolerated, and importantly, ones that are simple to deploy. Vaccines are our primary weapon in countering and controlling this threat. So let's turn now to Janssen's COVID-19 vaccine candidate and its one-dose regimen and what it could play in the urgent mass vaccination campaign needed now to fight this global pandemic. Here are some of the key factors. First, it's been studied in the largest COVID-19 vaccine trial to date in multiple countries, giving us more data to analyze and confidence in the results. Second, it is a replication incompetent vaccine, meaning that it has been engineered to express the spike protein and cannot propagate in the cells of a vaccinated individual. Third, it is a non-adjuvanted vaccine, so it does not use additional ingredients that would further increase local reactions such as redness or swelling, or systemic reactions such as fever and chills. Fourth, it's compatible within existing vaccine distribution channels. It can be stored for 3 months at normal refrigerator temperatures and has a 2-year shelf life when frozen. And last but certainly not least, the Janssen vaccine was specifically studied with a one-dose regimen. When the World Health Organization outlined its target product profile for a vaccine candidate, it identified a strong preference for a single-dose vaccine in an outbreak. And certainly this one-dose regimen offers important logistical and practical advantages for mass vaccination campaigns. It can lead to the ability to reach both individual and herd immunity more quickly. Essentially, it simplifies the process. People only have to make one appointment for their complete vaccination. A one-dose vaccination decreases the burden on the healthcare system and healthcare providers. And as such, this single-dose regimen also decreases health utilization costs. In addition to these factors, the data demonstrates strong efficacy that offers protection against COVID-19. The pivotal study met both co-primary endpoints, finding that AD26 is effective against symptomatic COVID-19. Significantly, the vaccine is highly effective in preventing severe COVID-19. The prevention of hospitalizations and deaths was a particularly important finding when you consider the burden this disease has placed on hospitals and healthcare workers. The findings regarding efficacy against newly emerging variants, such as the highly transmissible strain first identified in South Africa, are also important. Getting on top of these variants will be critical in our fight to control the virus. Notably, in the large phase 3 trial, if a participant who received the vaccine candidate did experience symptoms after infection, those breakthrough infections were milder, another welcome benefit both for individuals and the healthcare system. Beyond the protective effects, we also see that a single dose was demonstrated to be safe and well tolerated, and the sponsor has a comprehensive plan in place for ongoing monitoring. Janssen was very successful in enrolling a diverse study population, including older adults and those over the age of 60 who also had comorbidities. This is important, of course, because these are the individuals most at risk of progressing to severe COVID-19 with resultant higher rates of morbidity and mortality. The data reviewed today for this trial did not demonstrate safety concerns, including fever, in all of the assessed populations, and this includes older adults with comorbidities such as diabetes, hypertension, and obesity. In this trial, there were no severe allergic reactions, but as you have heard 2 days ago, the sponsor was made aware of a case of suspected anaphylaxis in a recently initiated trial with a current enrollment of over 40,000 vaccinated healthcare workers. Generally, hypersensitivity reactions following immunization were rare and non-serious. I fully support the sponsor's plan to amend the pivotal study and allow participants who received placebo to cross over to access the vaccine. This will allow continued safety monitoring, diminishing any reason to withdraw from the study, and give us longer-term data. And the sponsor's planned studies in special populations, including children and pregnant women, will provide important new data for our consideration. Finally, let's take a moment to consider a list of attributes that would be ideal for a COVID-19 vaccine, especially one authorized for emergency use authorization and to be used in mass immunization campaigns. We'd like to see an excellent safety profile and protective immunity, ideally with a single dose, and balanced immune responses. And we want to avoid vaccine-induced immunopathology. In terms of production and shipping, we need a vaccine that can be quickly mass-produced, that at normal refrigerator temperatures, and avoids the need for ultra-cold chain transport and can be stored long-term. Beyond this, we want to see a reasonable duration of immunity and efficacy. With the data on hand, we now see that the Janssen vaccine candidate checks nearly all the boxes. There are some longer-term items that will need to be further researched, but as discussed, we can expect answers to these important questions as part of the sponsor's ongoing investigation. In summary, COVID-19 continues to be a deadly pandemic, and we urgently need more vaccines under EUA to protect the millions of Americans who remain at risk. Today, we have seen clear and compelling evidence that the Janssen vaccine candidate is well tolerated, has an acceptable safety profile, and most importantly, is highly efficacious against COVID-19. To me, it is clear it meets the criteria for emergency use authorization. Thank you, and I will turn the microphone back to Dr. Van Hoof.
Thank you, Dr. Folland. And before we conclude, I just want to take a moment to say a few special thanks, certainly to our collaborators at U.S. Department of Health and Human Services, particularly FDA, CDC, and the National Institute of Allergy and Infectious Diseases, as well as the team at BARDA. A special thanks as well to all of the global trial sites and to the many trial participants. Our work would not have been possible without their involvement. Thank you. And we are now ready for questions.
I'd like to thank you all for a very clear presentation. I'd like to start off by asking specifically about the issue of the crossover, which you said was going to occur as quickly as possible, giving vaccine to the placebo recipients. This, as I take it, is a unblinding of the study. In other words, giving the vaccine only to the placebo recipients, not giving a placebo to the former vaccine or the vaccine recipients which would be a blinded crossover. Also, and I really don't think we ought to spend a whole lot of time on it, but I noticed in your briefing materials, you're also planning in your 3009, the two-dose study to give vaccine again to the placebo recipients, which will change that design completely. I don't think we want to spend much time on that point. It might come up in the discussion later on, but I do want clarification about that now, so we have that to keep in mind.
Thank you. Indeed, just to make sure that you understood it correctly, we are indeed proposing to do an open label crossover of the people who have received placebo, that they would receive a dose. This would happen in the study 3001 and also in study 3009. It would be subject of an amendment, and depending on the country, it might take some time for these amendments to be approved by the authorities. The thinking is that by crossing over the subjects, that we can keep subjects in the trial. We should not forget that we have really very intensively been selecting people that are at significant risk for COVID disease. And thus, there are some ethical challenges on keeping these people on placebo. There were quite some discussions in the past already around that topic. We have seen that people start in those countries where the products are approved, especially in the US, and that was part of the data that have been presented. We hope by offering the vaccine that we can keep people in the study. Although it's not the ideal design with a placebo group, we still would be able to compare relative efficacy between those people that were vaccinated a few months later than those people that were vaccinated initially. And that if their differences start to occur, that could be an indication of waning protection. For 3009, we are actually indeed offering a single dose of vaccine to the group that receives currently 2 doses of placebo. And so that means that that study at that moment is different in design, and at that moment you're comparing the 2-dose regimen with a single dose regimen.
And do you have enough power to show differences?
I would actually propose that I go over to our biostatistician who has been looking at those power calculations, and I would ask Dr. Bart Spiessens to take the floor. Dr. Spiessens?
Thank you, Dr. Van Hoof. So indeed, if you look at the 3001 study, when we do the placebo crossover, so we cross over the placebo participants, we do think that based on what we see in the Folman paper and simulations that we have done, that we do have sufficient power to make sure that we can detect waning if there would be waning of our vaccine. For the 3009 study, also there we will be comparing the 1 dose with the 2-dose vaccine, and also there we think we have enough power to make at least some comparisons if the incidence keeps being high as it is currently the case. Thank you.
Okay, thank you. Dr. Levy.
Hello, it's Ofer Levy with the Precision Vaccines Program. Can you see me?
No.
We hear you.
We hear you.
Okay, good. So I had a question for the sponsor regarding what is known with respect to innate immune activation by the AD26 vaccine vector. There's evidence in the literature that AD26 may engage pattern recognition receptors, potentially toll-like receptor 9 in the inflammasome, thereby inducing cytokines such as interferons. Is that known for this particular product? The other related query was that did any of the AD26 clinical trials, either for coronavirus or other indications, assess acute cytokine induction and its potential relationship to adaptive immune responses in the clinical responses? And finally, we note that there is some safety data for children from the Ebola program with this vector, and how will this position J&J for pediatric studies? Thank you.
Thank you. Actually, I will start with the last question, and then I would like to go to Dr. Zahn to comment on your questions on the innate immune responses. With regard to indeed, we do have extensive experience in pediatrics going down to the age of 4 months. We have specifically with our Ebola program done an extensive study going through different ages. We have observed that our immune responses are higher than in adults. There is a tendency to somewhat higher fever rate in the younger children, overall still very manageable. And so overall we feel that this platform experience encourages us to start fast with our pediatric program, and we are looking into, as I indicated already, we are looking into starting the vaccinating adolescents as of next week, and we hope to de-escalate in age over the next few months. With regard to the innate responses, Dr. Zhang, can you respond to this question?
Yes, thank you, Dr. Panov. I don't think you can see my camera. I guess it's not working at this moment, so excuse me for that. I'm Roland Zahn, and I'm the non-clinical lead for the viral vaccines program. And indeed, as you mentioned, for the AD26 vector, there have been multiple innate cytokine pattern recognition receptors described, like TLR9 and CGAS, STING pathways, as well as the inflammasome pathway. We have not studied this specifically for this vector. However, in our non-clinical safety studies, we have measured acute phase reaction after vaccine administration 24 hours later in the circulation. Thank you.
And what were the results? Thank you for that, Dr. Zahn. And what were the results of those studies?
So we saw CRP-induced relatively transiently 1 day after the immunization with AD26 at the 1 × 10^11 viral particle dose.
Okay,
Okay,
so
Dr.
Thank you. So although this vaccine doesn't have an adjuvant in it, it may be self-adjuvanted. Thank you.
Okay, Dr. Offit.
Yes, thank you, and thank you all for a very clear presentation. I'm trying to get a better understanding of kind of the strategy moving forward with this vaccine. Dr. Poland made an excellent case for all the advantages of a single-dose vaccine, but you're doing with that COVID-309 trial, you're doing a 2-dose trial, presumably because, as you showed in that phase 1/2A trial that was reported in the New England Journal of Medicine by Dr. Sadoff and others, that with that second dose, you had a sort of 2.6 to 2.9 increase in neutralizing antibodies, which may well confer more protection. If that's the case, move forward with this vaccine, and we find that with 2 doses we have a better clinical response. Does this then become a 2-dose trial, a 2-dose vaccine? Rather, in other words, for those who got, say, a single dose starting a couple weeks from now, then 6 or 8 months from now when we have the data for the 2-dose trial, are we asking them to come back for a second dose? I'm just trying to understand how you're positioning this.
There are The question is clear. When we started back in September and we had the data from our non-human primates model that actually did show that we had full protection in the lung with a single dose, that we also had protection in those monkeys even 4 months after vaccination, combined with the responses we had observed in humans, we decided to evaluate in parallel 2 vaccination schedules. First one is really testing the single-dose regimen and then the 2-dose regimen. Why did we choose a single-dose regimen? Because also based on all the discussions over the months that preceded, including guidance from WHO and others, it's clear that in a situation of an outbreak, in a raging epidemic, the big challenge is to get the epidemic under control, and that is where a single-dose regimen with rapid onset of protection is highly preferred. We do feel that the result of this study, where we did that it really has high efficacy against severe disease, specifically against hospitalization, and that with that, it actually fits that profile. An epidemic where you have mass vaccination programs, there's so much operational advantage in having a single-dose regimen, in addition to also being able to vaccinate more people with the same supplies, that we do feel that this regimen is really extremely well positioned for the use in outbreak situations. Now, indeed, there is remains to be seen what the benefits will be in terms of the differences versus the single-dose regimen. It can be indeed that the efficacy could be higher, specifically for moderate or for moderate. For severe hospitalization and death, it will be very difficult to be. So I do think that the judge is still out on what and how the data look and what to do with it. The other point is that, of course, even for a single-dose regimen or for the other vaccines, the big question mark still is how long does protection last and at what moment would it be needed. And so we do feel that also those data will help us to determine that this somewhat. But again, for the current situation with these emergency use, we do think that this requirement that the single-dose regimen has to fulfill.
No, I agree with that, and thank you for that answer. Just one quick follow-up, I might, Dr. Mato.
Very quick, because we got some deadlines ahead of us. Go ahead.
Sorry. You can see where the messaging is. Right? If you bring out a single-dose vaccine and say this is a single-dose vaccine, and then later find that something is better enough, I mean, clinically better enough to say that we recommend a second dose, you can see where that would be confusing to people, where they are thinking maybe I didn't get what I needed. But in any case, that's all. It's a messaging challenge is all. Thank you. Thanks, Dr. Mato.
You're welcome. Thank you.
Dr. Kurilla?
Thank you. Yeah, specifically in relationship to the seropositivity with regard to 26 that you highlighted, clearly looking across the different populations, there doesn't seem to be any gross impact on efficacy. I'm wondering if you were able to discern any differences in terms of some of the immunity can predict or not someone with a previously exposed to AD26 is just a natural have less of a risk that may wane quick or may respond differently forward in terms of the overall efficacy. For example, the broader spectrum with regard to some of the variants activities that you saw.
Right. That's a good question. So it's interference with pre-existing immunity. As Professor Schuitemaker, we have clearly seen in this that the immune responses were the cell of South Africa, the spectrum of people being seropositive at baseline. This is from our other AD26-based vaccines, where we have seen that there's really no significant interference with that pre-existing immunity against the vector. This seems to be a big difference with what has been reported for the adeno5 vectors. The big difference probably is related in the fact that these titers are in the people that are pre-immune are clearly lower than the titers that you see with AD5 seropositivity at baseline. But so overall, from this program, but it is in line with what we have seen with our other programs.
Thank you, Dr. Moore.
Thank you. I want to follow up on Dr. Carrillo's question. First, a very quick comment on unblinding, and I'm not picking on your vaccine because I've asked the same thing of the other vaccine makers. I'd really suggest that you try to do an NP-RT-PCR on all the participants before you unblind them, or at the time of unblinding, so that we have a better idea of virus shedding after vaccination, and potentially, if you have those samples, then you can later about variants. But my question and follow-up to Dr. Carrillo's question, which is really quite good, is that adenovirus latency and persistence is a black box. This, as you've pointed out, is a natural infection, and presumably some people will have prolonged infection and other people not with AD26 after exposure. Do you have any evidence or data on the persistence of the vaccine strain, or whether it's cleared from all vaccinees within days, weeks, months? Any data that would help us evaluate that?
Thank you for that question, and I'll go back to Dr. Zahn to discuss the biodistribution data that we know from there. I would like to reiterate, though, something which was already said before, that is the adeno 26 here is a non-replicating vector, so there's no multiplication in the vaccinee's body. So it's
now, let me just clarify that. It is not, it will not, it cannot, presumably from theoretically what we know, make infectious virions. But in terms of persisting as a latent either episome or pseudo-episome, I'm not certain that we can say anything about that. You haven't deleted, for instance, the N-terminal repeats or any other way that virus may use to persist long-term. So I caution about using that phrasing that it's not a dead virus.
I refer to Dr. Zahn to answer the details of your question. Dr. Zahn?
Yes, I'm Roland Zahn. Thank you for the question. Indeed, we have obviously looked into biodistribution of AD26. We have not done specific biodistribution studies with AD26-CoV2S, but with multiple other AD26 vaccine vectors. And we've seen that in rabbits, which we use as the main species for these distribution studies, that the vector DNA is mainly localized in the injection site and then distributed to the draining lymph nodes and a bit to the spleen. Here we have seen that the vector DNA is cleared from these tissues within 90 to 180 days. And that's a similar pattern as has been observed for other adenoviral vectors. So the vector seems to be cleared from the organism by natural mechanisms like division of cells or by immune mechanisms of infected cells. Thank you.
Okay, we're going to have to go on. We have a hard start for the open public hearing at 1:10 p.m. Eastern. So we are going to have to limit questions right now to, let's say, 2 more, and I'll call on Dr. McGinnis.
Thank you, Arnold. Can you hear me?
Yes.
I have a question, please, regarding the case definitions and the description of accompanying statistics. So in the briefing document, the sponsor does define under 7.1.1.5, they define moderate COVID-19, severe critical COVID-19, and mild COVID-19. Yet when we look at the analyses, we have these pooled, what I presume are pooled, and a description of something about protection against moderate to severe COVID-19. There is no case definition for moderate to severe COVID-19. So I want the clarification, please. These are ranked data, graded data. Is this a pooling of moderate and the severe pools to come up with this moderate to severe?
That is correct indeed.
Could you repeat that, please?
That is indeed correct. So moderate to severe is adding the moderate and severe together.
So it's pooled, so it's and, moderate and severe pooled together with an analysis run on that.
Yep.
Thank you.
Dr. Hildreth.
Okay. Yes, I had a question about the T-cell response to AD26. The participants all of them made strong antibody responses, neutralizing antibody responses. But I noticed that the response by CD4 positive T cells is only about 2/3 of them. How does that compare to your other AD26 vaccines that you developed?
Thank you. That question I'm going to refer to Professor Schuitenmaker. Professor Schuitenmaker?
Yes. In our other programs, we have seen similar, we call it good CD4 T cell responses. Also, if we compare for other vaccines, these are high responder rates. And also for our CD8 T cells, we see good responder rates also as compared to other vaccines. So does that address your question?
Well, I was asking the question because I think T cells are important for durable responses. And I know that some of your vaccines you say last for 2 years, so I wondered if those individuals have a higher T cell response than you see here.
Yes, and indeed, we see durability of our responses that indeed correlate with the good CD4 cell responses for the humoral immunity, but also we see prolonged durability of our CD8 T cell responses, which is really a feature of the platform, to your point. Yeah.
Thank you.
Okay,
Thank you.
One final question, I promise. Final from Dr. Kim before our very short lunch break.
So I have a question for Dr. Duogi. You know that one death in the AD26 group was excluded because the subject had tested positive for infection by PCR at the start of the study. How many other subjects in the study had positive COVID tests at the start of the study, and how were they distributed? That is, demographics and other information such.
Dr. Duogi?
Yeah, sorry. Can you hear me okay?
Yep.
Yes.
Yeah, so we had, that's true in the placebo group, we did have one PCR positive at baseline. We wanted to exclude those in our primary efficacy analysis. In terms of evidence, so we looked at seropositivity at baseline in all of our participants and then as well. And overall, we saw that about 9.6% were seropositive at study entry. And I'm just checking to see if we have more specific numbers on PCR. I don't see those here. Maybe I could just ask Dr. Stephens if you perhaps have that information.
Yes, thank you, Dr. Duogi. So indeed, we had 9.6% of the subjects that were SARS-CoV-2 positive at baseline, and they were well balanced between the placebo group and the vaccine group. Thank you.
Okay, well, thank you. I apologize because of our technical problems, which increased the time we had at the previous break, that we didn't have as much time as we had hoped for questions here. I would ask the Janssen team to please be available at the start of our broad discussion time for us in the afternoon, because we are certain to have some additional questions for you. Right now, we're going to adjourn for a very brief lunch and start up at 1:10 hard start for the open public hearing. So we'll see you at 1:10, open public hearing.
All right, hold on one moment. Welcome to the FDA's 164th VRPAC meeting. We are now going to be moving into our OPH session. So with that, Dr. Monto, would you please take it away?
Welcome to the open public hearing session. Please note that both the Food and Drug Administration and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the open public hearing session of the advisory committee, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages you, the open public hearing speaker, at the beginning of your written or oral statement to advise the committee of any financial relationship that you may have with the sponsor, its product, and if known, its direct competitors. For example, this financial information may include the sponsor's payment of your travel, lodging, or other expenses in connection with your attendance at the meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your statement, it will not preclude you from speaking. Over.
Good afternoon, everyone. This is Dr. Prabha Atreya. I am the designated federal officer for this meeting. So we're going to be starting the open public hearing speaker and open public hearing session. And we will start with Dr. Diana Zuckerman. Dr. Zuckerman, take it away. Thank you. You have 3 minutes.
Thank you. Can you hear me?
Yes, please.
I'm Dr. Diana Zuckerman, President of the National Center for Health Research. Next slide. Our center scrutinizes the safety and effectiveness of medical products, and we don't accept funding from companies that make those products. However, I inherited J&J stock, so my criticisms today are counter to my financial interests. I'm trained in epidemiology, was a former faculty member and researcher at Vassar, Yale, and Harvard, a former fellow in bioethics at Penn, and also worked at HHS. Please skip the next slide and go to the one titled Preventing Serious COVID. I'm concerned about the hype that this vaccine is effective specifically against moderate and severe COVID. Those are PR claims that are misleading. Not all symptomatic in terms of hospitalization and medical interventions, as the FDA did on page 33 of their briefing document. 10 study participants developed severe COVID at least 2 weeks after their shots, and only 5 developed severe COVID at least 4 weeks after their shots. Most were in the placebo group, but these are very small numbers, and the differences are not statistically significant. It's misleading to tell the public that nobody who was vaccinated was hospitalized unless you also tell them that only 5 people in the placebo group were hospitalized. The data indicate the vaccine is effective, but doesn't prove that the vaccine is especially effective against moderate and severe COVID. Next slide. After 28 days, there were 0 COVID cases for ages 75 and older in the vaccine arm and 4 cases in the placebo arm. Too few to draw conclusions about efficacy in the oldest patients. Next slide. The vaccine is effective, but the median follow-up is only 8 weeks after the shot. Does human immunity last only 2 months or 4 months or a year? We won't know unless the randomized controlled trial is continued. Last slide. In conclusion, the FDA guidance for COVID vaccine approval specified at least a year or 2 of follow-up. FDA's guidance for EUA drastically shortened that to a median of 2 months, and that's exactly what the companies provided. The companies said that the double-blinded studies would continue after the EUA, but that no longer seems likely, and FDA said today that approval might be based on 6 months. As soon as a vaccine is authorized, we start losing the placebo group. If FDA lets that happen, that's a huge loss for public health and a huge loss of information about how we can stay safe. The crossover design is something, but unless it allows at least 6 months of data, we will really be limited in what we know. So at the very least, let's be very honest with the public about what we do know and what we won't know. Thanks very much for the opportunity to speak today.
Thank you, Dr. Zuckerman. The next speaker is Dr. William Fitzsimmons.
I am William Fitzsimmons. I have no financial relationships to disclose. And with my collaborator, Anthony Coniglio, I'd like to thank you for the opportunity to make 2 points. First, support for the Janssen EUA and expansion of the Moderna and Pfizer-BioNTech EUAs to allow single-dose administration. Secondly, a recommendation for advancing the registration trial methodology so that active controlled non-inferiority studies can be used for new vaccine approvals. Next slide, please, slide 2. The Janssen single-dose efficacy rate for preventing moderate to severe COVID-19 is 66%. The single-dose efficacy rate of the Moderna vaccine over 28 days post-dose was 69.5%, which includes the first 2 weeks post-dose. Although the single-dose 28-day efficacy data with the Pfizer vaccine is not directly available, we know that the 21-day efficacy of single-dose is 52.4% and is well over 80% in the 3rd and 4th weeks after first dose. Next slide, slide 3. We support the EUA application for the Janssen vaccine, and the same scientific rationale would indicate that the EUAs for the Moderna and Pfizer vaccines be expanded to include a single-dose administration option. Next slide, slide 4. We ask that the FDA and the advisory committee advance methodological work to enable the performance of active controlled non-inferiority trials. Participants randomized to placebo have experienced significantly more severe infections, including COVID-19 related deaths, as seen in 7 participants receiving placebo in the Janssen study. Reports from ongoing placebo-controlled studies indicate some participants are requesting to leave the studies or are receiving vaccination under the current EUAs, both of which compromise the trials. Next slide, slide 5. Pfizer and Moderna data demonstrate that it should be feasible to propose a non-inferiority margin, and the FDA guidance document indicates a relative efficacy non-inferiority margin when comparing a new vaccine to an effective vaccine. Next slide, slide 6. Additional considerations for future COVID-19 vaccine registration trials include inclusion of patient populations, for example, cancer, autoimmune disease, and transplant recipients that were previously excluded from trials but are at increased risk of COVID-19 morbidity and mortality, antibody testing in these populations, and systematic protocol-defined testing for asymptomatic infections and transmissibility. In summary, we support approval of the Janssen EUA and expansion of Moderna and Pfizer-BioNTech EUAs to allow public health professionals to optimize vaccination for the prevention of severe infection hospitalizations, and deaths as quickly as possible. It is also vital that we re-examine the design and methodologies to facilitate the use of active controlled non-inferiority designs. Thank you very much.
The next speaker is Dr. Dasgupta.
Good afternoon. I'm a side effects surveillance scientist at the University of North Carolina in Chapel Hill. Dr. Lazar, Dr. Brownstein, and I have no conflicts to disclose. Next slide, please. This committee and FDA need reliable information on side effects, and the systems described by Dr. Shimabukuro and Dr. Anderson this morning are groundbreaking. Seriously, y'all, much respect. But there are two areas for improvement. Therefore, I'll share what we learned from deploying government adverse event reporting apps for drugs and medical devices across 13 countries in Europe, North America, and Africa. From the MMWR, early V-Safe reporters had a median age of 46 and 69% were women, seeming to reflect the vaccination-eligible healthcare workforce. But there's more to the story. In our apps, the earliest adopters matched exactly this demographic profile. Many were nurses who already knew about the importance of adverse event reporting. It was much harder to get run-of-the-mill patients to report because we weren't tuned into their motivations. When we asked, their answer was clear, quote, show that someone truly cares about our well-being. So to get sustained reporting from patients via digital tools, we had to tap into motivations that were different from those of early adopters. There are two main reasons why patients report. First is altruism, to prevent side effects in others. Second is social validation, because initial concerns often get dismissed by clinicians. We learned the hard way that too often surveillance systems take valuable information and give patients little in return. Next slide, please. This leads to our second point. Elderly African Americans have exceptionally high COVID morbidity and lower rates of digital access. Many have lived experiences of mistreatment by the healthcare system. Their adequate representation in health data is a form of social justice and equity. To respectfully gather their perspectives, Sentinel sites may be needed to supplement existing efforts beyond the digital divide. The key thing missing in the current data picture is community-based Sentinel sampling with the active participation of marginalized populations. Next slide, please. Can I just come out and say it? The public doesn't know what safety science is, that it even exists. To meet people where they are, we built crowdsourcing tools like Outbreaks Near Me that partner with companies like Facebook, SurveyMonkey, and Google to get population-level insights that are not available elsewhere. These tools can be adopted for in-person data collection. Next slide, please. Final point. The real-world data slides today betray the natural impulse to parse differences between vaccines. Patients do it too. Choice can improve prosocial behavior among those who are hesitant. Choosing between vaccines can be an expression of identity. In turn, expression of identity restores a sense of agency. We anticipate patient choice will be part of the end game as we try to reach those with lingering reservations into next year. Having a third vaccine will help create choices for patients and caregivers and get all of us vaccinated. Last night, The Lancet published our article detailing these comments. Last slide, please. We can be reached here if you have any questions. Thank you for your time.
Thank you, Dr. Dasgupta. Next speaker is Kermit Kubitz.
Good afternoon. I am Kermit Kubitz, and I support EUA for the Janssen vaccine because of the need for additional vaccines and clear positive benefit risk. So far, the path to vaccination is neither frictionless nor fast. My prior comments to this AdCom addressed the need to approve the Pfizer vaccine. Since then, my 84 and 86-year-old sisters have been vaccinated, but my 88-year-old brother, a Korean War-era Navy veteran, has not. He lives alone with caregivers coming in. His medical provider, Peninsula Family Medical Center in Tacoma, Washington, reported to me their, quote, great disappointment and frustration, unquote, that they would not be receiving the COVID-19 vaccine because state officials had chosen to divert the vaccine to other areas deemed more beneficial to the public. So having been vaccinated with the Moderna vaccine myself, I will have to travel to Tacoma to try to navigate the vaccine highway from my brother, with the VA and the state of Washington. We need more vaccines. Page 2, a structured benefit risk strongly supports emergency use of the Janssen vaccine. The pandemic is a serious disease. The only alternatives, the Pfizer and Moderna vaccines, are not fully licensed and limited in supply. The benefits, as revealed in the FDA and sponsor briefing documents, are efficacy above 70% with virtually complete protection against COVID-related hospital and deaths. The risks are limited, as shown by limited adverse events among 40,000 trial participants and low overall fever rates. The conclusion is that a well-understood method of production vaccine with high efficacy justifies EUA. Page 3. My recommendations are to note that adenovirus vaccines may have lesser efficacy in older adults with prior multiple adenovirus infections. It may be desirable to include an adjuvant or booster in future or fully licensed vaccines to promote more antigens and efficacy. In any case, it is important to approve the Janssen vaccine and move forward with community immunity. Thanks to Dr. Messonnier on the annual anniversary of her warning, February 25th, 2020, that we should plan for community spread and remote learning and working. Thank you. Bye.
Thank you, Mr. Kubitz. The next speaker is Dr. Kevin Letnes.
Thank you, committee, for allowing me to talk about serology testing and COVID-19 infection and vaccine monitoring. I'm Kevin Latness. I trained in immunology and rheumatology in the medical scientist training programs at the University of Iowa and Washington St. Louis. Going forward, we should continue to monitor safety and efficacy of vaccinations and determine the need, timing, and safety of booster vaccinations. To do this, our best tool is serologic conversion. Next slide. What does serology tests tell us? They tell us that we have developed immunity to COVID-19. To produce IgG antibodies, our immune system must recognize and respond to COVID-19 proteins by both B cells and T cells. The process of switching from IgM to IgG cannot occur without T cell help, a hallmark of adaptive immunity. As you have seen in data from all vaccine trials, IgG has strong correlation with neutralization assays and T cell assays. Please return to my previous slide. Assays have improved significantly. Improved specificities and the increasingly high prevalence of immunity to COVID-19 have eliminated early problems with false positives. I propose the EUA assays of highest value are those that measure IgG, those that measure quantifiable levels of antibody, those that detect spike protein, in particular the receptor binding domain, and those that are inexpensive and available for high throughput capacities. Next, the CDC recommendations on use of serology testing, last updated in November, need to include using serology to establish a threshold for protection and to monitor maintenance of immunity. From a clinical perspective, vaccine studies evaluate homogeneous populations of healthy individuals. They lack large amounts of data on significant variability in regards to age extremes, immunologic comorbidities, and medical treatments, factors that impact immunity. Vaccines, like drugs, are medical treatments. They come with relative risks and benefits that need to be assessed and monitored. So what can we hypothesize? 1, that some people may not respond with durable immunity. 2, that some people may hyperreact. 3, that at some point, immunity after infection and after vaccination is likely to wane. In these cases, serology testing is very helpful to track immunity. I will close with 2 cases demonstrating why serology testing is a valuable tool. 1, a 32-year-old lupus patient had COVID in the fall. She received her first vaccine dose in January and had significant and lingering post-vaccination symptoms. Testing for lupus activity revealed a lupus flare with a critical drop in her platelets. Serology testing showed evidence of antibodies, so I recommended postponing her second vaccine dose until her lupus is stabilized and her serologies wane. Second, a 67-year-old woman with severe autoimmune disease treated with immune-modifying medications received both vaccine doses in January. Serology testing this week showed she had not seroconverted. So my plan is to continue monitoring her serology over the next few months, and if persistently negative, revaccinate when the vaccine supplies are no longer limited. Thank you for your time.
thank you, Dr. Letnis. The next speaker is Dr. David Berger.
Hello. I'm a board-certified pediatrician in Tampa, Florida, and I specialize in preconception, infancy, and wellness, and patient. I am also a patient vaccine consultant. I also serve as an associate professor at the University of South Florida College of Nursing and the senior medical advisor of the Vaccine Consideration Project. Thank you for inviting me back to present to this committee. I have no conflicts of interest. To the technical staff, please note that I will not be using slide number 11. Before I start my presentation, I again want to express significant frustration with this process, which required us to submit slides and written comments before the vaccine data was available to the public. If the FDA truly values our input, it would give us sufficient time to review the data. Now, if you can please go to slide number 3 and start my presentation. My comments today are about the use of COVID-19 vaccines in pregnant women and people of childbearing age. No studies of pregnant women have been published. Americans are asking if they should get a vaccine if pregnant or trying to conceive. Each will weigh the options and need more information, and we must be open and honest with them about what is known and not known. Next slide, number 4. Morbidity associated with COVID-19 in pregnant women has been established with increased ICU admissions, preterm births, and admissions to a neonatal ICU. Next slide, 5. Ongoing hesitancy relative to vaccine continues. In a recent survey, at least 1/3 of people aged 15 to 50 say they would probably or definitely not take a vaccine even if determined to be safe and free of charge. Multiple polls have also shown women are more likely to have vaccine hesitancy than men. Therefore, large numbers of women of childbearing age have vaccine hesitancy. Next slide, 6. Hesitancy and confusion has likely increased due to conflicting statements from various organizations. Next slide, 7. We have learned that adenoviral vector DNA vaccines, such as this one, enters the nucleus, where the human DNA resides. Messenger RNA vaccines do not enter the nucleus. Next slide, number 8. This difference has led to questions about whether this vaccine could interfere with pregnancy, especially from conception through early fetal development. Upon finally being able to review the data 2 days ago, and with only 8 pregnant women included in the trials, safety and effectiveness conclusions cannot be made about this vaccine and pregnancy. Next slide, number 9. After researching this issue over the past 2 weeks with my research team, we could find no medical research or scientific opinion addressing whether a human embryo could be negatively impacted by this type of vaccine. I did locate a CDC article showing adenovirus can enter sperm, and I found one mouse study indicating adenovirus vector DNA does not show up in the DNA of mouse offspring. Yesterday, a vaccine researcher I spoke with suggested that the amount of vaccine particles injected into the arm that could reach testicles or the uterus may not be enough to be consequential. As per the agency's mission statement, the FDA is responsible for protecting the public health by ensuring the safety, efficacy, and security of human drugs. Applying this directive to COVID-19 vaccines in people of childbearing age, I believe the FDA has a responsibility to proceed with caution as more data is gathered. However the FDA decides to proceed, we can empower people by providing the information necessary to weigh the benefits and risks of taking a vaccine. Such decisions are very personal, and their decisions should be respected. Transparency on the part of the manufacturers and the government needs to be better. It should not be so hard to find information. In the end, it all comes down to informed consent.
Okay, thank you, Dr.
If you want to wrap up.
Yes, if I could just wrap up, I just have one more sentence, 3 more sentences. So informed consent by definition means individuals must be fully informed so they consent to taking or not taking the vaccine. Next and last slide. The Vaccine Considerations Project is building a repository of information related to vaccine safety concerns and efficacy. We will continue to tackle the issues of most concern to the public and share whatever information we find. Please reach out to us if you wish to join our endeavor. Thank you for your time.
Thank you, Dr. Berger. The next speaker is James Krupnik.
I have no financial relationships to disclose. Hi, I'm Jared Krupnik. I'm the president of Uniting for Action and the founder of the Vaccine Considerations Project. We're working to make sure that all health and safety concerns are given due consideration. Thank you very much for this opportunity. Next slide, please. Specifically, we're here today to address preventing health inequities. There's a prevailing concern that differences in efficacy between different vaccines could exacerbate health inequities. A solution is for the FDA to provide critical information for the public. I wasn't sure if this was within the FDA's mission, so I looked it up. Next slide, please. The mission says, in part, the FDA is responsible for helping the public get the accurate, science-based information they need to use medical products, in this case vaccines, to maintain and improve their health. Next slide, please. There are scientific differences between the vaccines. Currently, many public health experts are saying the important statistics are that each of the vaccines prevent hospitalizations and deaths. Those are indeed very important benefits of all the vaccines, but there are other severe potential impacts within families and communities beyond hospitalizations and deaths. Next slide, please. Some severe and potentially devastating consequences for individual families and communities include more missed work, more spread to non-vaccinated family members, more long-term health impacts. Next slide, please. The FDA needs to provide information about the differences in the vaccines to people to allow them to provide an informed consent. It erodes credibility to, on one hand, repeatedly say, trust the science, and then when it comes to vaccine differences, to effectively say, ignore the science. Next slide, please. If it's found that marginalized communities are faring worse, in part due to vaccine differences, it will further deepen mistrust and skepticism and decrease vaccine uptake. Next slide, please. Here's just a simple example of the type of comparative data that would be important for people to know. For families who are one missed paycheck away from going hungry or from losing their home, the difference in the science are critical for them to know. I implore the FDA to not just publish data, but to take ownership of ensuring that every individual who is considering a vaccine have all of the best scientific information they can have to make informed decisions for themselves and their family. Next slide, please. Our national team of professionals and graduate students are actively evaluating concerns and working to provide the information individuals need to make informed choices. We encourage everyone who shares our intentions to join our efforts. So go to vaccineconsiderations.com for more information related to this and to Dr. Berger's presentation, and to sign up to receive more information or to join our team. Thank you very much.
Thank you, Mr. Krupnik. The next speaker is Benjamin Newton.
Hi, thank you so much. I'm here to talk about how we can save the most lives. I have a financial interest in Moderna, and if you follow my recommendations, I will be harmed financially. I encourage you to approve this vaccine. Many people with PEG allergies need it. It's an important first dose. Next slide. You can see that the vaccine is insufficient to stop the spread of the South African variant based upon South African efficacy. You need a 100% vaccination rate, which is unlikely. Next slide. The tested vaccine doses are not magic. Dosing and regimen for all vaccines was decided for commercial reasons based upon what was known at the time the study commenced. Zero-bridging is a real and accepted practice at the FDA and CDC. It is actually required by the EUA filing. I would encourage us to use all what we know about biology and not just what was tested, so that we can actually follow the science. Next slide. The mRNA vaccines boost, the adenovirus vaccines don't boost effectively. The one-dose efficacy is listed at the top, and you can see the boost increase for Pfizer and Moderna is 16-fold and 7-fold, which is fantastic. Next slide. What this leads to is some amazing vaccine supply math. Doubling the dose provides less than twice the protection. Each additional shot increases protection 10-fold. This means that for any supply of vaccine, every person in the world can be fully protected. The right questions to ask are, what is the manufacturing capacity? How many people? How many doses will each person require? And what we see if we ask those questions on the next slide is that using the single dose size for Pfizer and Moderna and the American doses currently produced per month for Pfizer and Moderna, using zero bridging, we can see that a 1 microgram dose for Pfizer and a 1.5 microgram dose for Moderna is sufficient for 90% efficacy. That leads to 2.2 billion people protected each month, which is enough to bring COVID to an end in a very short period of time. Next slide. Ethics. COVID has led to a lot of ethical issues and difficult ethical choices, and we've all had to make them. since the last time I spoke to VRPAC, 900,000 people have died of COVID. So next slide. Right here, what we have is a regulatory bottleneck. We should be on slide 8 here, not a manufacturing bottleneck. I encourage each of you to have political courage, lower the doses for Moderna and Pfizer. Who's doing this already? Britain has already figured this out. Monsef Slawi already commented on the logical extension. I've put together a YouTube video. It's about as interesting as watching paint dry to talk about any questions you might have. I do thank you for your time and service. Everyone without a vaccine has the same R0 as an anti-vaxxer. While you've listened to me speak, 6 additional people have died of COVID. I urge you to act with all due haste. Thank you very much for your time.
Thank you, Dr. Newton. The next speaker is Dr. Sidney Wolf.
I'm Dr. Sidney Wolf of the Public Citizen Health Research Group. I have no conflicts of interest. I support granting an EUA for Janssen vaccine despite decreased efficacy in older people with comorbidities. With the current EUA availability of 2 COVID vaccines, the third probably next week, it will be neither practically nor ethically feasible to continue recruiting new participants to placebo-controlled trials unless they will ultimately get vaccinated, whether initially randomized to the vaccine or placebo group. On December 10th, before your committee, I stated that, quote, an important unresolved conflict exists. If an EUA is granted for widespread use, should the 19,000 participants in the Pfizer trial who received a placebo be notified of this and be offered a vaccine by Pfizer, clearly encouraging them to stay in the trial? Status uninformed, trial participants might otherwise leave the trial to try and get vaccinated with the Pfizer or any other EUA available vaccine. Parenthetically, a couple thousand people have left the Janssen trial for exactly this reason. The unblinding vaccine-providing proposal has important advantages. Once an EUA is granted, the ethical obligation to both inform all placebo recipients of their status and offer them the vaccine within the context of the clinical trial is met. The originally vaccinated group could be compared with the newly vaccinated group to continually compare rates of new COVID infection with increasing duration of vaccination as well as adverse reactions. Pfizer's stated preference was, quote, this is during the Pfizer hearing on the 10th of December, was that such individuals be vaccinated within the study in order that both safety and efficacy data can be continued to be collected. We believe this approach will minimize the number of current participants who choose to withdraw from the study once a vaccine is available and will maximize the collection of data that can inform long-term safety and efficacy of the Pfizer vaccine. Now, 2.5 months later than December 10th, Janssen now proposes that, quote, this is their slide 62 this morning, upon authorization by a regulatory authority, all placebo participants will receive 1 dose of their vaccine. If granted by the FDA for Janssen's and subsequent EUA granted COVID-19 vaccines, all placebo participants will later get a vaccine. But what about the 19,000 Pfizer subjects and 15,000 Moderna subjects who were randomized to placebo groups? At the time of EUA authorizations, both companies had similarly, Moderna, like I just quoted from Pfizer, had similarly expressed their preference that subjects previously given placebos be notified of this and offered a vaccine. As of now, how many of the original 34,000 Pfizer and Moderna placebo recipients have been notified of the status and offered a vaccine? And what will occur and when will this occur for the almost 20,000 Janssen placebo recipients who were risking their lives, as were the people without knowing it in the Pfizer-Moderna studies, in order to find out that they are eligible for an EUA?
Time.
Thank you very much.
Thank you, Dr. Wolf. The next set of our presenters did not submit any slides for their presentation, so we will only hear their verbal comments. Thank you. The next speaker is Kim Witzak.
Good afternoon. My name is Kim Witzak, and I'm speaking on behalf of Woody Matters, a drug safety organization started after the death of my husband due to an undisclosed side effect of antidepressants. We represent the voice of families who live every day with the consequences of the current drug safety system, effective and accessible medical treatments. There is an excitement in the air. You can feel the energy of excitement growing. People are looking for hope, and these vaccines seem to be providing it. Hope is a powerful motivator. But what is hope based on? On the surface, the efficacy of J&J's vaccine may not be as high as the other two investigational vaccines on the market, but it is one shot and doesn't need the extreme preparation like the others. Since it is all but guaranteed that J&J will receive emergency authorization this weekend, I'm going to use my time to address some general concerns I have. The public has not been explained what emergency use authorization really means. Most assume it means FDA approval. We may never have FDA approval on any of these vaccines, especially if we lose the placebo control group on the ongoing phase 3 clinical trials. These vaccines need to continually be framed as investigational, and we are learning as we go in the real world with all the risks that that entails. It seems EUA has become the new standard. With new variants popping up, there will be an endless market for potential booster shots. Warp speed, testing, then deploy, and hope for the best seems to be the new acceptable strategy. Is the public being given true informed consent and made aware that these vaccines are still investigational and may not stop transmission, may not stop your life, has not been tested on all types of people like those with immune deficiencies and those who are pregnant? And now we are seeing celebrities, politicians, influencers joining in on the mass vaccination effort while telling the public the shots are safe. Speaking of safe, how can the public be assured that harm's adverse events are being taken seriously? The adverse events that we have seen occurred in the short term are being quickly dismissed and accepted as the vaccine is working. It's priming our immune systems. I was personally workers that experienced horrific side effects after their first shot. They took the proper reporting measures to the FDA, CDC, and the companies. To this day, none of them have been contacted or followed up by the companies. However, contrast this with the Reuters story from earlier this week with the headline, "First months of shots find no safety issues with Pfizer, BioNTech, Moderna vaccines." Of course, we still know nothing about the long-term impact on our immune systems, fertility, and other health-related issues. Ultimately, in this current pandemic environment, the public is the real-world clinical trial. It is one big human experiment. And thank you for your time, and I appreciate your careful consideration of my comments.
Thank you, Ms. Witzak. The next speaker is Ms. Anne Lewandowski.
Hello, my name is Anne Lewandowski, and I am the Executive Director of the Wisconsin Immunization Neighborhood. I have no conflicts. I would like to begin by thanking the sponsors and members of VRPAC for their time today. As a public health professional, I am thrilled to see a one-dose stable vaccine that can be distributed to hard-to-reach communities. I'm also thrilled to see that some groups are providing high acceptability of this vaccine. I'm deeply appreciative of the sponsor included transmission data points and to help us communicate to the public that these vaccines do more to protect not just the individual, they help protect the community. We support the 5-dose vial size as appropriate for small clinic locations and trying to get into a hard-to-reach community. We ask that you consider a flexible minimum order size and shipping amounts for the vaccine. We very much support Dr. Office and others' suggestions that clear messaging should be made for the public on whether or not this may turn into a two-dose series in the future. Like others, I support the comments that many of these populations do not have adequate data. We must help the public understand where data exists and what gaps may be, and at what time frame these questions and data gaps may be clarified. Of particular interest, as many have already noted, autoimmune disorders and pregnancies are critical for many people in the United States as they make decisions to be vaccinated. Thank you.
Hello? Apologies.
Yep,
Was I
Prabha,
skipped?
yep, we're back. Yeah, we're back on track now. Prabha?
Okay. The next speaker is Ms. Sarah Gustafson.
Hi, thank you.
Could you speak up
My
a
name is
little
Sarah.
bit? I think the volume is low.
All right, I will take off my headset then. My name is Sarah Christophers. I am the Policy Advocacy Director at the National Women's Health Network, a nonprofit advocacy organization that has been bringing the voices of women to the FDA for 45 years. We are supported by our members and do not accept financial support from drug or device makers, and I have no conflicts of interest to disclose. We believe that an emergency use authorization based on the data presented this week is appropriate under the current circumstances. A one-dose vaccine effective in preventing severe disease, including against several known variants, that can be stored for 3 months at normal refrigeration temperatures with fewer logistical constraints has the potential to markedly reduce hospitalizations and deaths. With 2 authorized vaccines currently on the market, we now have real-world data about how logistical challenges in distribution have hampered equitable access. The Janssen vaccine represents a big leap forward for access, particularly in low-income, rural, and other underserved communities. However, because it's difficult to make an apples-to-apples comparison between vaccines authorized based on data collected before new variants are believed to have been in widespread circulation and today's data, there is a significant concern that headline numbers are already leading to a sense among the public that there are first and second class vaccines, with the latter relegated to low-income, rural, or otherwise marginalized communities. That has the potential to exacerbate existing mistrust. Public health authorities must address these perceptions head-on. Secondly, and as we've heard in previous meetings of this committee, CDC data indicate that Black and Indigenous people living in the U.S. are roughly 4 times more likely to be hospitalized from COVID-19 and roughly 3 times more likely to die from the virus than their white counterparts. Racism, both systemic and interpersonal, healthcare disparities, and increased workplace exposure are all factors. When examining today's clinical trial data, we see that the data for racial and ethnic groups track closely with each group's share of the U.S. population, but do not account for the disproportionate impact of the pandemic on different communities. Given that disparity of impact, which was already well established at the time that trials were begun, the sponsor should have sought to enroll Black, Latino, and Indigenous participants relative to their vulnerability to the virus, not share of total population. Thank you for your consideration.
Thank you very much. The next speaker is Ms. Linda D.
Hi, my name is Linda D. I'm from AIDS Action Baltimore. I've been a community rep on a number of antiviral advisory committee hearings, and I have no financial relationship with the sponsor. I support the EU application given the risk-benefit ratio presented here. Back in October, I had a laundry list of issues, many of which have been addressed by the sponsor, including the intention to use an open-label crossover study for phase 3 placebo-armed participants. I'm actually surprised by the large number of people between 65 and 74, the many racial and ethnic minorities, and people with pre-existing COVID-19 outcome-affecting morbidities. I mean, I've never seen this before in 35 years of doing this work, this many. At least we've started to enroll the correct people in the studies here. Some data was also provided on pregnancy outcomes for trial participants, and the sponsor intends to conduct a trial on children, pregnant women, and immunocompromised people. People with HIV were included at the outset of the study, and at least some study data was provided. There are many people with HIV who are interested in being vaccine study participants. I hope all sponsors will do a better job of recruiting people with HIV for future studies. Outreach to the HIV community will undoubtedly help recruitment. AD26 has lower efficacy than the messenger RNA vaccines. This may be affected by the location of the studies, but there is benefit regarding hospitalizations and death. There is also a great benefit in no rate-limiting storage or transportation requirements and a one-dose regimen, although the results of two-dose studies will be very important. And I also share Dr. Offit's concerns about a two-dose vaccine regimen. Hopefully, efficacy in people 60 and over with comorbidities will be confirmed. Thromboembolic events possibly related to the use of Ad26 is concerning and should be followed carefully. The sponsor will also be studying real-world effectiveness and evolving viral variants using genetic sequencing and immunogenicity data. The FDA should require specific duration and transmission studies. The agency should also make similar study recommendations for all the above for future studies and further address both authorization, BLA, and post-marketing requirements. The FDA's planned guidance update is a great start. I'm committing to using every opportunity to stress for the record that the government must address both vaccine hesitancy issues and vaccination access digital divide issues to promote trust and enroll more people of color and other underrepresented people in vaccine trials. To do otherwise would be a disgraceful continuation of generational neglect. Once again, I'd like to thank the FDA for their tireless work and VRBPAC members for your dedicated service and commitment and for the opportunity to comment.
Thank you, Ms. D. The next speaker is Ms. Nisa Shafi.
Yes, good afternoon. I'm Nisa Shafi. I'm present today on behalf of the National Consumers League. I have no conflicts of interest to disclose. Our organization extends its gratitude to the Vaccines and Related Biological Products Advisory Committee for the opportunity to amplify consumer voices regarding the Janssen Biotech COVID-19 vaccine. For over 120 years, NCL has championed efforts to increase vaccine education, safety, and access for consumers. As consumer advocates, we thank the Food and Drug Administration for their commitment to fostering public trust throughout the development and approval of a vaccine for COVID-19. We have been encouraged by the transparency and opportunities for engagement afforded to the public during this process. Consumers are relying on the FDA more than ever for guidance pertaining to treatments for COVID-19, and preserving their confidence in the agency is of vital importance at this time. Emergency use authorization, while not intended to replace randomized clinical trials, has been a critical component to the nation's pandemic strategy. NCL appreciates the FDA's recognition of clinical trials as a vital component to demonstrating safety and efficacy of a treatment. We are encouraged by reports indicating that the Janssen Biotech vaccine has proven to be effective against hospitalizations and deaths from COVID-19. The added benefit of another vaccine is to decrease virus mutation. Presently, 3 far more contagious variants of COVID-19 spread and could hamper efforts to quell the virus. We are reassured that the Janssen vaccine has demonstrated efficacy against certain variants. As new data is collected, we call on the FDA to perform post-market surveillance to monitor ongoing efficacy. Vaccine hesitancy and social determinants of health remain critical obstacles in the vaccine rollout process. The Janssen Biotech single-shot vaccine has the potential to increase access for hard-to-reach communities, bringing us closer to herd immunity. This week, we marked a grim milestone, as 500,000 Americans have now perished from this relentless virus. Amidst this loss, the continued development of vaccines for COVID-19 has provided the nation with much-needed hope and respite. As the committee deliberates on the Janssen Biotech COVID-19 vaccine, we request the agency to consider the benefits of its release for historically disadvantaged communities, for which this vaccine would be logistically more accessible than the prior 2 vaccines. Thank you to the committee for your considerations of our views. Through our consumer education work, NCL will continue to support the FDA in its efforts to develop a safe, effective, and expedited pathway towards a vaccine for COVID-19. Thank you.
Thank you, Ms. Shafi. The next speaker is Dr. Peter Doshi.
Hello, and thank you. I'm Peter Doshi. I'm on the faculty at the University of Maryland and a medical journal editor at the BMJ. I have no relevant conflicts of interest. No one's paid for my attendance, and these comments are my own. First point, I'm nervous about the prospect of there never being a COVID vaccine that meets the FDA's approval standard. The agency has already authorized 2 COVID vaccines as meeting the EUA standard of may be effective. Granting another EUA to Janssen would begin to create a kind of marketplace of vaccines good enough to be authorized but never approved. The briefing documents say that Janssen's seeking an EUA, but they don't say why. My question is, if Janssen is fully confident in the data, why not seek a full approval, a BLA? Looking forward, I worry about FDA lowering its approval standards. Last June, FDA outlined its expectations for an approvable vaccine, saying participant follow-up should continue, quote, for at least 1 to 2 years. We know Moderna and Pfizer can't meet this standard as placebo recipients are already being vaccinated. And in its briefing document, Janssen says that if an EUA is granted, they will unblind their trial. It's quickly seeming that the only way a vaccine will ever be approved is if FDA lowers its standards to the may be effective standard of the EUA. Is this what we want? If the FDA now believes that a few months of follow-up is sufficient to be certain benefit outweighs risk, the agency needs to tell us why it changed its mind. We thankfully have a waning epidemic in the U.S. right now, and manufacturing capacity of already EUA'd vaccines continues to grow. The argument that we don't have the luxury of time to demand better evidence doesn't hold as much water as it might have 2 months ago. Second, I worry about process. The way it's supposed to work is the FDA asks the advisory committee for its honest, independent view. But the media reporting on this suggests an EUA is a foregone conclusion. I want to know if FDA is doing anything to ensure advisory committee members can truly vote their mind and not bow to the pressure that there is only one right decision. Third, it's unreasonable to accept Janssen's labeling of its primary endpoint moderate to severe critical COVID-19 because it includes what most would call mild disease. A lab-positive test plus 2 symptoms like cough and headache would be sufficient. Everyone knows that the majority of COVID cases are mild, yet in Janssen's trial there were only 4 cases of mild COVID compared with 390 so-called moderate cases. See page 17. Clearly, Janssen's moderate is what everybody else would call mild. The case definition of severe COVID also needs scrutiny as PCR-positive cases with no other symptoms other than a blood oxygen saturation of 93% or less would qualify. There's a real urgency to stand back right now and look at the forest here as well as the trees, and I urge the committee to consider the effect FDA's decisions may have on the entire regulatory approval process. Thank you.
Thank you, Dr. Doshi. The next speaker is Dr. Robert Kaplan.
So a faculty member at the hello, this is Robert Kaplan. I am a distinguished professor emeritus at the UCLA Fielding School of Public Health, and I'm also a faculty member at the Clinical Excellence Research Center at the Stanford University School of Medicine. I'm employed by Stanford, and I have no conflicts of interest to report. With colleagues, I've reviewed the data prior to the EUA hearings for Johnson, Pfizer, and the Moderna vaccines. We consider this to be part of our responsibility as academic research scientists. But as interested scientists, we have some concerns. The public has been saturated with non-peer-reviewed press releases that shape public opinion and have created very high expectations. We believe that constructive feedback improves and clarifies data interpretation. The scientific community has embraced an open data movement that emphasizes posting of data for reanalysis by independent scientists like us. None of these safeguards are in place with the EUA reviews. The briefing document is excellent, but it raises many questions, which various colleagues have commented on today. We have concerns because EUAs are for emergencies and they should be temporary. EUA might use a lower standard to speed early dissemination, but continuing use should depend on emerging data. The trials have included between 30,000 and 44,000 participants, large sample sizes. However, we have more experience. 66 million doses have now been administered in the U.S. Yet we don't have access to data on how many people have been infected post-vaccine. We have minimal information about serious adverse reactions outside of trials. Now, VSafe and VERS certainly are a step in the right direction, but they don't use representative samples from the population. So what needs to be done? First, we need more transparency. Some of the vaccines have been developed at public expense and are being paid for using public resources. Therefore, the data should be made public. We need independent analyses by investigators who are not employed by the manufacturers. Importantly, the data must be available more than 48 hours prior to these hearings. Second, the public access surveillance system should be a condition of the EUA. During the EUA, there should be continuous monitoring post-vaccination for COVID cases, deaths, and serious adverse reactions. This could be done on a population basis or based on true representative samples of vaccine recipients. And third, we need to preserve control groups. There's been a precipitous decline in COVID cases in countries that have and have not started vaccine programs. Without control groups, we won't be able to figure out causation. In the interest of transparency, please remember that if it is in the public interest, it should be in the public domain. Thanks again for listening today.
Thank you, Dr. Kaplan. The next speaker and the last speaker of the open public hearing session is Mr. Michael Ward.
Hello and good afternoon. My name is Michael Ward, and I'm the Vice President of Public Policy at the Alliance for Aging Research. I have no financial conflicts to disclose. The Alliance is one of 3 convening members of the COVID-19 Vaccine Education and Equity Project, along with Healthy Women and the National Caucus and Center on Black Aging. As you can see at covidvaccineproject.org, we are joined by more than 175 leading organizations representing a wide range of patients, professionals, and diverse communities. We are focused on promoting widespread and equitable access to COVID-19 vaccinations and information, especially in hard-hit communities. As an organization committed to advocating on behalf of older Americans, the Alliance is thankful that the scientists and researchers at Janssen have successfully developed a safe, effective vaccine. Moreover, we appreciate that individuals aged 60 and older and people of color were prioritized in the study design, resulting in significant enrollment of clinical trial participants from groups disproportionately susceptible to severe outcomes and death from COVID-19. We are impressed that the vaccines thus far have been so effective. However, the primary endpoints in the Janssen trial differ from previously authorized COVID-19 vaccines, as did levels of community transmission and the documented presence of emerging variants during the trial period. We hope the FDA and other agencies within HHS will work with us and others to shape messaging and communicate the encouraging takeaway that all FDA-authorized vaccines will prevent many from contracting SARS-CoV-2 and prevent most serious medical complications arising from COVID-19. Further, the ability of the Janssen vaccine to provide protection against severe disease and death resulting from the B.1.351 variant is critical. We also appreciate the preliminary data made available by Janssen regarding the impact of a range of comorbidities on efficacy. We encourage monitoring for the age 60 and older population that has pre-existing conditions, especially hypertension and diabetes, across all COVID-19 vaccines with larger subgroups of patients. Such comorbidities are endemic. Improving the understanding of the influence of these comorbidities will be helpful not only for the Janssen vaccine, but as other COVID-19 vaccines are developed and updated. Janssen is conducting a second clinical trial to test a 2-shot regimen of this vaccine. As Dr. Offit noted this morning, stakeholders should begin evaluating policy issues now around the results of the ongoing Ensemble 2 trial. For example, if a 2-shot regimen induces an enhanced level of protection, will patients who received a single shot be given the option to receive a second shot? What level of enhanced protection will merit reserving a percentage of available vaccine to enable distribution of a second shot, given current excess demand? These are not easy questions, but ones worth considering as soon as possible. In conclusion, the Alliance is thankful that an additional tool may be publicly available soon in our fight to end this pandemic. We encourage the agency to work in concert with colleagues throughout HHS to advance public confidence and ensure equitable vaccine access. Thank you.
Thank you, Mr. Ward. This concludes the open public hearing session, and then we will go into the regular session. Dr. Mancho, would you introduce the next speaker, Dr. Rachel Zhang and Dr. Yosefa Hector? Thank you very much.
Yes, it's my pleasure to introduce for the FDA presentation and describing the voting questions which will follow our discussion, the FDA presentations are being given by Rachel Zhang and Josefa Hefter. Please.
Thank you very much. All right, so first I'll give a brief introduction of the Janssen COVID-19 vaccine and take you through the clinical development program to date. Then we will take a closer look at the efficacy data from the phase 3 study. Then I'll turn it over to Dr. Hefter to take you through the safety data, the pharmacovigilance plans, and summarize the benefit-risk assessment in context of the proposed use under EUA. So the Janssen COVID-19 vaccine is a recombinant replication-incompetent adenovirus type 26 vector vaccine, which encodes the SARS-CoV-2 spike protein produced in PER-C6 cells. The vaccine is administered intramuscularly as a single-dose regimen at the dose of 5 × 10^10 viral particles. The proposed indication under EUA is for active immunization to prevent COVID-19 caused by SARS-CoV-2 in individuals 18 years of age and older. Shown here are the ongoing studies with the Janssen COVID-19 vaccine. All the studies are randomized, double-blinded, and placebo-controlled. I will give you a brief overview of each study, except for study 1002, which is a phase 1 non-US IND study, and then focus on study 3001, which is the study submitted to support the EUA application. Study 1001 is an ongoing phase 1/2 safety and immunogenicity study in approximately 1,000 participants, with 2/3 of the participants between the ages of 18 to 55 and 1/3 65 years of age and older. This study assessed 2 dose levels and a 1 and 2 dose vaccination regimen. Interim study results showed that a single dose at 5 × 10^10 viral particles was able to induce SARS-CoV-2 binding and neutralizing antibodies in both age cohorts. There was a Th1 skewed CD4 T cell response elicited. Study 2001 is an ongoing phase 2 study in healthy adults and 2 dose regimens. Interim immunogenicity assessments at day 29 show the vaccine elicited SARS-CoV-2 neutralizing antibodies in the adult cohort consistent with results from study 1001. This study was initiated on August 31st, 2020, and review of SAEs from this study up to the time of EUA submission revealed no safety concerns to date. Study 3009 is an ongoing phase 3 efficacy, safety, and immunogenicity study of a 2-dose vaccine regimen, spaced 56 days apart in approximately 30,000 participants. This will be a multinational study and will include sites in North and South America, Africa, Asia, and Europe. The study was initiated on November 16, 2020, and enrollment is still ongoing. No safety concerns have been identified to date based on review of blinded SAE reports up to the time of the EUA submission. 3001 is the study used to support the EUA application. This is an ongoing phase 3 efficacy, safety, and immunogenicity study in 44,325 participants across the U.S., South Africa, and 6 countries in Latin America. Participants were stratified by 2 pre-specified age cohorts and randomized 1-to-1 to receive a single dose of vaccine at 5 × 10^10 viral particles or saline placebo. Enrollment was staggered in stages based on age and comorbidity, so that participants without comorbidities were enrolled earlier than those with comorbidities. The protocol specified that at least 30% of the study population would be participants 60 years of age and older. This study was initiated on September 21st, 2020, and the total planned study duration is 2 years. This slide summarizes the scheduled visits and assessments for study 3001. Primary analysis was triggered on January 22nd when there was a median follow-up of 58 days in the overall study population. Active surveillance for COVID-19 is conducted via an e-diary with prompts twice weekly for the first year of the study and then bi-weekly thereafter. Solicited local and systemic adverse reactions are collected for 7 days post-vaccination and unsolicited adverse events through 28 days post-vaccination in the safety subset. All participants will be followed for medically attended AEs for the first 6 months after vaccination, and SAEs and medically attended AEs leading to study discontinuation will be followed for the entire study duration of 2 years. Blood samples will be collected at scheduled visits for immunogenicity assessments and for the asymptomatic infection endpoint. Shown here are the case definitions for moderate COVID-19, which is a positive SARS-CoV-2 PCR plus any one of the following new or worsening respiratory symptoms as listed on the left-hand side, or any two of the following new or worsening systemic symptoms as listed on the right-hand side. The case definition for severe critical COVID-19 is defined as a positive SARS-CoV-2 PCR plus any one of the following new or worsening signs or symptoms as listed in the box with specific parameters for each further specified in the protocol. All cases meeting the severe critical definition and all cases meeting the moderate case definition as shown in the previous slide that included more than 3 signs and or symptoms were evaluated by an independent blinded adjudication committee. Only cases adjudicated as severe by the adjudication committee were included in the severe critical COVID-19 endpoint. The study included co-primary efficacy endpoints of vaccine efficacy to prevent protocol-defined moderate to severe critical COVID-19 confirmed by the central laboratory occurring at least 14 days and at least 28 days after vaccination. The original study protocol had a single primary endpoint of onset at least 14 days after vaccination, and the 28-day endpoint was added as a co-primary endpoint in December while the study was ongoing. The primary efficacy success criterion is met if the null hypothesis of VE less than or equal to 30% is rejected and the VE point estimate of 50% or greater for both co-primary endpoints. The primary efficacy analysis will be triggered with an accrual of at least 42 moderate to severe critical COVID-19 cases, at least 6 moderate to severe critical COVID-19 cases among participants 60 years of age and older, and at least 5 severe critical cases of COVID-19 in the placebo group with a favorable vaccine to placebo split for both primary endpoints. To align with the median follow-up requirement as specified in FDA's guidance document, the primary analysis was not conducted until at least 50% of the participants in the study have had 8 weeks follow-up post-vaccination. Secondary endpoints evaluated in the study include vaccine efficacy to prevent any symptomatic COVID-19, including mild, moderate, and severe disease, COVID-19 for FDA harmonized case definition, efficacy against severe critical COVID-19, COVID-19 requiring medical intervention, COVID-19 related deaths, and asymptomatic COVID-19 as inferred through seroconversion using serology against the nucleocapsid protein. The protocol specified that COVID-19 cases diagnosed by a positive SARS-CoV-2 PCR obtained using an FDA-authorized test at a local laboratory be sent to the central laboratory at the University of Washington for confirmation. The statistical analysis plan further stipulated that the primary analysis would be triggered and based on the centrally confirmed cases at the time of data cutoff. Due to the high incidence rate of COVID-19 during the study, not all positive PCR tests accrued by the time of the trigger for the primary analysis have been confirmed by the central laboratory. At the time of the primary analysis, about 70% of the accrued cases have undergone the central confirmation process. Another 18% were in the shipping process, and 12% were received by the central laboratory but still being processed. At the time of the data cutoff, there was high concordance observed between the local PCR results and central lab confirmation at around 90.3%. The majority of discordant cases were those with low viral loads, which may have been impacted by the freeze-thaw cycle during the shipping process. In today's discussion, we will present only results from centrally confirmed cases for the primary efficacy analysis as specified by the protocol. For subgroup analyses, we will also include results from those cases with positive PCR from a local laboratory and still awaiting confirmation by the central laboratory to increase the number of cases and the precision of the estimates. The study analysis populations are shown in the table on this slide. The full analysis set included all randomized participants with a documented study vaccine administration. The protocol set used for the efficacy analysis included all participants in the full analysis set who had no immunologic or virologic evidence of prior SARS-CoV-2 infection at baseline. The safety subset is a subset of participants used for the analysis of solicited adverse reactions and unsolicited adverse events through 28 days post-vaccination. At the time of the primary analysis, the median follow-up duration for all participants in the efficacy and safety analysis populations was 8 weeks post-vaccination. Phased enrollment by age group and comorbidity resulted in differences in follow-up duration between participants in these groups, with approximately 2-week difference in the median follow-up time between the first group enrolled, which are participants 18 to 59 without comorbidities, and the last group enrolled, which are participants 60 years and older with comorbidities. Next, we will take a closer look at the efficacy results from Study 3001. This table shows the demographic characteristics of the per protocol set, which was the population used for the analysis of efficacy. The demographic characteristics among vaccine and placebo participants were similar. The median age in the study was 53, with about 20% of participants 65 years of age and older. There was a slightly higher percentage of males compared to females. 62% of participants were white, 17% were black or African American, 8% Alaska Native or American Indian, including American Indians from Latin America countries, 3% Asian, and 0.3% Native Hawaiian or Pacific Islander. In terms of ethnicity, 45% identified as Hispanic or Latino. About 47% of participants are in study sites in the U.S., 40% in Latin America countries, of which a majority are in Brazil, and about 13% in South Africa. About 40% of participants had one or more comorbid conditions associated with an increased risk of progression to severe COVID-19, as listed by the CDC at the time of study initiation. Overall, the demographics from the U.S. participants in the study are similar to the global demographics, as shown in these slides, except for some variations in race and ethnicity composition. This table shows the disposition of participants from the study. The proportion of participants excluded from the per protocol set were balanced between the treatment groups, with the vast majority of those excluded due to positive baseline SARS-CoV-2 status. Overall, few participants were discontinued or lost to follow-up. As of the data cutoff date for the primary analysis, 5.3% of participants in the vaccine group and 5.8% of participants in the placebo group were unblinded by request as they became eligible to receive an authorized COVID-19 vaccine under EUA. A slightly greater proportion of participants 60 years of age and older were unblinded at 6.6% compared to those 18 to 59 years of age at 4.4%. And the vast majority, about 93% of participants who were unblinded were from U.S. study sites. Shown here are the co-primary efficacy endpoints of protocol-defined moderate to severe critical COVID-19 confirmed by the central laboratory with onset at least 14 days and at least 28 days after vaccination. In the overall study population, vaccine efficacy after 14 days was 66.9% with a lower bound of 59 and 66.1% with a lower bound of 55 after 28 days. Efficacy for the two pre-specified age cohorts of 18 to 59 and 60 years and older were comparable with the efficacy in the overall population. For the subgroup analyses, centrally confirmed cases as well as cases with positive local PCR, which are still awaiting confirmation by the central lab, were included. As shown here, efficacy was similar across different age groups and sex. There was some variation in efficacy across geographic regions, which will be discussed in further detail in a later slide. Efficacy across race and ethnicity were generally consistent with that observed in the overall study population, but a small number of participants and cases in some racial groups resulted in large confidence intervals around the point estimate and limited interpretation of those results. When looking across age groups and comorbidity, there was a lower efficacy point estimate observed in the subgroup of participants 60 years of age and older with comorbidities with onset at least 28 days after vaccination. However, the confidence intervals are wide and the uncertainty of the point estimate is large. This wide confidence interval may be attributable to a lower number of cases due to the shorter follow-up duration in this subgroup compared to the other subgroups, and with a slightly greater proportion of participants in the subgroup who were unblinded due to eligibility for an authorized COVID-19 vaccine under EUA compared to the younger cohorts. There was also overall a smaller number of participants in this subgroup compared to the other subgroups. The VE estimate increased and the confidence interval narrowed as the number of cases included in this analysis increased. For example, when looking at the VE at the 14-day endpoint, which included all cases in the 28-day endpoint, and when comparing centrally confirmed cases only to this analysis, which includes non-centrally confirmed cases as well. This indicates that the apparent lower VE in this subgroup potentially reflects imprecision associated with a smaller number of cases. There was a lower efficacy estimate for participants with comorbidities compared to those without, but only for the endpoint of onset at least 28 days post-vaccination, which, similar to what was discussed in the last slide, may be reflective of a smaller number of cases. For a majority of individual comorbid conditions, interpretation of the results is limited by the small sample size, low incidence of COVID-19 in the subgroup. However, for subgroups with higher incidence of COVID-19, such as in participants with obesity, the VE appeared to be similar to the VE estimate in the overall study population. The study included a little more than 4,000 subjects who were SARS-CoV-2 seropositive at baseline. The number of cases observed in this subgroup was small, and at this time, there is insufficient data to evaluate vaccine efficacy in previously infected individuals. Efficacy against any symptomatic COVID-19, including mild disease, and efficacy based on less restrictive case definition, the FDA harmonized case definition, with onset at least 14 days or 28 days after vaccination, were overall similar to results obtained for the primary efficacy analysis of efficacy of moderate to severe critical COVID-19. There were only 4 central confirmed mild COVID-19 cases, 1 in vaccine group, 3 in the placebo group, with onset at least 14 days post-vaccination, indicating that the moderate to severe critical primary efficacy endpoint definition captured almost all cases of symptomatic COVID-19. Vaccine efficacy against centrally confirmed adjudicated severe critical COVID-19 cases in the overall study population with onset at least 14 days after vaccination was 76.7% with a lower bound of 54.6 and 85.4% with a lower bound of 54.2 after 28 days. Point estimates of efficacy against severe critical disease were lower with wide confidence intervals in participants 60 years of age and older compared to the younger participants when evaluating only centrally confirmed cases. When non-centrally confirmed cases were also included, the VE estimate for participants 60 years of age and older increased and the confidence interval narrowed and was more similar to the efficacy estimates for the younger cohort and the overall population. The endpoint of COVID-19 requiring medical intervention is defined as participant requiring hospitalization, ICU admission, mechanical ventilation, and/or ECMO. A post-hoc analysis of all COVID-19 related hospitalizations was performed by counting all hospitalizations recorded in medical resource utilization forms, SAE forms, and clinical event listings such as during a severe critical COVID-19 episode. In the setting of a positive PCR at the onset of the COVID-19 episode or onset of the adverse events. These data indicate vaccine efficacy in the prevention of severe COVID-19 requiring medical intervention, with no COVID-19 related hospitalizations seen in the vaccine group following 28 days after vaccination. In the subgroup of participants 60 years of age and older with comorbidities, after 14 days post-vaccination, 2 out of the 22 moderate to severe COVID-19 cases in vaccine recipients resulted in hospitalization, both prior to 28 days, compared to 11 hospitalizations out of 63 cases in the placebo recipients, with 5 occurring after 28 days. As of February 5th, 2021, which is an additional 2 weeks after the primary analysis data cutoff date, there were a total of 25 deaths reported in the study, with 5 in the vaccine arm and 20 in the placebo arm. Of these, there were 7 COVID-19 related deaths, all in participants in the placebo group and from study sites in South Africa. 1 death was in a participant who was SARS-CoV-2 PCR positive at baseline and who developed symptoms 10 days after vaccination. As you can see in this table, all subjects had one or more comorbidities, which may be associated with an increased risk of more severe COVID-19. Asymptomatic infection was defined as a participant who does not fulfill criteria for suspected COVID-19 based on signs and symptoms and has a positive SARS-CoV-2 PCR or develops a positive serology based on SARS-CoV-2 nucleocapsid serology during the study. There's no scheduled PCR screening specified in the protocol, so most of the data will be based on N serology collected at scheduled time points. Based on available day 29 N serology data, efficacy for this endpoint was modest with wide confidence interval that crosses 0. Day 71 N serology results are only available from a subset of approximately 2,800 subjects, which is a very small proportion of the overall study population. Available serology is also not evenly distributed across demographic groups and geographic locations. Data is limited at this time to make a conclusion about vaccine efficacy against asymptomatic infection. And also note that the protocol specified this endpoint will only be evaluated once 15,000 samples from day 71 are queued. So what is done has been an interim analysis. During the conduct of the study, from study initiation on September 21st through the data cutoff date of January 22nd, new SARS-CoV-2 variants emerged in geographic regions where the study took place. In a subgroup analysis of vaccine efficacy against moderate to severe critical COVID-19 in the United States, South Africa, and Brazil, there was lower efficacy observed in South Africa compared to the United States. Vaccine efficacy against severe critical COVID-19 was comparably high across the 3 countries, although there was a wide confidence interval around the point estimates for the United States and Brazil. During sequencing of COVID-19 cases in the study to inform the vaccine efficacy analysis by region is incomplete at this time. As of February 12th, 71.7% of cases which have been centrally confirmed by the data cutoff date of January 22nd have been sequenced. Only samples with a sufficient viral load were able to be sequenced. Prioritization for sequencing was given to moderate to severe critical cases and cases with onset at least 14 days after vaccination. As of February 12th, there were no cases identified from the study to be from B117 or P1 lineages. In the United States, 73.5% of cases have been sequenced, of which 96% were identified as the SARS-CoV-2 Wuhan H1 variant D614G. In South Africa, 66.7% of cases have been sequenced, of which 94% were identified as the B1351 variant. In Brazil, 69% of cases have been sequenced, of which 69% were identified as a variant of the P2 lineage, and 30% were identified as the Wuhan variant. Because strain sequencing of COVID-19 cases in the study is incomplete at the time of this analysis, and due to selection bias involved in prioritizing the cases to be sequenced first, vaccine efficacy against specific SARS-CoV-2 variants cannot be evaluated at this time. So in summary, the data from the primary efficacy analysis with a cutoff date of January 22nd, 2021, and a median follow-up for efficacy of 8 weeks post-vaccination met the pre-specified success criteria established in the study protocol. Efficacy of the vaccine to prevent protocol-defined moderate to severe critical COVID-19 occurring at least 14 days after vaccination was 66.9% with a lower bound of 59 and 66.1% with a lower bound of 55 at least 28 days after vaccination. Efficacy against the key secondary endpoint of prevention of severe critical COVID-19 was 76.7% with a lower bound of 54.6 for onset at least 14 days after vaccination, and 85.4% with a lower bound of 54.2 for onset at least 28 days after vaccination. There was efficacy against COVID-19 requiring medical intervention, with 2 COVID-19 related hospitalizations in the vaccine group after 14 days compared to 29 in the placebo group. After 28 days, there were no COVID-19 related hospitalizations in the vaccine group compared to 16 in the placebo group. Efficacy outcomes across demographic subgroups were generally consistent with the efficacy seen in the overall study population. Although vaccine efficacy in participants 60 years of age and over was overall similar to observed in the younger participants, a lower efficacy estimate against prevention of COVID-19 with onset at least 28 days was seen in a subgroup of participants 60 years of age and older with comorbidities. However, for this and several other subgroups, The VE estimate increased and the confidence interval narrowed with inclusion of more cases, indicating that the observed result potentially reflects imprecision associated with a smaller number of cases in this subgroup. The vaccine was effective in reducing COVID-19 related hospitalizations in this subgroup. Finally, there was country-to-country variation in vaccine efficacy in the setting of different predominant variant strains circulating around the time of the study, but the confidence intervals were overlapping, and efficacy was more similar across countries when evaluating the endpoint of prevention of severe critical COVID-19. So with that, I will turn it over to Dr. Hector, who will take you through the rest of the presentation.
Thank you, Dr. Zhang. I want to start by going over the safety monitoring in Study 3001. This is a graphical depiction of the monitoring throughout the study. Solicited adverse reactions were collected via an e-diary for 7 days after vaccination in the safety subset, which included 6,736 participants. Unsolicited adverse events were also collected in the safety subset for 28 days after vaccination and recorded in the electronic case report form at the day 29 visit. Medically attended adverse events are captured through 6 months post-vaccination. Serious adverse events and adverse events leading to study discontinuation are captured through the entire study period. In addition, spontaneous reports of AEs to investigators, regardless of seriousness or severity, were recorded in the case report form at any time. The arrow in the middle of this slide marks the approximate safety evaluations completed prior to the data cutoff point. Here we have the disposition in the safety population. This is overall similar to what was previously presented by Dr. Zhang for the efficacy population. Follow-up duration, unblinding to treatment, and discontinuations occurred at similar rates between treatment arms. The safety subset, the population for analysis of solicited and unsolicited adverse events, was about 15% of the total safety population. 99.9% of individuals in the safety subset completed follow-up through day 29. Subjects in the safety subset were enrolled in 3 Tier 1 countries. The Tier 1 countries were selected based on rapid startup capacity and projected incidence rates for COVID-19 that would allow for rapid efficacy signal detection. At the site level, investigators questioned participants on their willingness to be part of the safety subset. Selection and randomization of the participants was then completed through a web-based randomization system. The demographics of the subset were similar to those of the entire safety population, the full analysis set, with respect to sex and age. However, a larger percentage of participants in the safety subset were white, 83.4%, compared to the full analysis set, 58.7%. Geographically, the safety subset was limited to the participants in the United States, South Africa, and Brazil. Fewer participants in the safety subset compared to the full analysis set were seropositive at baseline, 4.5% compared to 9.6%, and had at least one comorbidity, 34.1% versus 40.8%. Here you can see the rates of solicited local reactions broken down by age group. The most commonly reported solicited local reaction was pain. Grade 3 events were rare for solicited local reactions. Overall, there was a lower rate of solicited reactions in the older cohort compared to the younger age group. Among participants in the vaccine group, the overall rate of local adverse reactions was similar between those who were seronegative for SARS-CoV-2 at baseline and those who were seropositive. The same was true for each individual local adverse reaction. Here are the rates of solicited systemic reactions, also broken down by age group. The most commonly reported solicited systemic reactions were headache, fatigue, and myalgia. Few systemic reactions were grade 3. Again, you can see that the rates were higher in the younger cohort compared to the older cohort. As with solicited local reactions, for systemic reactions in vaccine recipients, there was no significant difference among those who were seropositive or seronegative at baseline. This is a broad overview of all unsolicited adverse events collected through the protocol-specified methods. Overall, rates of medically attended adverse events and serious adverse events were balanced between groups. This remained true when you look by age cohorts as well. Through the data cutoff, 19 deaths were reported, with 3 in the vaccine group. There were no vaccine-related deaths. There were also no AEs that led to study discontinuation. Unsolicited AEs within the safety subset were reported at similar rates in the 28 days following vaccine. 13.1% in the vaccine group and 12% in the placebo group. This table shows the breakdown of unsolicited AEs in the first 28 days in the safety subset by system organ class and preferred term. Events that occurred in at least 1% of vaccine recipients are included. By preferred term, the most commonly reported unsolicited adverse event in the vaccine group was chills. As this was not recorded as a solicited adverse reaction, this may represent vaccine reactogenicity. FDA conducted standard measure queries, or SMQs, using FDA-developed software to evaluate for constellations of unsolicited adverse events. Hearing loss associated with tinnitus on day 34. Hearing improved and their event resolved by day 69. Causality assessment of these tinnitus events was also confounded by the presence of risk factors in participants in Study 3001. In FDA's assessment, the vaccine cannot be excluded as a contributing factor to these events. Overall, rashes were reported more frequently in vaccine recipients than placebo recipients. Urticaria specifically was reported in 5 vaccine recipients compared to 1 placebo recipient in the 7 days following vaccination. Moving to serious adverse events. Per the study's investigators, 7 serious adverse events were considered related to the vaccine. I will go through FDA's assessment of these events. In FDA's assessment, 3 of these events were considered likely related to the vaccine. The first is a 42-year-old male who experienced hypersensitivity following the vaccine. On day 3, he began to have urticaria, which became more diffuse over the following days. On day 5, he had angioedema of the lips and reported sensation of an itchy, tight throat. He did not experience any respiratory distress and he was not hypoxic. The event did not meet Brighton criteria for anaphylaxis. The second event was pain in the injected arm on day 1, which progressed to include more of the upper extremity. Electroconductive studies on day 15 showed intact sensory and motor nerves in the affected region and no denervation of muscles. The subject's symptoms, as well as assessment, were ongoing at the time of data cutoff. The third event was extreme generalized weakness in a 35-year-old male associated with multiple systemic symptoms, including fever and headache. The subject was hospitalized for evaluation, where a mild elevation in CPK consistent with myositis was noted, and a demyelinating disorder was excluded. Symptoms resolved by day 4. 4 SAEs were considered to have an indeterminate but not likely relationship to vaccine in FDA's assessment. This included 2 cases of facial paralysis considered SAEs on the basis of medical importance by the investigator. These occurred on days 3 and 16. 2 events of facial paralysis were also noted in the placebo group in a similar time frame on days 2 and 29. 1 additional case of facial paralysis was reported in the vaccine group on day 19. However, review of the clinical information revealed no facial asymmetry as well as intact cranial nerves, making the diagnosis of facial paralysis unlikely. Reports of Guillain-Barré syndrome were also balanced between vaccine and placebo arms, with 1 event happening in each group on days 16 and 10 respectively. There was a case of pericarditis in a 68-year-old male 16 days following vaccination. No etiology for the diagnosis was determined. While the vaccine cannot be excluded as a cause of this event, review of the AD26 safety database did not reveal cases of pericarditis or carditis. In addition to the SAEs already mentioned, yesterday, Janssen reported to the FDA that an anaphylactic reaction, the details of which are still under investigation, had occurred in an individual who received the Janssen COVID-19 vaccine as a participant in another ongoing study. Prior to the data cutoff, 19 deaths were reported, with 3 in the vaccine group and 16 in the placebo group. An additional 6 deaths were reported between January 22nd and February 5th. 2 in the vaccine group and 4 in the placebo group. None of the deaths were considered related to the vaccine. The deaths in the vaccine group were as follows. a 61-year-old was diagnosed with pneumonia on day 13 and died on day 24. A 42-year-old individual with HIV was hospitalized with a lung abscess and died on day 59 following prolonged hospitalization. And a 66-year-old woke up with shortness of breath on day 45 and died prior to EMS arrival. No autopsy was performed and the cause of death is unknown. The two deaths following data cutoff were also considered unrelated and included an individual who collapsed at home and an individual who died from decompensated heart disease. In the placebo group, 6 deaths prior to data cutoff and 1 following were related to COVID-19. Study participants with childbearing potential were screened for pregnancy prior to vaccination. Participants were excluded if they were pregnant or planned to become pregnant within 3 months of vaccine administration. 8 pregnancies were reported through the January 22, 2021 cutoff, 4 vaccine and 4 placebo. In 7 participants, vaccination was within 30 days of the last menstrual period, and in 1 vaccine recipient, vaccination was prior to LNP. Among vaccine recipients, 1 spontaneous abortion and 1 ectopic pregnancy was reported. 2 pregnancies were ongoing with outcomes unknown at this time. To summarize the safety data, the information provided by the sponsor was safety data available in greater than 42,000 participants with a median safety follow-up of 58 days was adequate for review and to make conclusions about the safety of the Janssen COVID-19 vaccine in the context of the proposed indication on population for intended use under EUA. Reactogenicity, particularly injection site pain, headache, fatigue, and myalgias, were frequent but mostly mild to moderate. Overall, less reactogenicity was seen in the age cohort 60 years and older. A single SAE of hypersensitivity was seen in the vaccine group, as well as more cases of urticaria. Finally, we noted numerical imbalances in thromboembolic events and tinnitus. The assessment of causality for these events was confounded by the presence of risk factors in participants, and vaccine cannot be excluded as a contributing factor to these events. We will now move to the pharmacovigilance plan and future studies. Janssen submitted the pharmacovigilance plan to monitor safety concerns that could be associated with the AG26-CoV-2 vaccine. Important potential risks include vaccine-associated disease, anaphylaxis, and thromboembolic events. Important missing information includes use in pregnancy and lactation, use in immunocompromised individuals, use in individuals with autoimmune or inflammatory disorders, use in frail individuals with comorbidities, interaction with other vaccines, long-term safety, and use in pediatrics. Pharmacovigilance activities include adverse event reporting. Adverse event reporting under the EUA may come from vaccine recipients, vaccination providers, or the sponsor. First, vaccine recipients will be notified that AEs can be reported to theirs through the fact sheet for recipients and caregivers. Another source of AE reports from recipients is the vSafe program, which is a smartphone-based program that uses text messaging and web surveys from the CDC to check in with vaccine recipients for health problems after vaccination. Reports from vaccine recipients are voluntary. AE reporting by vaccine providers and the sponsor is mandatory. Both the sponsor and vaccine providers administering Janssen COVID-19 vaccine must report to theirs the following information associated with the vaccine: vaccine administration errors, whether or not associated with an adverse event, serious adverse events irrespective of attribution to vaccination, cases of multi-inflammatory syndrome in children or adults, and cases of COVID-19 that result in hospitalization or death. In addition, the applicant will also conduct periodic aggregate review of safety data and submit periodic safety reports at monthly intervals for FDA review. Each periodic safety report is required to contain a narrative summary and an analysis of adverse events submitted during their reporting interval, including interval and cumulative counts by age group, special population, and adverse events of special interest, newly identified safety concerns in the interval, and actions taken since the last report because of adverse experiences. Both FDA and CDC will take a collaborative and complementary approach on reviewing AEs. FDA will individually review all serious adverse events on a daily basis. FDA will also examine other sources for AEs, such as the literature, and will perform data mining to determine if AEs are disproportionately reported for the candidate vaccine compared to all other vaccines in theirs. Any potential safety signals identified will be investigated. The sponsor provided a description of studies they are currently planning on conducting. These studies include completing of long-term follow-up from ongoing clinical trials, as well as the following active surveillance studies. A pregnancy study, which will be conducted as a multi-country observational prospective cohort study of pregnant women vaccinated with Ad26-CoV-2S in order to assess the occurrence of obstetrical neonatal outcomes among women administered the vaccine during pregnancy. An active surveillance study of safety, which will be conducted as a retrospective observational propensity-scored match cohort study using health insurance claims and electronic health records in order to assess the risk of developing pre-specified adverse events of special interest during specific risk windows following administration of the Janssen COVID-19 vaccine. An active surveillance study of effectiveness will also be conducted as a retrospective observational propensity-scored match cohort study using health insurance claims and electronic health records in order to estimate the effectiveness of Ad26-CoV-2S to prevent medically attended COVID-19 in individuals who are vaccinated according to national immunization recommendations. The sponsor has provided protocols and milestone dates for these studies, and FDA is reviewing the protocols and will provide feedback. Finally, I will review the overall risk-benefit assessment. The benefits of the Janssen COVID-19 vaccine include reduced risk of symptomatic COVID-19 14 days following vaccination, as well as reduced risk of severe critical COVID-19 at least 14 days following vaccination. This includes a reduction in COVID-19 related deaths and hospitalization. The efficacy against severe critical disease was similar across demographic regions. Overall, the efficacy was generally consistent across demographic groups. Finally, as the vaccine is administered as a single dose, it may provide operational benefits to mass vaccination campaigns. The risks associated with the Janssen COVID-19 vaccine include reactogenicity, especially injection pain, headache, fatigue, and myalgias. An SAE of hypersensitivity as well as non-serious urticaria were reported and likely associated with the vaccine. Finally, the vaccine cannot be excluded as a contributing factor to thromboembolic events and events of tinnitus. There remain several data gaps and areas of unknown risk, including the duration of protection, efficacy against asymptomatic infection, transmission, and new variants, safety in subpopulations such as pregnant and lactating women, pediatrics, immunocompromised individuals, and individuals with SARS-CoV-2 infection, and information on adverse events that are uncommon or need longer follow-up to detect. Finally, at this time, the evidence suggests risk of vaccine-enhanced disease is low, but a longer follow-up duration is needed to fully assess this risk. With that, I will conclude with the voting question for the VRPAC members. Based on the totality of the scientific evidence available, do the benefits of the Janssen COVID-19 vaccine outweigh its risks for use in individuals 18 years of age and older? And that concludes my presentation.
Thanks to both of you. I must compliment you on not only on the presentation, but on the briefing document, which was very comprehensive and easy to follow. Before we go on to a few questions, we're going to have a break, and then we're going to continue in the first part of the discussion with a question period for both you, if the members wish to do so, and for others. I wanted to ask you, Dr. Zhang, specifically about the difference between the endpoints, moderate and severe, versus the harmonized case definition. There seemed to be only 4 cases that were different, 4 additional cases. So can we conclude that really the moderate and severe case definition is just about equivalent to all symptomatic cases.
Yes, you're correct, Dr. Monto. When looking at mild cases, centrally confirmed mild cases, there are only 4 additional cases included. So really the efficacy estimates for moderate to severe or based on any severity or based on the FDA harmonized definition was basically the same.
Okay. Could I ask?
Okay, I don't know if my video is on.
That's okay. Go ahead, Dr. Miser.
Okay.
We can hear you.
Okay, thank you. First of all, I want to make an acknowledgement, and then I have a question for the FDA. First of all, get myself back in here. First of all, I want to second Dr. Monto's comment about the FDA. This has been an extraordinary amount of data that has been digested. I believe it was submitted to the FDA on February 4, so that's 22 days ago. And to get all of this data in such a presentable form is remarkable, and I just want to thank you all for what you've done, Dr. Zhang and Dr. Hefton and everybody else at CBER. Second point I'd like to make briefly is there were comments in the public hearing about wide confidence intervals in some of the endpoints. But remember, that's inevitable. This was a very large study with over 43,000 subjects enrolled and randomized. And as you break down each group by smaller and smaller numbers, the confidence intervals are going to be wider because there will be fewer events. So in view of the urgency of having information about an additional fact, I think the Janssen team has done a remarkable job in getting this data to us and to the FDA. And I think in particular, their representation of different races in the trial is really, I think it's the best that we've seen among the 3 submissions. So all I can say is I think a lot of people have done a remarkable job. The question I have relates to study 2001, and Dr. Zhang, I think maybe you can comment on this. There were 660 individuals 12 to 17 years of age who were enrolled in that safety and immunogenicity study. And I didn't see any suggestion of additional attempts to generate efficacy data. And can you comment on how the FDA is going to deal with that information? Is that, my concern is that should not be a basis on which an EUA should be administered to adolescents, or at least I think we have to exercise caution in using that as a sufficient basis to address an EUA. Over. Thank you.
Thank you for that question, Dr. Meisner. So just to clarify, the study 2001, the adolescent cohort has not yet been enrolled. So that has those subjects have not been included in the study yet. And that study is, you're correct, it's just a safety and immunogenicity study with secondary exploratory endpoints for efficacy to be included. And of course, we're still in discussion with all the sponsors about pediatric plans and what will be the basis for the indication down to those age ranges.
Okay, thank you. I'm sorry, who was just speaking?
This was Rachel Zhang.
Okay. Oh, okay. I guess it's a delay here,
Dr.
Rachel.
Okay, let's move on to Dr. Hildreth.
Yes. I have a question about the percentages of American Indians and Alaska Natives included in the trials. According to the data from the sponsor, on a global basis, there were 10% of such individuals enrolled. When they showed the data for the U.S., it was only 1%. And I don't really understand how that can be. They can have 10% on a global level, and it drops by 90% when you look at the U.S. itself. So can you explain that disparity to me, please? Thank you.
Thank you for that question. So yes, so I think that the terminology may not be totally encompassing, but the American Indian Alaska Native population shown in the slide includes the American Indian population in Latin America countries as well. So those populations in South America and Central America as well. So of those 1%, you're right, came from the U.S. study sites, and the rest are from mostly the South American, Central American sites.
Okay, thank you.
Okay, 2 more questions, and then we're going to go into our break. And at the discussion, when the discussion starts, we'll have a free-for-all. We'll be asking questions, both of you, FDA group, and also of the sponsors. So that's the only way we can attempt to stay on schedule. So next, Dr. Rubin.
Hi, thanks. That was a terrific presentation, and I echo what everyone else said. Just a quick question about the testing. The PCR test isn't that complicated, and is it so important to have centrally confirmed testing? How often were there discrepancies between what was acquired locally and the central adjudication?
Thank you for that question. So yes, the protocol was set out that way, that all the tests should be shipped to University of Washington for central confirmation. When looking at the tests that had already undergone that central confirmation process, there was a high concordance rate in the test. So those tested positive in the local laboratory and those in the central laboratory, 90.3%. So as mentioned in the presentation, most of the discordant results were those samples with very low viral loads, and probably with a prolonged shipping time from some regions where the study was conducted to the University of Washington, the freeze-thaw cycle might have impacted those samples as well. But all of the tests used in the local laboratories were all FDA authorized tests as well.
Okay, before the break, we'll hear Dr. Sawyer.
There you go, Dr. Sawyer. I muted you. There you go. Go ahead.
My question relates to a slide in it's on the same topic of the PCR testing. Relates to a slide in Dr. Zhang's presentation, as well as Table 18 in the briefing document. Although there's concordance, as you just stated, in general, between the testing done at the sites and the centrally confirmed testing, in this table, all of the differences appear to be in the placebo group, which seems a little bit odd. Are we to interpret in general that one of these tests is more sensitive and the other is less specific, or how are you interpreting the differences?
I'm sorry, I have to find the table you're referring to, but the tests, as I mentioned, both using the local laboratory and at the central laboratory, University of Washington, are all FDA authorized and 99.9% with sensitivity and I think similar specificity as well. So in terms of the samples analyzed, like I mentioned in the presentation and in our briefing document, there was some selection bias involved in which samples got sequenced first. So again, those samples with more severe cases, samples that had onset 14 days or after, so like choosing those samples that would have met the primary endpoint basically, were selected to be sequenced first to try to generate as much data as possible. So because of the selection bias in prioritizing which samples were sequenced first, oh sorry, now I'm talking about something else. I'm talking about PCR results. So no, I apologize. I have to find the table that you're talking about, but I don't remember there being a very big difference between the vaccine and placebo groups in terms of PCR testing results.
Okay, thank you. We're going to take a 10-minute break now. When we come back, we're going to be hearing from Dr. Peter Marks before we go into a general question and answer session. And I hope the Janssen team is also going to be available because the committee has residual questions they want to ask. So 10-minute break. Thank you.
All right, everybody. All right, just be a second as we are going to come back. All right, welcome back to our 164th meeting of the VRPAC Advisory Committee. So, Arnold, Dr. Monto, I'm going to hand it back to you.
Right, and it's my pleasure to introduce Dr. Peter Marks, the head of the Center for Biologics Evaluation and Research, familiarly called CBER. Dr. Marks.
Thanks very much, Dr. Monto. So I just want to take a moment here to, for those who might not have been here this morning, to thank everyone once again. Really want to thank the committee members for the time in going through a lot of data. Also want to thank the sponsor for a very clear presentation and for their participation in this process, our public speakers, and the FDA staff who really have worked tirelessly over the past month after working the month before and the month before with these various emergency use authorizations. So tremendous amount of work and tremendous amount of thanks to all of them. Please let me also call out the advisory committee staff. Dr. Atreya and her staff have done an incredible job putting together all of the logistics here to make this happen. A lot of planning has to go into these meetings, and then having them get executed is really a lot of work. So thank you so much for all you've done here. Just to finish off here, I just want to thank everyone from the committee. We'll look forward to a very robust discussion this afternoon. We're looking forward to hearing the committee's dialogue, and I'm going to just turn it over to Dr. Monto now. Thank you so much, and have a good rest of afternoon discussion.
Right, thank you, Dr. Marks. We're going to try to have some question and answers for a limited time. We'll say about 10 minutes to finish up the questions on the FDA report, and then about 20 minutes questions for the sponsor that are hanging over since their presentation. So let's just go ahead, and I see Dr. Fuller's got her hand raised. Dr. Fuller.
Amazing. I'm first in line. Look at that. Thank you, Dr. Monto. So this is actually...
Sometimes you're lucky.
Look at that, yes. This is for the Janssen group as well as for Dr. Marks. And I asked this question this morning. Clearly, the reduction of disease and moderate to severe to hospitalizations is critical for ending this pandemic. But in the long run, we really do have to stop infection in order that we don't give the coronavirus the many opportunities to mutate and to actually adapt to get to a best fit for itself. So I want to commend Janssen for doing the global study, including people around the world. But we know that nobody's going to be safe until we're all able to shut down the virus replication. So in that light, what is happening with both the Pfizer and Moderna studies to follow the asymptomatic infection and shedding, as well as for Janssen, what are you going to do? I think that was in your table 7.166. Could you tell us if you got additional data or you're planning, what studies you're planning to follow the vaccine effect also on infection asymptomatically and shedding? Important for our school systems and so many things.
If I can interrupt, this gives me the opportunity to give us all an admonition. We are reviewing the Janssen vaccine here. So we need to be very specific in what we are looking at. So when getting answers to your question, let's talk about what is going on with this vaccine. So FDA, I think you mentioned.
Vaccine.
Right. this vaccine, right?
Yes, it includes what's going on in the...
I understand there are global questions, but that's not our remit right now. Rachel?
All right. So I'll actually defer to Janssen to talk about what their plans are in terms of asymptomatic infection and transmission studies.
Mr. Chairman, shall I comment on this question?
Yes, please.
Yeah, so as
we
already
try to be
outlined,
orderly, but things never turn out to be orderly during the discussion.
no
please,
problem at
go
all. So as already discussed in the presentation, the way that we want to address this is to look at non-symptomatic seroconversion against N protein. As indicated, we do have preliminary data on that that actually are limited to about 1,300 samples in the placebo group and 1,300 samples in the active group. And where we do see that we have, depending on how you look exactly at it, about 70%, we observe 70% efficacy. However, we want also to stress that these are preliminary data to the point that was made also by the colleagues from FDA. It's a limited number of samples, and also in the study protocol, we have planned to do this once we have at least 15,000 people, and we look at the time point between day 29 and month 6. So it is certainly part of our plan to continue to do this and to take those samples and to look at asymptomatic infection. But the preliminary data that we have today encourages from two perspectives. On one hand, it suggests, if it's confirmed, that indeed it has impact on, or it prevents asymptomatic infection to a large degree. And the other point is that sometimes it was hypothesized that eventually you can shift all of your symptomatic patients into the non-symptomatic, so people without symptoms, in which case you could eventually even increase transmission, and certainly that seems not to be the case. So concretely, we have an active plan. We will do that based on seroconversion between day 29 and month 6, and the preliminary data we have are very encouraging.
All right,
thank you.
That's important to know.
Dr. Levy. Dr. Levy, please.
Yeah. I wanted to thank the FDA representatives for their most recent presentation at this hearing. I had a general question. For some of the imbalances seen with adverse effects, such as tinnitus, for example, I'm wondering whether FDA also looked at the broader pool of data for the ADVAC 26 platform, and whether that gave any guidance as to whether such imbalances were seen in other vaccine studies with the same adenovirus 26 platform. Thank you.
Hi, yeah, thank you for that question. So we did look across for tinnitus across the development program of the COVID-19 vaccine. Because it's reported as an AESI, it doesn't come up as a serious adverse event throughout, but there were no SAEs that were notable, that were reported aside from the one that I had previously mentioned that occurred in the phase 1 study, and then one additional event of tinnitus that occurred in the ongoing 2-dose study 3009 that remains blinded. I would defer to Janssen to report on the larger safety database for all of the Ad26 vaccines.
We have indeed looked.
Go ahead.
No, I just wanted to it was suggested to look into the platform database. We have actually done a screen on that particular phenomenon, tinnitus, and we have not seen that previously with the platform.
Thank you.
Okay, thank you. Dr. Chatterjee.
Arnold, we had a little, we have some question, and just to clarify, the questions now, right now, are for FDA, the FDA portion, correct? That's where we're focused right now?
We're trying to do that.
Okay, so if the FDA representatives could both keep their cameras on, that would be great, and that would help guide the, there we go, thank you.
To discipline the committee is rather difficult, so. Hey, Dr. Chatterjee, is this for FDA?
Yes. So
Okay.
my question is, I had trouble with the briefing document trying to distinguish between where the cutoff was for severe disease. Was it, because the way the data were presented, it was moderate to severe and then severe critical. And so I couldn't figure out, is this a continuum? Where's the cutoff?
Thank you for that question. Yes, there is a cutoff, and in our briefing document and in one of the slides in my presentation, there is a separate analysis for just severe critical disease. And as I mentioned in the presentation, to meet that severe critical disease endpoint, it has to undergo adjudication by a blinded independent adjudication committee. And what their decision is based on when they review the data, that is the final determination of whether that case is severe or not. So it is a distinct endpoint.
So when they're talking about moderate to severe disease, that is separate from the critical, severe critical, right? And that's dependent on the adjudication committee. Is that correct?
The moderate to severe disease endpoint includes all the severe disease.
Okay, got it. Thank you.
Okay, Dr. Lee, is this for FDA?
Yes. Yes,
Okay.
sir, it is. So my question to the FDA is, I did notice in the document that you indicated about 70% or over 70% of the, you have done strain sequencing on that. And so I think one of my questions is, obviously we haven't seen that before. When we see these results again at some point, will we be able to see the efficacy by strain? Because obviously there's a lot of interest in that. Thank you.
So I will defer to Janssen to talk about their plans for finishing up the sequencing and what kind of analysis we can get.
Hey, we try
Thank
to
you.
segregate by, but it doesn't work. Dr. Van Hoek.
Thank you for that question. Indeed, it is our aim to continue the sequencing of the cases that we observe. Sometimes there might be challenges because you need to have a minimum amount of viral load to make reliable sequencing, but we certainly want to get it at a higher percentage than what we have now. And all of that would be intended then to be sufficient to do strain efficacy analysis, and it's our intent to include this in the BLA we plan to submit later this year.
Okay, thank you.
Dr. Gantz.
Thank you so much. My camera doesn't seem to be working. I wasn't
still
clear.
hear
I wasn't clear who we could ask questions of and who may have this data. So if it's all right, I'd like to go ahead and ask my questions, and then people can defer to whomever. Is that all right?
That's okay.
Okay, thank you. So I had,
disciplining the group is impossible. So.
Yeah, it's just hard because we don't know the timeline. Anyway, there is, as been mentioned a couple of times, a little bit of discordance between the immune responses. And the real questions are, if an individual doesn't have an antibody response, which was some of the individuals in there, do they have a T-cell response? So in essence, is there individuals who have no response to the vaccine, and what percentage of it? I assume that it's low. And in that vein, as we're trying to understand the immunity to these vaccines and how that pertains to the efficacy of the vaccines, is there any immune data from those who had infection? So particularly those who were hospitalized or had more severe. Were there actually attempts at getting further immunologic studies on them so we can understand, had their antibodies dropped, had their T-cell responses dropped, and in addition, are those being correlated to whether or not people actually had whatever levels of antibodies? I think there was some very brief mention of there was some correlates of the level of antibody and then disease, but that would be important to know so we understand in the future how to boost individuals moving forward. And I did hope that I hear a lot about pediatric studies. I know they're in the works. If we could get a better timeline on that, because we're getting a lot of questions about when these studies may then be completed. Thank you.
I think that this is for Dr. Van Hoek.
Thank you. Your first question related to the immune readouts and the correlates of protection, and that certainly is a very critical question. That is also part of the objectives of this trial. It's actually a work that is planned to be done in collaboration with NIAID and with Professor Peter Gilbert, who is specialized in doing correlates of protection. That work has started, but it's still work in progress, so too early to report back on. We have seen in our non-human primate studies that there was a correlation between the neutralizing titers and the protection, although this was not absolute, which might imply indeed that the cell-mediated immunity is an important component. And again, there the future will need to tell us going forward. In general, with the platform, we have seen that usually good responders on the humoral side are also good responders on the cellular side, although sometimes you do see discordances.
Okay. Dr. Hildreth. You're muted.
Sorry about that. Thank you, Dr. Mondo. I'd like to follow up on a comment that Dr. Poland made about the breakthrough infections of those who got the vaccine being milder. And can you refer us or me to the data that confirms that the breakthrough infections had a milder disease when they got the vaccine?
It's actually a figure which is in the briefing document, and it's figure 16, page 65, which actually looked at the amount of symptoms that people have when they have breakthrough infections, and you see that in general, if there are breakthrough infections, people present with less symptoms.
Page 65?
Yeah, and this is, I don't know if our team could pull up the slide that actually shows this. Let me...
I think that's a fairly important point that needs to be made when this is discussed with the public, to know that the breakthrough infections were milder. That seems a very important point that needs to be made. So
there's some technical challenge because I see it in the system that it is up, but I don't see it appearing in the Adobe.
Okay, I'll find it.
No, he's we're waiting for Ted to connect his Ted to connect his, but that's all right.
I'll find it. Thank you.
Arnold, go ahead. Arnold, just as a comment that came in, just so we can keep on track. Is it open now to both? Like I said, they wanted to know because we are.
I capitulate. I think it's open to both. Is that there any? Let's do this. Are there any questions specific to the FDA? And then we can really focus on questions for the next few minutes for the sponsor. So any of those 4 or 5 people who have their hands raised right now, who really want to talk to the FDA.
Go ahead and
Dr.
unmute then
Perlman,
if you want to talk.
talk to the FDA, you're next.
Dr. Kim.
Kim, okay.
Dr. Kim, don't forget to unmute your own phone.
Still muted.
Your phone. Not your camera, your phone, Dr. Kim. Nope, not yet. All right, let's go to one of those while Dr. Kim's getting his phone. He muted himself in his phone. That's all right. Oh, you there, Dr. Kim? Go ahead, Doctor. Go ahead, Dr. Monto.
Okay, Dr. Moore, I'm just going to go down the list from now on. I've given up. I tried it.
I have a question that I'd like to ask generally the FDA because I don't know the answer.
Okay,
to this.
good.
And you may not know the answer to this either, but live adenovirus vaccines are very old, right? They were invented in the 1960s.
The military has used them a lot.
I'm sorry?
I said the military has used adenovirus vaccines a lot.
Right, and my impression is that they have a tremendous safety profile, but I'm not expert on this. Is there anything that we know from the history of adenovirus-based vaccines that we should be particularly worried about? Because now we're taking two things that we know something about. One is adenovirus vaccines, and two, SARS-CoV-2 spike protein vaccines, which at least we have 4 months worth of data on, and putting them together, and neither one of them raises tremendous concerns to me in terms of the general science behind the vaccines, but I could be missing something. I'm just asking, is there any institutional memory of these vaccines and other questions that have been raised, and should we be concerned in a particular way about these vaccines?
I'll see if anyone else from the FDA wants to opine on this. It's certainly sort of a little outside the scope of this EUA review.
I think that answers my question. Thank you very much. And by the way, you guys have done just a tremendous job.
Okay, why don't we, at this point, since I promised the committee that they would be able to ask questions of the Janssen team, since we had to terminate our questions, Dr. Hefner, Dr. Zhang, you're off the hook for now. And we're going to move to a few questions to the Janssen team. We're not going to allow unlimited number of questions because we really do need time for our own discussion before the vote. So, Dr. Hildreth, you have another question, or was yours answered?
Thank you. Mine was answered.
Okay. Dr. Kurilla. You're muted.
Dr. Corella, you're muted your own phone. Are you unmuted now? It says you don't have your audio connected, Dr. Corella. Here you go. We're down here. I'll unmute you right now. Hold on. I got you. Give me 1 second. And now you can talk. Go ahead.
Thank you. Thank you. So in looking at the cellular immunity that you measured, it looked like there was evidence that well after 28 days you were continuing to see increases in some cases. And I'm wondering from some of the earlier studies, the phase 1-2, where you may have longer data, are you continuing to collect immunogenicity data from those groups, and does that suggest, does that at least provide the potential for estimating when you might need to boost, or possibly also the duration that you may see from a single dose? Any clues or insights from that?
Dr. Von Hoof, did you mute yourself again?
No, it was my error. So,
there you go.
Yes, yeah, so indeed to your question, we have designed our phase 1 study such that we do monitor these subjects over the next 24 months, and that we will monitor their cell-mediated immunity and the humoral responses, which will help us, at least to some extent, in guiding us when a boost might be needed. We do know historically from the platform experience that we do see quite good persistence even after 1 to 2 years. We have seen that with our Ebola vaccine, but it was a 2-dose vaccine, but also with Zika, we see good persistence at least up to 1 year.
Okay, Dr. Pergam.
Hopefully you can hear me, since it seems like muting's been a problem. Okay, good. Good. So just a question about the vector itself and looking at immunity to the vector over time. Most of the adenovirus vaccines that you've done have been short-term prime boost or just a single dose. It's not really been long-term where you've gone back and revaccinated. Is there a plan to look at the AD26 immunity over the longer course to see if it might preclude or might limit the boost of responses at a later date?
This is a very important question indeed, and we do have an HIV vaccine program that is running, where we are in a clinical proof of concept study in South Africa. We also have a phase 3 running in the Western world, and that's a study in which we give up to 4 injections, and we do see that as you continue to give injections, you do see a continued rise in antibody levels. We also have experience with people who were vaccinated several years ago with the HIV, a prototype of the HIV vaccine that were boosted several years later. And it seems to be no problem at all to get that booster response. But it's certainly a very valid point. These vectors are immunogenic to some extent, but it looks like the overall viral load that you give with the vaccine is sufficient to overcome that eventual hindrance.
Thank you. Dr. Perlman.
So I have a question about one group of patients, namely the ones who are greater than 60 with the comorbidities who have only a pretty, not a great protection rate. And so there's a few parts of this that I find, well, I don't know really what the answer is. So we talked a little bit about immune responses, and I know that you're going to be looking at that, but do we expect this population to respond to adenovirus as well as people who are not over 60 with comorbidities? And do you think that when you go to the dose regimen, will this help with this group? As it is now, I believe that the people who receive this, they will feel like they're getting a vaccine that doesn't work as well, and so we don't want to have 2 classes of recipients who feel like we're increasing health inequities.
This certainly is a question that is very important, and once we have had that observation, we went deep into the details to really understand the numbers. And perhaps I can share a few slides. It is essentially, we are in line with the observations that already were shared by the colleagues from FDA, which, as you see, this is actually the first plot data that give the efficacies. They are ordered in a somewhat different way. On the top, you see the dose after 14 days. On the bottom, at the 28 days. And you see actually that when you have sufficient number of cases, also for the people over 60 with comorbidities, you have quite decent vaccine efficacies for the moderates and severe, with also confidence intervals that are quite good. It is when you go to the 28 days lower, and you go really into that category with over 60 with comorbidities, where you observed that lower point estimate, however, also already indicated by FDA with that very wide confidence interval, which is linked actually to the very low numbers. What you see on the right-hand side is there was a statistical analysis made, the Cox proportional hazard test, and the p-values indicated that there was no evidence of statistical significance when the three-way interaction was checked, so age and comorbidities. Of course, that is the question here is how is this? And then an interesting finding, which I would like to share, is when we looked closely to the Kaplan-Meier curves. And what I'll guide you here through the slide itself. You do see in blue the studies, the active group. The dotted line is the overall study population. The full line is the population that received the vaccine, with so that cohort with comorbidities. And the gray lines are dotted line overall study population. And then the full gray line is over 60. And what you do see is first the split for the people over 60 with comorbidities is exactly the same moment, and the trajectory is really very similar to the trajectory for the overall population. So for me, this really indicates that there's something weird is happening here, not that much with the vaccine line, but with the placebo line, which in our view, to the point made earlier, is really linked to that overall shorter follow-up, because you do see a significant drop of the population that is available for that duration of follow-up. Now, and what is also important but not seen here is that when we look then to those cases and we look individually, then we see that those are somewhat randomly distributed over different countries, but there were 2 countries where there's sufficient numbers to reach efficacy. It was the US and South Africa, and in both cases it was around 70%, 70, with regards to the moderate and severe. Now, what is certainly very reassuring, and also our colleagues from the FDA have mentioned this, is that when you look really to what is important for this population with regard to severe endpoints, then we did see very clearly that we had a high efficacy, 82%, seeing 2 versus 11 hospitalizations after day 14. So statistically significant with confidence interval above 0, which was really very encouraging. And we also see 0 against 5 after day 28, which is in line with the observation of hospitalization for the rest of the study population. In addition, and this is of course not statistically relevant because it's only 2 cases, but it is encouraging to see that you have no cases of death in this population due to COVID, while you had 2 that was related to, that was in the placebo group, that was COVID related. Of course, one of the challenges that we have is that despite having 44,000 people, once you go doing subgroup analysis of subgroups, you end up with low numbers. But so if when we take all of this together, we do feel that specifically these numbers that are in front of us give us sufficient confidence to say, yes, this population will benefit also from this vaccine. Thank you.
Okay, I've got about 8 hands raised, and we really need to close out the discussion. So I'm just going to call on Dr. Marasco to have the final question.
Sure. Can you hear me now?
Thank you.
Can you? Okay, good. Fine. Thank you. Well,
I didn't get to introduce myself earlier, but I work at the Dana-Farber Cancer Institute. And my question is really related to the patients that we see. So there's a large number of patients on checkpoint blockade inhibitors and in different stages of chemotherapy or with a distant history of cancer. So how do you plan to roll out the vaccine to these particular patients, or are you planning on studying them at all?
Yeah, we are actually planning on studying them, and we are already in discussion with some centers that have expressed interest to start these studies as soon as possible. Out of principle, we didn't want to start those studies before we had evidence of efficacy, not to put them unnecessarily at risk. But based on the efficacy now observed and under the assumption that the emergency use would approve, we would certainly start these studies.
Okay, so thank you very much. We're going to move now to the general discussion. There may still be questions for the sponsor. So if it's not too late where you are, please stand by. Why don't we start our general discussion, and I'll call on Dr. Rubin to lead us off.
Thank you. Thank you. I'm having some problems with my webcam that won't seem to turn on. So
we hear you, though.
Great, thanks. I guess my biggest concern, and it doesn't really speak to the approval as much as to how we use the vaccine. There is this ongoing study with 2 doses. If that proves to be superior, what do we do? Because we have a vaccine now that has good efficacy that everyone is going to compare to the existing vaccines and say it does not look quite as good. We have a second dose that might well, and after what we just heard from Dr. Van Hoof, that there might well be a better response. But we're going to have a large number of people who've gotten a single dose out there. What do we do for them, including the participants in the trial? We won't have a way of capturing them the way that we do for the current vaccines, because it's sort of built into the program. It seems like a big logistical problem.
The simple answer from an FDA standpoint, and we may want to ask our colleagues at FDA to comment, is that this is something that the ACIP will need to consider, that what is in front of us is whether this vaccine as a 1-dose formulation should be approved. But I see this also as an issue, especially if the 2-dose formulation in study 3009 proves to be more efficacious. Other comments? Dr. Gruber, please.
Dr. Gruber, please unmute your own phone.
From a clinical study that evaluated 1 dose, and we have a study ongoing where 2 doses are evaluated. I think what we need to keep in mind, in addition to have conversations also with other health policymakers and the ACIP, is that these, if authorized, this is an emergency use authorization to really mitigate, hopefully, the devastating effects of the current pandemic. The question about, if data show that 2 doses are going to be more effective than 1 dose, you can really, address it by looking at a biologics license application and see what the dose and schedule would be approved there. And if, God forbid, this pandemic drags out, that we'll have to have these emergency use authorizations in effect, and we have data then to see that, or to demonstrate that 2 doses work better than 1 dose, there's always the provision to amend the emergency use authorization to allow 2 doses. I understand that there are logistical issues and operational issues that need to be sorted out, but from a regulatory perspective, I think we have means to address that. Over.
Okay, thank you. I think that clarifies the situation. It's still a difficult situation, but Dr. Kim?
Yes, thank you. So in the briefing document today and during today's presentations and discussion, Janssen championed Ad26's effectiveness against moderate to severe COVID. I'd like Janssen to reconsider this claim. Earlier this afternoon and during OPH, 2 of the commenters, I believe they were Drs. Doshi and Zuckerman, mentioned basically a total lack of mild COVID cases in the study. And honestly, that's you had mentioned this as well, Dr. Manto. And Dr. Zhang, during her presentation, discussed the lack of mild cases. And this is basically a case, basically a situation where Janssen's definition of mild case versus moderate case are inconsistent with FDA's and with CDC, as well as the other 2 vaccine manufacturers for whom the EUAs were administered. So we're different. So the mild case is defined as a single symptom, whereas moderate case was defined as 2 or more symptoms by Janssen. And for others, we're talking about a situation where the standard definition of symptomatic COVID is 2 or more of the symptoms. And so I would like to see Janssen revise their claim or their position in saying instead of saying there's a 67% moderate to severe COVID vaccine effectiveness, that they be consistent with what's currently in use for FDA and others by saying that the 67% vaccine effectiveness applies to symptomatic COVID. So just something that I'd like us to have some consistency in this.
Dr. Gruber, would you care to comment, or should we go to Dr. Van Hoff?
Yes, I think I would like for Dr. Van Hoff to take a stab at that. I mean, all I want to say is it's really difficult because these were pre-specified case definitions, and the primary analysis was really, specified looking at these definitions. So going back now retrospectively and change that, I think will provide with difficulties. But I also would like to give the sponsor a chance to comment on that.
Thank you, Dr. Gruber. No, I fully agree. Based for methodological reasons, it would not be wise to go back. A good thing is that we do have, as part of the analysis, also looked at protection against symptomatic infection and also protection against COVID infection according to the FDA definition. We will make sure that in our publications those numbers are also mentioned such that there is less potential for confusion. And so from that perspective, I hope that will help also to avoid confusion.
Right, I think that would be a great help if both are presented. So whoever is reading the briefing documents can figure it out.
Sure, I would like to come back to the discussion on the 2 versus 1 doses. As I said before, we still need to wait to see what the 2 doses will give us incremental value, and we overall feel that with the efficacy we see in hospitalizations and death and critical disease, that 1 dose already delivers quite a lot on the promise. Thank you.
Thank you. Dr. Hildreth.
Oh, I don't have a question. I'm sorry.
Okay, could you put your hand down?
Yes, sir.
Okay, thank you. Dr. McInnes.
Hello. Thank you, Arnold. This question about the case definitions, I think it's very important to be able to report out by the FDA concordant definition, and it sort of comes from my question before lunch today about the case definitions and how you've lumped things together that I think leads to some confusion. My actual question pertains to your placebo and to your actual description of your vaccine. I presume that you probably used saline as a placebo, but I searched for the word saline in your briefing document for the sponsor. Does not reveal anything, so I'd like to please know what your placebo was. And then I have a question that regards what your vaccine actually looks like and how you handled blinding. And the reason this comes up is because in the FDA document, they describe the product as being colorless to slightly yellow, clear to very opalescent, which I'm not really sure what that is actually looking like. I don't think there is a description of the vaccine in the sponsor's briefing document. If in fact it is not clear or colorless in a match to whatever the placebo is, how did you handle blinding, please?
Yeah, so it's a very good question, and all of the vaccines were prepared by a pharmacist who was unblinded, but was independent then from the study, and who was also accountable for the vaccine preparation. Because of differences in appearances, there was also a blinding tape, so a translucent yellow tape that was wrapped around the syringe after drawing up the suspension, and then the masked syringes was handed over to the nurse, who administered then the vaccine to the participant in a blinded fashion.
And was it saline?
No, it was diluent. It is the same buffer as we use for the vaccine.
It's a buffer. It's a vehicle or a buffer?
It's a buffer, but let me double check with the experts.
Thank you.
Thank you. Thank you. Yeah, it is.
It is.
I will come back to it when I have the final confirmation. I have some communication challenges here, working virtual. Sorry.
Dr. Rubin.
I don't know how I do that. Sorry, I'm sorry. Please, my hand shouldn't have been up.
Okay,
down now.
Dr. Chatterjee.
Yes, I have a comment to offer, Dr. Monto, and then I have a couple of questions as well. The questions are very brief, I promise, and so is the comment. The comment is with regard to the concerns around the 2-dose versus the 1-dose. You know, I think that we are in such a fluid situation that what happens 2, 4, 6 months down the road is going to be very difficult for anybody to really say for sure. We currently have 2 2-dose vaccines that have been authorized and are being used. Well, there's already discussion about will you need a booster dose and how will we figure out, you know, how to give those booster doses. So I think this is going to evolve over time. Can people hear me?
Yes, we can. Yes.
Okay, thank you. The questions I have, there are actually 2 and fairly easy. The first one is, in the COVID 3001 study, I noticed that taste and smell changes were not included in the moderate to severe or severe to critical case definitions. And I was just curious about why that was the case.
Dr. Van Hoff?
I'm afraid you can't leave.
Okay. Could you repeat the question, please? I didn't fully understand it.
Please
So
repeat,
changes,
Dr. Chatterjee.
yeah, changes to taste and smell, which are pretty common symptoms for people who have COVID. They don't appear to have been included in the case definitions for the moderate to severe or severe critical cases. I was just curious why they were not included. Because when I looked at the list that was on the chart, they were not there.
I'm pulling up now the definition of the that was used, and as you will see, change to olfaction and taste is actually part of the definition. It is part of the definition, so you might have overlooked it.
Yeah, I'm sorry, I can't tell you where I saw it, but I did make a note when I was reading the briefing document. The other question is with regard to the distribution based on sex. So in several of the tables, it said 45% male and 45% female. And I was just curious where the other 10% went. Were they not reported, or were they other gender something?
I'm going to refer to Dr. Dworgi for that question if she has more details on that. Meanwhile, I need to correct myself. An earlier question: the placebo used was indeed saline. I was wrong. It was saline that was used, but it was taped like I described. Yeah, it is. Can Dr. Bart Spieters, our statistician, comment on the lacking information on the gender? Bart?
Yes, I can, Dr. Van Hoof, thank you. I'm Bart Spieters, I'm a clinical biostatistician at Janssen. So I assume that the 45% that you refer to is that in each, both the vaccine group as well as in the placebo group, there were 45% of females, but there were then approximately 55% of males in each of the two groups. Is that the percentage that you refer to?
Again, I don't have the reference, unfortunately, of the actual chart, but the table. But I did see that they were. It said both of them said 45%, so that confused me. Maybe it was just the females.
It is actually indeed a strange way of putting it on the table. I agree. And the way that it is mentioned on the line is sex, female, 45%. Hence, assuming the 55% is indeed male.
Okay, thank you for the clarification.
Okay, Dr. Offit.
Yeah, I have just a practical question, but one that I get asked a lot. I noticed that in your trial, in your briefing document, 26% of younger patients took antipyretics around the time of getting vaccinated. There are studies, one in the Czech Republic, another in Australia, looking at influenza vaccine as well as a variety of other vaccines, showing that that can lower the immune response. I just wonder whether you had any data on whether or not the choice to use antipyretics in any way affected the immune response.
I'll immediately go to Dr. Dworgi for the question, but I do know that we actually did screen indeed intensively the literature about impact on use of antibiotics on immune responses. What is most striking is that there's no consistent report, but indeed when it's taken prophylactically, it seems to impact. When it's taken reactively, it seems to be less clear. We have never recommended the use prophylactically, but we did recommend once there was fever starting or headache starting to use an antibiotic. So it was reactive use and not prophylactic use. I don't know, Dr. Dworgi, if you want to add something?
Yeah, hello, can you hear me? Sorry, there's an incredible delay. Okay, yeah, because we didn't require prophylactic use, it's very difficult to assess. We encourage people to take antipyretics symptomatically, so we really don't have a means to make that comparison officially. And as Johan said, I think the literature are conflicting in terms of what the impact potentially could be. Certainly we will have some immunogenicity data. We can look to see if with the data that we have, we can make some sort of assessment based on antipyretic use, but we have not yet done that.
Yeah, I think the literature was all prophylactic, where it did affect, not. I don't know if there was a literature sort of post developing fever, but thank you.
Dr. Chatterjee, you're still up there. Do you have? Are you still okay? Thank you, Dr. McGinnis. You have another question?
No, I just took it down. Sorry.
Okay, thank you. Dr. Fuller.
Yes, thank you. This is for the sponsor as well as for FDA. Given that, as my colleagues have said, this is a very fluid situation where things are changing rapidly, and we want to know some follow-up things that will be important to managing this pandemic and hopefully getting us out of it. So to the sponsor, you enrolled 44,000 people around the world, which is wonderful, and you propose a crossover study to retain them. I want to know if you have indications that will work well for the people that you have enrolled. And secondly, and this is important for things like the response to the adenovector, to the pregnancy, to the asymptomatic infection, to the duration of immunity, all those things we need to know. And then I'd like to address that question to the FDA in terms of the follow-up with the Moderna and Janssen studies in terms of keeping the people in the trial still in the trial.
No other vaccines,
so
Ovita.
Excuse me?
No other vaccines. Off limits.
Well, it's about the whole vaccination process, Dr. Michael.
Okay,
so,
but
they can answer if they want to or not. It's up to them. I just put the question out there because it's important.
We do hope that by in the hypothesis that we have the emergency use application authorized, we do hope that offering the crossover to the vaccine will also incentivize people to stay in the study. Because the reason to leave the study, that's what we have seen happening, is to get access to the vaccine. Many of the people participated also because they want to contribute to the science and seem to understand indeed that by continuing to be in the study, monitored for safety, monitored for immunogenicity, will help science to progress and will help to address some of these critical questions. Of course, we don't have a crystal ball, and this is not guaranteed, but we do think that by offering the vaccine, we can really be and hope to be successful in retaining the people in the study. If we don't do this, we have seen that many people are already leaving the study, and that by vaccines becoming more and more available, this will become impossible. So that's why we have decided to go that route.
How long will it be before the crossover occurs? Have you determined that?
That is actually depends on the countries where it is happening, but we would like to make this happen within the next coming weeks to months.
Okay, I have a follow-up for Dr. Van Hoop. Do you have any assurance if vaccination is being given to hold people in the study, that once they are vaccinated and know they are vaccinated, because you're basically unblinding, they won't leave the study at that point because they have achieved what they're after, getting vaccinated. And you'll have the worst of both worlds because you will now have an unblinded study of people. You will basically have an open label study. Has that been considered at all?
Well, that is certainly a risk that exists. We do think that risk can be mitigated by community engagement with the participants. I think that apart from having the vaccine, there is also the follow-up that we do in terms of eventual workup of infections that would occur. So there is also the follow-up that is important. We also follow up with regard to the immune responses. So It's also part of the benefit that they have by participating to the study, that they will know that there will be follow-up if COVID would occur, but also that they will know if their antibody levels would drop extremely low or whatever. So I think there are some incentives which we hope can help to mitigate that risk.
Thank you. Dr. Levy.
Hello. This is a question for Dr. Van Hoof. Can you hear me?
Yes.
You can.
Wonderful. My question to the sponsor relates to the dose of the vaccine. In other words, you know, you have certain efficacy with the single dose regimen with the current dose, and yet you feel compelled to start another study to look at a two-dose regimen in hopes of gaining more efficacy. Have you considered an intermediate dose from the phase 1 in between the current dose and the higher dose to see if that might give more efficacy in a single shot regimen, and or the addition of an adjuvant?
Yeah, these are very good questions. Of course, we should not forget that this was a race against the time. We obtained the sequence of the gene in mid-January. We evaluated 12 transgene candidates to see which one would give the most immunogenicity. With regard to the dose of the platform used, we actually do have quite experience with the platform, having tested it now in many of our other research programs. So as you know, in vaccinology, it is always the challenge to find the right balance between maximum immunogenicity and acceptable tolerability. We know historically with these vectors, with the Adeno-26, that there were two doses that we know work good. We know the 5-10-10, which is for most of our programs. We know the 1-10-11 gives occasionally somewhat higher immune responses, but also is more reactogenic. And that was the reason why we have selected these two doses. Then, based on the phase 1 results, we have seen that they give very similar immunogenicity, while it was more reactogenic, at least certainly in the younger people. And therefore, we decided to go for the 5-10-10. Now, in vaccinology, very often, it's not that much a matter of only giving the amount of antigen, but also the schedules are important. And that is the other element. And you have seen study 2001, which is evaluating several schedules. That is actually one element we are doing there, is we want to test also the robustness of the immune response and immune memory by giving these people who have received a single dose, giving them a very late boost of a small amount of antigen, and then check whether within days you do see a rise in antibodies, which is a hallmark normally of a robust immune memory and amnestic response. And that is work that is still ongoing.
Thank you, Dr. Van Hoof. How about the older individuals who have comorbidities? Older people have less reactogenicity. Can you give a higher dose in that group and get better protection?
That is a theoretical question, but again, I would like to go back to the analysis we did. And we do see that we have there pretty high protection against hospitalization, and also death was 0.2, but hospitalization was pretty high. So I'm not sure whether adding the more would necessarily result in higher results. We do think the antibody levels play an important role. We should not forget also that this vaccine has very strong cell-mediated immune responses. And when you look to the effect on severe disease, kicks in as of day 7, when there's hardly any antibody present. So it is reasonable to assume that also cell-mediated immune responses play a role here. And there we do know that it does not necessarily increase that much with increased dose.
Thank you.
Okay, Dr. Kurilla.
Thank you. Thank you. Yeah, this is a comment, I guess, for both the sponsor and the FDA. You know, I mean, ideally, it would be fantastic if your vaccine induced lifelong immunity. But well, that's a tremendous aspirational goal. I'm not sure any of us are really looking for that or have that expectation. But that raises the issue. You're talking about a 1, now perhaps a 2 dose for the primary vaccination, but then the expectation is that there may have to be ideally annual booster shots that people are talking about, and they're talking about boosters for the other vaccines. But we've also seen that down the road where we may have to anticipate a strain change, that's being referred to as boosters, and I'm seeing quite a bit of confusion of what is meant by a booster. And so we have the initial primary vaccination with a 2-dose regimen, which you end up with calling that a prime boost. Then we're talking about needing another boost, and then we're talking about a strain change that's a boost. It's going to become very confusing, and I think there needs to be some general agreement on the nomenclature here so that it is not just another a problem for the general public to understand what's going on as to why they need another vaccine at this time. Just please give it some thought.
That's a fair point, yeah.
Okay, Dr. Portnoy.
Thank you. I've just turned on my microphone and my camera. Things are a little bit delayed. As an allergist, I'm concerned about allergic reactions to the vaccine, in particular anaphylaxis, which has been reported with other vaccines. I was wondering if you had information about sensitization rates in terms of IgE to the adenovirus in people who receive your vaccine. I know anaphylactic reactions are likely to show up as more people receive it because it's a low frequency event, but what are the ingredients in the vaccine, and have you looked at them as possible sources of anaphylactic reactions?
For this, I'm going to refer to Dr. Duogi if she can comment on that, and also on the excipients that are present in the vaccine.
Hello, can you hear me?
Yep.
Yes.
Okay. Sorry, I think the camera's delayed. Yes, so we have not seen a lot of hypersensitivity in our platform data. So just with respect to the IgE questions, we have not had a trigger to explore that. In terms of the excipients that we have, yeah, I just would like to pull it up so I can read it to you, if that would be helpful. I mean, we've used a lot of excipients that are basically considered common in terms of buffers. Yeah, so sodium chloride, citric acid monohydrate, trisodium citrate dihydrate, polysorbate 80, HBCD, ethanol, sodium, hydrochloric acid, and water for injection are our excipients.
Okay. Yeah, and you've not found any evidence of IgE development to the adenovirus. Have you looked at it, or have you thought about doing that?
To my knowledge, we have not looked at that. I mean, there's certainly from a clinical perspective, we have not had the impetus to consider that. So yeah, it's I think an open question. We don't have reason to expect that the adenovirus itself would cause that, just based on what we know about natural infection. You know, there doesn't appear to be IgE-mediated manifestations associated with natural infection, so we wouldn't expect to see that with the adeno itself. So again, if there's any source, you know, the excipients would be more likely than the virus itself.
Okay, great. Thank you.
Dr. Kim.
I'd like to follow up on what Dr. Gruber said earlier. When I mentioned that this brought up the discussion on case definition, I wasn't referring to changing any of the study protocols or retroactively modifying things to better suit the needs of the outcome. The Janssen protocol's first primary outcome was moderate to severe COVID disease, And the secondary outcome was symptomatic COVID. And symptomatic COVID is basically the same as moderate to severe COVID, because altogether out of the entire study population, there were only 6 cases of a mild COVID. So the FDA case definition and Janssen case definition for symptomatic COVID, essentially, they are the same. So just to comment on that, nothing will be changed if Janssen proceeds with the vaccine effectiveness against symptomatic COVID as opposed to moderate to severe COVID. And that would be consistent with all the other definitions that are out there, and that's not changing anything from Janssen's study protocol. And the other thing I would like to bring up is I'd like to follow up on what Dr. Moore said this morning. It's a two-part question, so please bear with me as I set up the question. The FDA briefing document stated this, regarding the benefit of the Ad26 for individuals with prior infection with SARS-CoV-2, there were limited cases of COVID-19 among study participants with positive SARS-CoV-2 infection status at baseline. The study was not designed to assess the benefit of individuals with prior SARS-CoV-2 infection. So the first part of the question I have is, are there enough data to look into whether those who are present, those who were COVID positive at baseline, mounted a more robust immune response? So that's my first question, first half of the question. And this, and I want to bring up Dr. Moore's concern again, or point again. He brought up possibly testing the subjects for evidence of infection in the placebo group at the time of unblinding before administering the vaccine. So this could help answer the second part of that question. So the second part of the question is, can the single dose of Ad26 serve as a de facto immunity booster for COVID? This will be relevant for the 19 million or so adults who've already tested positive and might be candidates for a one-dose vaccine.
On that one, can you hear me? Yeah. So
Yes.
With regard to the crossover to the placebos, I can confirm to you that everyone will be swapped and a blood sample will be taken to look into seroconversion. So that is part of the. With regards to the immune responses observed in the people who were seropositive, I would like to ask Professor Schuitsmaker to comment. I think she has the information, the details on that. Professor Schuitsmaker?
Yes. Can you hear me?
Yep.
Because I don't think my camera is working, but I can do it off camera. So in our phase 1/2A study, we had few individuals that were seropositive at baseline. And there we indeed saw, sort of boosting effect of our vaccine, resulting in some of the individuals in very high antibody titers. So it seems to have a beneficial effect, vaccination in people who are seropositive at baseline. Over.
Do you have enough data to conduct a robust analysis?
No, well, we will do, of course, this analysis in our phase 3 study. Now that we have identified who were seropositive, we will look at the immune responses 4 weeks after. So that is a work in progress. For the people in our phase 1/2A study where we have done this analysis, there were too few to do a robust analysis, but work in progress.
Thank you.
Over.
Dr. Meisner?
Yes. I'd like to ask 2 brief questions, if I may. The first question is, you define asymptomatic serologically by antibodies to the nucleocapsid protein. And Is the nucleocapsid protein unique to SARS-CoV-2 so that there isn't cross-reaction with seasonal coronaviruses, for example? Is that really a reasonable way to determine if someone's been infected? And then the second question is on the Janssen briefing document on page 73 out of 118. I think there's a typographic error, but it says that reactogenicity was evaluated in people under 18 years of age, and I think that was supposed to be greater than 18 years of age, but again, I'm interested in its use in adolescence if that did occur.
With regard to the letter, it is indeed a typo where it should read over 18 years of age. With regard to your first question, I will go in a minute to Professor Schuitenmaker, but I do know that we asked ourselves the question, to what extent can we validate that seroconversion against N protein as a hallmark for infection? And we did that by going to look to all the cases and validate and check to what extent all the cases that we had detected by PCR, to what extent they were actually seroconverting, yes or no. And over to Professor Schuitenmaker to give you the results of those and to comment also on the question about potential cross-reaction.
Yes, so there's delay, so again my camera will turn on when I finish my answer. We validated indeed the N-IgG seroconversion or N-antibody seroconversion by looking back in seropositive cases whether they also had a PCR, so whether the cases that we had, the PCR positive cases, had seroconverted. This was the first analysis for N seroconversion on day 29. And there we observed that 90% of the PCR positive cases had seroconverted, and it was interesting to see that people who were PCR positive too close to day 29 had not yet seroconverted. So there is a slight delay there, but there was overall very good concordance between people who had a PCR positive result and a seroconversion for N antibodies. I missed the second question. Johan, can you repeat?
That was a typo in the briefing book. That's no problem. That was okay. That's solved.
Okay. Okay. Thank you.
Cody, I can tell you from a paper we have under review right now, there are cross reactions between seasonal coronavirus infections, low level, and the full N protein of SARS-CoV-2. But the other thing that's going on now is that we are practically not seeing seasonal coronaviruses, just like we're not seeing influenza viruses right now. But it is something we will have to watch for going forward.
Yes. And do you think, is that true everywhere, Arnold, in the world, for example, in Africa, that the
I have no idea outside our area, but it is something that needs to be checked on. Yep. Thank you. Next is Dr. Sawyer.
Good. Somebody already asked my question. I'm good. Thank you.
Okay. Dr. Pergam. We're moving along quite nicely.
Thank you. Thanks. Dr. Van Hoof, I have a question. In relationship to the new variants, we have, a couple of variants of interest at the moment. Since there has been evidence that maybe a little less response to the vaccine in the South African strain, as an example, is there efforts by the company to start working on additional updates to the vaccine that might include these new variants, and what would the time frame be to make that change if you needed to?
That's a very good question. First, I would like to come back to what we have observed in South Africa, where observing the efficacy against the severe endpoints, the efficacy was quite high, as we have said. And also, even for the moderate and severe, we did see that the efficacy gradually increased. So I think the jury is still out on whether there's a new generation vaccine needed. This being said, we are not complacent, and we are in the making of a new variant vaccine that should enter the phase 1 trials before summer. Also depending on, while we will monitor in parallel on how the situation is evolving. But again, I would like to say that what we observe is that also against moderate and severe, that there was efficacy, but efficacy increased over time and was substantially higher by day 56 than it was earlier. So I think the judge is still out there. Some elements, and we have not, these are data that are fresh from the laboratories, but we also do see that we have functional activities that are related to the Fc fragment of the antibody. And Professor Schuitenmaker referred to that. And we are evaluating to what extent these functions are impacted by the variant, yes or no, because we do hypothesize that these mutations should not impact those, and we have preliminary data that suggests indeed that those functional activities are preserved. This being said, we don't want to take risks, and that's why we are preparing also, if need be, a second generation vaccine, and that could be in phase 1 before summer.
Okay, Dr. Moore.
Dr. Pergam just asked the question I was going to ask, but following up on his question, and it's more of a question to FDA, is how much change, or is there any change genetically that can be made to the vaccine without triggering a full re-examination through the EUA, meaning that you have to do full phase 3 trials? Is there any change that can be done, or do we, how much clinical or wiggle room do we have on something like that?
Dr. Gruber.
Yeah, this is Marion Gruber. I'm trying to start my webcam here. So, well, I think you know what I look like. So the
we hear you.
You can hear me, right? Yeah.
Yes, we can.
Okay, so on the question about making adjustments to the original vaccines to really get protection against emerging variants, we have actually just recently, was it last week, amended our EUA guidance document to really discuss the type of data that we would need to see to authorize a modified vaccine against some of these variants of concerns. And we basically would address it by way of immunogenicity bridging studies. We would not request clinical disease endpoint efficacy studies. Does it answer your question?
I guess it does, because he signed off. Okay, Dr. Marasco.
Yes, I'd like to address this to sponsor. So you have a lot of experience with adenoviral vectors, and do you have a sense to what extent they're activating the innate immune system? I mean, do you have cytokine levels or any other quantitative parameter which may allow this adenoviral vector to act as an adjuvant without adjuvanting it? I mean, do you have science on that?
For that one, I'm going to refer again to Dr. Zahn, who is our preclinical expert. Dr. Zhang?
Yes, I'm back on. Yes, this is Oran. So indeed, we have done studies with Ad26 vectors, not with this specific Ad26 Corvus 2 vector, but with other adenoviral vectors coding for HIV or vaccine inserts. And there we have seen indeed that multiple cytokines are induced either measured systemically after administration to humans or in vitro by stimulating PBMCs with giving the Ad26 vector. So we think indeed there is an innate stimulus by the vaccine vector itself, also for this vector.
So are you suggesting that that is adequate to explain its immunogenicity or potentially enhanced immunogenicity? For example, interferon activation.
Yes, it's certainly one of the factors which drives the immune response and which activates the adaptive immune response to a vaccine insert of this vector.
Thank you.
Thank you. Dr. Gans.
Hi, all. Thank you. I wanted to follow up on a question that I asked previously because it wasn't really answered, and it was really to just get some more details on the study 2001 and 3006 that are planned for hopefully enrollment this year, and particularly I understand next week starting with the 12 and 18-year-olds. There's some indication in the table that those will be done in the same geographic locations that were done just with an extension down in those populations. But I just wanted to ensure that we're actually going to get good demographic representation within our pediatric populations for those two studies. So that's my first part of the question. My second part is people are starting to have issues enrolling children into these studies, and I'm wondering what is the backup plan for that for your particular vaccine?
Yes, as mentioned in the earlier, we do have a trial that will start recruiting adolescents very soon. Actually, we hope this week, but it will be next week. With regard to the pediatric plan and the speed with which we will recruit, I'm going over to Dr. Dwogi, who is closer to the details of those studies. But indeed, we do also plan to recruit in several countries, including the US, but also other countries. Dr. Dwogi?
Yes, and thank you for the question. Yes, so we are going to be running these trials in multiple countries. Some will be different than the ones where we're conducting the efficacy studies, but there's a lot of overlap. And from the operations team, they've done an extensive feasibility to look at where there might be interest in vaccinating certain age groups. And as you can imagine, depending on the age of the child, there is more or less interest in enrolling. So our plan, and of course I don't have a crystal ball, is to really diversify and make sure that we're in a number of different locations and partner with investigators that really have good experience and track record in recruiting all age groups such that we can conduct these trials as expeditiously as possible. And so from a demographic point of view, I think we should have quite a mix. And again, we'll have to see how things start up, but we are hoping that we can recruit very quickly. Of course, the first data set that would come forward is in the oldest adolescents, the 16 and 17-year-olds, and then move down in age range over the course of this year. But the aim is really to generate as much data as we can this year. And one last point, just the totality of the data set from the two trials that we intend to generate will be over 3,000 children.
Thank you.
Dr. Kurilla. You seem to be the last.
Thank you, Arnold.
If anybody, to my great surprise, we have come to the end of the list.
Okay.
We have some more people who may want to make, so we have some extra time for comments if necessary. Dr. Carrillo.
Well, thank you, Arnold. We've seen recently of some in vitro studies of neutralization of virus by serum from patients from subjects who have been vaccinated, but we've also seen quite a bit of variability between neutralization of variants based on whether they're point mutations or whether they are actual clinical isolates. But we don't have any direct correlation with the clinical efficacy of those studies that have been done. You're in a unique situation where you can actually do that, and I'm wondering, do you have those in vitro studies going on, and do you have any results to share at this point?
Indeed, we do have. Can you hear me? Because my camera is frozen, but so.
Yes.
Yeah, we do indeed. This is an important question. We actually are looking at using sera from our phase 1 studies and later on probably also phase 3. And in collaboration with academic labs, both in South Africa and the UK, we are conducting these studies. We do have data that look to the neutralization of the UK variant that actually, like for the other vaccines, we observed indeed a drop in neutralization. But what was interesting as an observation is that by day 71, that drop had substantially decreased. So it looks like over time there's some qualitative improvement, for lack of a better term, let's call it maturation or affinity, that shows that the drop in neutralization went from about 8-fold drop to a 3-fold drop. So it was an interesting observation. At the same time, it's also interesting to see that even in presence of such a drop, of course this was for the UK variant, but we have also some preliminary data from South Africa that go in the same direction. But even in presence of that drop, it still seems to work. So it looks like it is a biomarker, but perhaps not the only one that correlates with protection.
Oh, good. Thank you.
Dr. Marks.
Thanks very much, Dr. Mono. I was hoping that the committee could potentially make some comments. I want to follow up on something that Dr. Perlman brought up. I think there was an explanation given by the company about the fact that there is this difference between 72% and 42% between people over 60 with and without medical comorbidities. 42% in those with, 72 without in this group for that was seen. And there's also the issue that the two medically attended cases in people over 60 were also in those with medical comorbidities. Now, granted, I'm going to be the first one to say that that's a group that we're dealing with very small numbers, but I think it would be nice to hear some comment about the comfort there, because particularly in that group, there may be people looking at that with a lot of scrutiny vis-a-vis other things that have been done in the past, not to bring in other vaccines from previously, but I think it would be good to hear some comments about that based on what Dr. Perlman brought up.
And I also noticed in some of the earlier immunologic studies, older people seem to get an antibody response slower than younger individuals. Could that have any effect on this finding based on the time they have been under study? So we'd welcome comments from the committee. Dr. Sawyer.
Thank you. Sorry, I've been having audio issues too. I was going to ask with regard to Dr. Mark's question whether we are going to get data from the two-dose study in adults over 60 with comorbidities after their first dose. Is there a sufficient space between the doses that we could get some additional information from that study? And I guess that's a sponsored question.
Let's go on to Dr. Wharton and gather our questions together about this topic. Dr. Wharton?
Thank you. Because of the way the staged recruitment, the follow-up period for that group, the people over age 60 with comorbidities, was less. And so I wonder if there's been any additional accrual of events in that group since the data set was closed out that would help narrow, that would have a larger number of events and perhaps narrow the confidence interval.
Dr. Van Hoff, do you have any reply?
Yeah, indeed, as Dr. Wharton just said, the point is indeed that this study is still ongoing and additional cases are accumulating, but of course we are completely blinded. I cannot comment on any results there. But so we do have roughly close to 400 cases that are accumulated. I didn't have the breakdown in that particular cohort, so we would have to look into that. I would like to come back though to the observation that we shared when we looked to hospitalizations in that group that was statistically significant with the difference between the placebo and the active group, despite the low numbers, and that was very encouraging. With regard to the two-dose schedule, the current protocol doesn't allow us to go and look, so it will only be when we have sufficient cases accumulated after the second dose that we would do an interim analysis, at what time point we could look at the difference between the single and a two-dose and also the efficacy of those two doses. Again, it remains to be seen whether there is benefit from the second dose. That's something the data would have to tell us.
Thank you. Dr. Marks, have we satisfied your concern, and what are the options in our voting, which is going to coming up very shortly, if we do continue to have concerns about this?
Sorry, who was that for, Arnold? I just want to make sure that, who are you calling out?
Marks. Dr. Marks or Dr. Gruber?
Oh,
Sorry,
there we go.
Peter Marks. Sorry, I must have disconnected myself. Sorry about that. I think it sounds like that, the committee, other committee members don't seem to have a concern with it. I think it's been asked and answered. If it comes up, I think we can handle it after you, after you take a vote if there are issues.
As with anything in these fluid situations, there's no right or wrong answer to a lot of these questions. Dr. Gruber?
No, I actually, I wanted to make a comment, but it has been, I'll take it back. I'm going to be quiet right now.
Okay. Are we ready to vote? Anybody from the group want to make any further comments? The procedure we're going to do is we are now going to vote, and after the vote, anyone, you don't have to, is going to be able to go on and explain their vote. So we are voting on the question based on the totality. Went away. based on the totality of scientific evidence available, do the benefits of the Janssen COVID-19 vaccine outweigh its risks for use in individuals 18 years of age and older? Dr. Hayes.
Thank you, Dr. Monto. Can everybody hear me fine?
Yes.
Great. Okay, so we're going to pull up the slide that has all of our members and temporary voting members on it. Here we go. Thank you. So our members and temporary voting members as seen on the slide, excluding the industry representative, will be voting in today's meeting. And in regards to the process, Dr. Monto just read the question for the record, but you may have to restate it. And then afterwards, all members and temporary voting members will cast their vote by selecting one of the voting options, which include yes, no, or abstain. And you'll have 2 minutes to cast your vote after the question is read. Once all the votes have been placed, we will broadcast the results and read the individual votes aloud for the record. And just please note that once you cast your vote, you may change your vote within the 2-minute time frame. However, once the poll has closed, all votes will be considered final. Does anyone have any questions related to the voting process before we get started?
This is Wayne Maraska. My screen's frozen, so I can't see what you're displaying.
Yeah, and I'd
Go ahead.
add,
If your screen froze, go ahead and take a moment to just log out, log back in. We will hold until you're in. So just don't hang up your phone. In the meantime, Dr. Montel, there is a question, or Kathleen, there is a question as well.
My screen just went blank.
Yeah. So go ahead and that's all right. So while those people are resetting, go ahead, Dr. Rubin.
If I add, I think a lot of us are delayed by probably 2 minutes on the video. So if we vote, by the time it comes up, we won't have been able to vote.
So if you
we will make sure everybody can see. Hold on, Kathleen. We will make sure everybody can see the vote up on screen before we start the timer and all that. We will confirm it. Okay. So it's not as video. It's just what you can see on the screen. So let's just make sure everybody is seeing a live feed of what should be up right now. Is you should all see the temporary voting members' names all listed.
Okay.
I don't. Oh, yes.
All right, Kathleen, take it away.
Okay, is everyone on? Everyone can see?
Not yet.
No,
Yeah,
I can't see.
I don't see the slide.
Mike, would you like us to raise our hands if we can see the voting? Would that make the most sense?
Yes, we haven't pulled the voting stuff up yet. So right now, do you see a slide with all your names on it?
Yes. We do now.
No.
I just did. I just see it.
Yes.
I just see it now.
Yes.
We will make sure, and plus we know when everybody casts, we will make sure we know when everybody casts their vote. So those of you, and I'm double checking, Dr. Wayne Mascaro, if that's you, you are not logged in at all. That's because you're on a frozen screen. So you need to log in. That's why.
No, I'm off. I have a live screen.
Okay, you're good now? All right. So go ahead, Kathleen, while we go forward.
Does anyone else have any questions just about the voting process or procedure while you're getting logged back in? Okay.
I'm sorry, Kathleen. I'm still, just this second, I'm still several minutes behind the YouTube feed here. So should I log out and log back on? Will that get me back into sync?
You shouldn't be watching this on YouTube. You should be watching this here in the meeting room.
I am, but but this screen is many minutes behind the YouTube. I just went and looked at the YouTube, so I'm still
right.
There's going to be a delay in the YouTube. So
there's a delay in YouTube. There's like a 30-second or so delay for feeding the YouTube. So
YouTube is way ahead of me. Way ahead of me.
I think we're all having issues with our screen because I'm having it too. It's just delayed, so just make sure, please, that our votes get in.
We'll make sure that everybody's vote submitted.
Yes. Again, let us go ahead and Kathleen, go ahead. As she'll mentioned, she will not close the vote until we make sure all of you have had the opportunity.
Correct. So, Mike, if you could pull up the voting plot, and then Dr. Monto, if you could just please read the voting question aloud for the record.
For the record, based on the totality of scientific evidence available, do the benefits of the Janssen COVID-19 vaccine outweigh its risks for use in individuals 18 years of age and older?
Thank you. So you should be able to see the voting pod. If everyone could submit their vote, you have 2 minutes or longer if I don't see your vote in.
I see yes 100% on my. Is everybody seeing?
No, Arnold.
Yep.
Arnold. Yep.
Down below. Okay, I got you.
Yep. So.
We've got about 30 seconds left. Okay, so it looks like all the votes are in. If we could close the poll at this time and broadcast the results, and then I'll read the votes for the record. Dr. Meisner, yes. Dr. Lee, yes. Dr. Perlman, yes. Dr. Monto, yes. Dr. Chatterjee, yes. Dr. Fuller, yes. Dr. Portnoy, yes. Dr. Morosco, yes. Sorry, we had a couple people here that accidentally voted. Dr. Pergam, yes. Dr. Levy, yes. Dr. Offit, yes. Dr. Moore, yes. Dr. Carilla, yes. Dr. Cohn, yes. Dr. Kim, yes. Dr. Rubin, yes. Dr. McInnes, yes. Dr. Gans, yes. Dr. Wharton, yes. Dr. Hildreth, yes. Dr. Sawyer, yes. And that concludes the vote. Since the majority voted yes, we do have a favorable vote. And I will now hand the meeting back over to Dr. Monto for the voting explanation. So thank you, everybody.
Thank you all. Now, if anybody wants to explain their vote, please raise your hands and I'll call on you. This is not a requirement. Dr. Chatterjee.
Thank you, Dr. Monto. Just wanted to say that despite the concerns that were raised during the discussion, I think what we have to keep in mind is that we're still in the midst of this deadly pandemic. There is a shortage of vaccines that are currently authorized. and I think authorization of this vaccine will help meet the needs at the moment.
Thank you, Dr. Chatterjee. Anybody? Oh, Dr. Perlman.
Yeah, the only thing I just want to agree with that and also add that I hope we keep getting new information about the vaccine efficacy and safety. In some ways, we have information that's supportive, but it'd be nice to have even more in the future.
Thank you, Dr. Perlman. Dr. Rubin.
I agree as well. I think it's a relatively easy call. This clearly gets way over the bar, and it's nice to have a single-dose vaccine. It is a bit challenging to know how to use it clinically right now, but the demand is so large that it clearly has a place. It is a very changing environment, though, so I think having new information coming out constantly will really help us understand how best to apply this.
Dr. Portnoy.
Great, thank you. Yes, I believe that the vaccine is as safe and as effective as other vaccines that are currently approved, such as influenza. The main thing to keep in mind is that we're dealing with a pandemic right now, this is like a very urgent thing. It's as opposed to an endemic virus that most of the viral vaccines that we use treat. And so there's an urgency to get this done. We're in a race between the virus mutating, new variants coming out that can cause further disease and stopping it. So the fewer people who are infected with the virus, the less opportunity it has to emerge as a more virulent strain. So we're in a hurry. We need to get this vaccine out. I do believe that the evidence supports its safety and effectiveness, and therefore I think it's great that we're able to have this vaccine.
Dr. Moore.
This is a comment not so much to the committee, but to the public and to reporters who may be watching this on YouTube or whatever. But the point is if you go back to December, the early days of December, when we first went through this process, those trials involved about 45,000 people. Now, 55 million people, which is 1,000-fold more people, have been vaccinated in just the last 2 months. At that time, we had comparable amounts of time to look at safety and efficacy of the vaccine as we do today. As of February 26th, things are looking good. That could change tomorrow, but this whole, my whole point is that this process, the EUA process, does seem to have worked, despite my own personal concerns about it, say 6 months ago. It does seem to have worked. And listening to particularly Dr. Shima Bukagoro's talk today, it was quite clear that there was nothing surprising in terms of the safety and efficacy of the previously approved vaccines that occurred, and they're being monitored. So in terms of vaccine hesitancy, one should be at least aware that experts are trying to take a look at this and trying to give the best possible answers in this emergency.
Thank you, Dr. Moore, and I would add that the increased confidence with the process can be measured by the changing votes that we have had in subsequent reviews. We are very comfortable now with the procedure as well as the vaccines that we are approving. Dr. Meisner.
Thank you. I agree with everything that has been said. I would also add the comment, Dr. Monto, that there is no, I think it's important that people do not think that one vaccine is better than other. And this falls under the purview of the CDC, the ACIP, obviously, but hopefully they will emphasize that there is no preference for one vaccine over another, and all vaccines work with what appears to be equal efficacy and equal safety as of this time.
And in this environment, whatever you can get.
Yes.
Is the conclusion. Dr. Levy, final remarks.
Yes, can you hear me?
Yes.
Okay, in addition to the safety and efficacy that we were impressed with as a committee, is the storage at 4 degrees, which is very practical for rural areas in these United States and around the world. This is a global challenge. The single dose, and also not to be forgotten is the experience with the AD26 platform in other vaccine programs, including in children and the potential pediatric indications. So I just wanted to end with those comments. Thank you.
Always good to end with positive comments. I'd like to turn the meeting over to Dr. Atreya for formal closing now. Thank you all. Thanks to the committee and thanks to all the participants. It's been a very smooth, with a few technical glitches and positive meeting. Prala?
Thank you, Dr. Montel.
Into preparing for this meeting. Thank you. I hope you had my final comments. Thank you all for your time and effort that you put into this process and giving us the recommendations. Greatly appreciate it. The meeting is adjourned now.
Thank you. Bye until next time.
Thank you for that introduction. Good day, everyone. For the next 15 minutes, we're going to be talking about pediatric exclusivity. I note that this presentation reflects my views and shouldn't be construed to represent FDA's views or policies. So over the next coming slides, we're going to be discussing the BPCA, the Best Pharmaceuticals for Children Act, which created the pediatric exclusivity construct. We're going to talk about the basics of what it is and how it works. We're going to talk about PPSRs and written requests, the sponsor side and the FDA side of developing pediatric studies. We'll be discussing the requirements for sponsors to fairly respond to an FDA-generated written request if they want exclusivity. And at the end, I'll provide some resources, some challenge questions, and a recap overview of the 10,000-foot level of the process and components of pediatric exclusivity. So, pediatric exclusivity, what is it? Basically, like other exclusivities, it is a financial incentive for sponsors. And in this case, it's a financial incentive for sponsors to study drugs in children. It's appropriate under the statutory construct where information relating to the use of a new drug in the pediatric population may produce health benefits in that population. It is voluntary. Sponsors do not have to undertake the studies that are set forth by the agency as part of the pediatric written request process. This is in contrast to PREA studies under the Pediatric Research Equity Act, which can be required in some circumstances. And importantly, it's 6 months of additional exclusivity that's added to a sponsor's existing patent or exclusivity life. And the statute specifies that it needs to be a listed patent or NCE, 3-year, or orphan to solve a common problem, which is historically Drugs were not often labeled for use in children. Children were often not included in clinical trials, and a lot of approved drugs lacked a pediatric indication